Good morning. I'm Jonathan King. I'm Senior Scientific Advisor in the Division of Behavioral and Social Research at NIA, and this is the Cognitive Aging Summit 4, the fourth one. The very first week that I came to NIA as a program official in 2007 was, in fact, the very first week, the very first meeting of the Cognitive Aging Summit. And since then, there have been 2 additional ones, and this is the 4th. And it's such an august event. We know it's really great because we've moved to Roman numerals, or as a child of my acquaintance has recently called them, Super Bowl numerals. The only other place you see that. In any case, I'd like to introduce the speakers this morning, and first speaker is Dr. Richard Hodis, who's the director of the National Institute on Aging. Dr. Hodis.
Thank you, John. Thank you, everyone, for participating. I'd like to open on a little bit of a personal note. It goes back about 18 years now when I got a phone call from Dr. Lee Dockery. And Lee, I think you're listening, and I'm sorry you're not here, but it's great to be participating with you again. I think, honestly, I've never met a more warm, sincere, and caring person. Even the folks in my office who remember him were asking when they knew this conference was happening. It took only a little longer to learn that in addition to his wonderful warmth and personality, that he was a visionary. And his vision struck home because I think it's very much what all of us embrace at the National Institute on Aging, NIH, all of us included. The notion is that by trying to understand diseases and pathologies, but also in a complementary approach to understand the events that happen normally, with normal aging, for example, we have our best strategy for identifying pathways that are indicative of ways in which we can improve lifespan, increase resilience, and that's the theme here. So even as we spend a lot of time dealing with the pathologies of aging, we spend a lot of time in these sessions, in these summits, inspiring us and informing us over these past decades now to understand what happens in the normal course of aging and when it establishes the greatest resilience and pathways forward for success in aging, and when there are vulnerabilities with Roman numerals. Now takes it a step further and takes the lessons learned that individual differences in the way we age can also provide important clues to the pathways that we need to understand in order to maximize and optimize the course of aging. Individuals, racial ethnic groups, person by person. The approach that we're taking here with technologies that have been developing over these years and decades to allow us to do so give us great promise, hope, and excitement. And so with all of you, I look forward to these next couple of days. Lee, it's great to be with you again and look forward to these contributions you're going to be making. Next, Jonathan.
And next up is Dr. Julie Gerberding from the Foundation for NIH.
Good morning. Thank you. It's really wonderful to be here and live and in color instead of on the little Zoom camera like the last time I interacted with this group. When I was on the red line this morning coming here, I was thinking about cognitive aging, and partly because I'm at an age and stage where that's increasingly relevant, but partially because I was thinking about my family. And I almost have 2 signposts of this process. Just recently, my senior uncle passed away from advanced Parkinson's and dementia. Had a really, really rough clinical course in the last few years of his life. But the other signpost is my mom, who will be celebrating her 91st birthday on next Friday, who lives alone, is a master bridge player, who drives, who goes to every South Dakota State University Jackrabbits football and basketball game, and is really completely intact, with the exception of a cane that she loves to terrorize us with when she disagrees with us. So, just in one family, we have really the spectrum of cognitive aging, the good and the bad part of the story. So I really love what Richard said about trying to understand the determinants and why people do age differently in this respect. The Foundation for NIH has a long tradition of interacting in this space. And I think not only are we proud of the collaborations that we've been able to establish with NIA and the McKnight Foundation, et cetera, we also are leaning into some new areas that I think are relevant to this topic as well. I'll just mention one of them, sound health, and the importance of music and the potential therapeutic impact of music on cognition and hopefully healthy aging. So we look forward to your work. Thank you for all you are contributing to this really increasingly important space, and I hope you enjoy the meeting. Thank you.
Wonderful. And our third speaker today, Dr. Michael Dockery, who's the chair of the Board of Trustees of the McKnight Brain Research Foundation.
Well, thank you all. Thank you, Dr. King and Dr. Hodis and Dr. Gerberding, and thank you also to the organizing committee, which is listed in your brochure. They worked really hard to put together a wonderful program, and I'm excited to see all that's going to be happening. But also a final thank you to our FNIH staff who organized everything, and I think you'll see that it's going to be great. Wanted to just start with a little bit of history. And the McKnight Brain Research Foundation was established in 1999 by Evelyn F. McKnight, who was a retired nurse and the former wife of William L. McKnight. And he was the chairman and CEO of 3M for over 40 years. And for those of you who've heard this story, please indulge me while I share it with the rest. Mr. and Mrs. McKnight were interested in the brain and memory and in aging. And Mr. McKnight would often comment after leaving a social gathering, why is it that I cannot remember their name until 10 minutes after I've left them? And they would drive around Miami Beach looking for land to build a brain institute. But in the end, it was Mrs. McKnight who left her legacy to honor her deceased husband and establish the McKnight Brain Research Foundation. And we were given the challenge that it was to support the research of the brain to accomplish alleviation of memory loss of the aging. Now, there was nothing wrong with their memory, and we've all had these little experiences as well, but we don't really understand how the brain works specifically with memory. How do we form a memory? How do we store it? How do we catalog it, retrieve it, alter it, reuse it? We don't really understand this so well, and there's many steps along the way where this process can break down. And as we've heard in our aging society, we really want to be sure that people can age well into their golden years. While my father, Chair Emeritus Dr. Lee Dockery, and I are two of the original trustees, and there's supposed to be some slides showing with this. Am I supposed to click it? Aha. So that's Mrs. McKnight, sorry. We've now expanded to 8 trustees, corporate trustee, and executive director, which are all listed on the slide here. And we look forward to this summit and to meeting you over these next 2 days. As the foundation is celebrating its 25th anniversary this year, we're also excited to be launching a new public awareness campaign to help people better understand what cognitive aging is and the changes that can be expected with age, as well as some simple lifestyle modifications that may help optimize brain health across the lifespan. We're kicking off our Brain Works, Optimize Your Brain Span campaign. It will start this Friday and leverage the momentum that we're building here over the next few days. So starting Friday, if you'd visit our website, mcknightbrain.org/brainworks. Now, while the McKnights never did build their Brain Institute, in the 25 years that have ensued since we sought to embrace our charge, we knew that this challenge would require collaboration. And therefore, we have established partnerships with 4 separate brain institutes. These are at the University of Alabama at Birmingham, University of Arizona, University of Florida, and the University of Miami. We also have a research partnership in cognitive aging with the NIA through the Foundation for the NIH. And as this slide notes, over $200 million in research specifically targeting cognitive aging, age-related cognitive decline, and memory loss has occurred through direct contributions as well as strategic initiatives with several partnerships. Now, these include the 4 brain institutes, the National Institute on Aging through the FNIH, as well as the American Federation for Aging Research and the American Brain Foundation, to name a few. We've hosted 3 national cognitive aging summits, and today is our 4th. The challenge that we have is to educate the public and the scientific community to differentiate cognitive aging and age-related cognitive decline from the other cognitive and neurodegenerative disorders. Now, you all are the scientists in the forefront of your colleagues, so who better to help us identify the areas of critical need and interest and opportunity on which we should focus our collective efforts? Which items hold the best promise for development? And what are the new cutting-edge concepts that need to be explored and developed? Who is that person within your institute or outside of it with whom you can connect and learn and grow and collaborate together to make something better? So thank you for accepting our challenge and look forward to this meeting.
And now the last speaker of our session, Dr. Madhav Thampasetti, who's the Vice Chair of the Board of Trustees of the McKnight Brain Research Foundation.
Thank you, John. Good morning, everyone. It's a real privilege and a pleasure to be here this morning. Welcome to the 4th Cognitive Aging Summit. I'm here as Vice Chair of the McKnight Brain Research Foundation. In my original day job, I'm a senior investigator at the National Institute on Aging in the intramural program. And so I think I'm the only one in this room who has an official reason to celebrate 2 important anniversaries. The 25th anniversary of the McKnight Brain Research Foundation and the 50th anniversary of the National Institute on Aging. So what I'd like to do today is to take you through where we've come from in terms of the 4 cognitive aging summits. I know some of you are attending this meeting for the very first time. There are some others who've been here for all 4 meetings. So a brief overview of where we began and where we've arrived. So the first Cognitive Aging Summit was in 2007. And John, I didn't know that that's when you came to the NIA. That's when I came to the NIA as well. And so this led to what we think is one of the most important strategic partnerships that the McKnight Brain Research Foundation has established, which is the partnership in cognitive aging between the NIA and the McKnight Brain Research Foundation through the FNIH. Recommendations from this first summit in 2007 led to the establishment of this partnership in 2009 and led to the deployment of about $28 million in federal research funds over 5 years to establish the neural and behavioral profiles of cognitive aging, and was really the impetus to push translational research forward towards pilot intervention studies for age-related cognitive decline. The second summit happened 3 years later, and of all of the important outcomes from this summit, this is my favorite. This is an editorial that Molly and John and Susan Resnick and Peter Rapp, my colleagues, published in the Journals of Gerontology, calling attention to what is at the very heart of the McKnight Brain Research Foundation's mission, which is to understand age-related cognitive impairment as distinct from neuropathologically defined clinical syndromes. This is another impactful article from the same collection of articles in the Journals of Gerontology, and in many ways it foretold the future to where we are today, highlighting the importance of studying individual level differences and raising the potential that an enhanced understanding of individual level differences might one day lead to individualized interventions for age-related cognitive decline. John and Molly have a fantastic knack of writing not just snazzy articles, but also giving them pithy titles. And this was what came out of the 3rd Cognitive Aging Summit, Lost and Found in Translation. And what that particular theme focused on was on cognitive reserve and resilience, highlighting the increasing importance of these concepts towards understanding both age-related as well as pathology-related cognitive change, and highlighted important gaps in our shared understanding of how to characterize terms such as reserve, resilience, and maintenance, warning us that if we aren't speaking the same language, we'll be lost in translation. And one of the key outcomes from that meeting was the Cognitive Resilience Collaboratory, which suggests the overarching term resilience and the definition of 3 key concepts: cognitive reserve, brain maintenance, and brain reserve. One of the most exciting outcomes from the third Cognitive Aging Summit, one of my favorites, is the initiative called STARS, which is Successful Trajectories of Aging, Reserve and Resilience in Rats. This is led by my colleague, Dr. Peter Rapp, in the intramural program of the NIA. And STARS goes to the heart of trying to understand in an experimental paradigm individual differences that might explain both cognitive reserve and resilience, as well as susceptibility to age-related cognitive decline. And what STARS aims to do is to provide an open data bank of longitudinal phenotypic data, neuroimaging scans, tissues, and other biospecimens for the wider scientific community, and was identified as a direct response to a need Cognitive Aging Summit in 2017. These are pictures from the installation of the magnet for the STARS initiative. Peter Rapp declined to send a picture because he didn't want his youthful good looks to be a distraction at this meeting. But for those of you who will attend the interinstitutional meeting at the University of Florida, I'm delighted that Peter's going to be giving one of the keynote talks at this meeting, where I'm sure you'll learn a lot about this initiative that we're very excited about. In fact, I was privileged just a couple of days back to look at the first fMRI scans from the STARS initiative. And so I think this is going to be a terrific resource for the research community at large. Which brings us to the theme for this fourth Cognitive Aging Summit, which is a focus on individual differences and the development of personalized and precision approaches to the remediation of age-related cognitive decline. The sessions are grouped into thematic groups, focusing on environments from both systemic as well as all the way to clinical trials. What we hope is that deliberations and discussions from these themes will eventually lead to cross-cutting and interdisciplinary work that might bring us closer to interventions for age-related cognitive impairment and cognitive aging. I'd like to remind myself that a focus on the individual is not a reductionist approach. We hope that this will bring us closer to understanding universal problems. And I'm reminded of one of my favorite quotes from Carl Rogers, saying that what is most personal is most universal. So thank you, everyone. Welcome to the fourth Cognitive Aging Summit, and I hope you have a terrific meeting. Thank you very much.
Well, thank you very much. This was just the first session of a very, very full program. So I'm going to take the opportunity right here to remind speakers and guests that we are going to maintain a completely on-time performance. So although we're going to get a few extra minutes here because of the remarks being very pithy, during the scientific sessions, all the speakers know what their time limit is. I would just like to point out that they can see it right here, and I can see it from there. And I'm not a shy person, and but seriously, there is going to be a robust period of time to discuss all of things in the general session. So now, having been very efficient for the introductory session, we have a break now until 8:45. See you then.
Good morning, everyone. If I could have the rest of you start making your way towards your seats, we'll actually get started with the first session of really exciting science. So again, welcome. Good morning. I'll be chairing the first session. My name is Saul Valeda. I'm an associate professor at the University of California in San Francisco, and I will be moderating. Just as a reminder, we're going to have 4 speakers this morning. The questions will follow all 4 talks, and we're going to have an open discussion at the end, and I'll also open it for questions from everyone. So please write down your favorite question, have it handy, because I am going to come back to you at the end of the session. And really, really broadly, I just want to point out, we're going to start the day really with this idea of, precision medicine, individual trajectories, and brain aging, and we're going to start outside of the brain very early in the morning. So really focusing on targeting the periphery as an alternative to delay and potentially even reverse cognitive aging. And with that, I'll start with the speakers, and I'll introduce each one as they come. So first, let's see. Do I do this? Yes, perfect. Look at that. I'd like to introduce Dr. Tara Walker, who's coming to us from the University of Queensland in Australia. I really just mad props on being able to come up here with the jet lag. I'm really looking forward to your talk. So please help me in welcoming you, and I'm really excited to hear.
Thanks, Sol. I'm really happy to be here. Okay, the brain has a remarkable ability to maintain plasticity throughout life. And one way that the brain can do this is via the continuous addition of new neurons throughout life in a process called adult neurogenesis. So adult neurogenesis occurs in the brain in a number of niches, and I'm interested in studying one of these, the hippocampus, a particular area of the brain that's important for many learning and memory functions. So neurogenesis in the brain significantly decreases with age, and also in a number of neurodegenerative conditions. And this is associated with reduction or loss in a number of cognitive functions. The good news, however, is that this reduction in neurogenesis can be reversed or declined by a number of external factors. And one of the strongest and physiological ways that we can increase neurogenesis is via physical exercise. So it was discovered almost more than 20 years ago, actually, now, that when mice are given free access to a running wheel, they love to run, and this leads to a significant increase in the number of proliferating neural precursor cells, an increase in neurogenesis, an increase in synaptic plasticity, and a concomitant increase in spatial learning and memory function. However, in the intervening 20 years, despite significant amounts of research, we still do not really understand the mechanisms by which exercise increases neurogenesis. So the major focus of my lab over the last years has been really to understand the underlying mechanisms by which exercise can boost neuron generation in the brain, and to then use this information to be able to identify novel strategies to reverse the neuronal loss that's associated with aging and neurodegeneration. So many years ago now, in a very elegant study, Saul Valida and colleagues showed that blood-borne factors are able to regulate neurogenesis. They showed, using a heterochronic parabiosis model, whereby the circulatory systems of young and old mice were attached, that there are factors factors circulating in the blood that are able to mediate both neurogenesis, synaptic plasticity, and cognitive abilities. So we hypothesized, given the sort of systemic nature of the exercise stimulus, that perhaps a similar thing is occurring, that there are factors released into the blood following an exercise that are able to mediate neurogenesis. So to test this hypothesis, we performed this very simple experiment where we took the blood serum from either standard housed or exercising mice and cultured this together with primary hippocampal cells in an assay called the neurosphere assay. And we could show that the serum from the running mice led to a significant increase in the number of neurospheres generated compared to the control serum or untreated cells, indicating that indeed there are factors released into the blood following exercise that are able to stimulate neural precursor proliferation. So, of course, the next obvious question was, what are these factors? So to identify potential exokines, we performed a proteomic screen, again on the blood of standard or running mice, and identified a number of candidate proteins that were significantly upregulated in the blood following exercise. A pathway analysis revealed that platelet activation may be a significant factor underlying this response. And we identified one factor in particular called platelet factor 4, significantly upregulated in the blood following exercise. So just a little bit about platelets. So platelets are the small blood cells, anucleate cells that are found in the blood that are typically known for their role in wound healing. However, platelets are now being shown to have many other alternative functions beyond their role in wound healing. And they also are associated, their dysfunction is also associated with many types of neurodegenerative disorders. What's exciting about platelets is, however, that they've also been shown to contain a wide plethora of bioactive molecules. So there's over 1,000 bioactive molecules that are contained within platelets, and these have been shown to have recently to have exciting new applications in regenerative therapy. So we first wanted to determine whether platelets are actually activated by exercise. So to do this, we used a flow cytometric analysis of the blood isolated from exercising and standard house mice. And we could show that as little as 1 day of exercise was enough to activate platelets. To determine whether platelets are actually required to mediate the neurogenic response to exercise, we performed an experiment where we depleted platelets by inducing thrombocytopenia in mice using antiplatelet serum. And we did this during the exercise period. As you can see here, as expected, the control mice had the expected increase in neurogenesis following exercise. But excitingly, you could see that the platelet-depleted mice, this response was lost, indicating that indeed platelets are required for the exercise-mediated response of neurogenesis. So as I mentioned, platelets are activated by exercise, and they release a number of factors into the blood following this response. And one of the major factors that was released that we identified in our proteomic screen was platelet factor 4. We confirmed this result using an ELISA-based screen as well. So we wanted to determine whether PF4 alone could mediate the, or could mimic the response to exercise that we see in neurogenesis. So to do this, we first tested this in vitro by culturing, again, isolated cells from the hippocampus in the neurosphere assay, together with platelet factor 4. And as you can see here, compared to the controls, which is the dotted line, platelet factor 4 led to a significant increase in the number of neurospheres generated. It also led to an increase in the neuronal lineage differentiation potential of these neurospheres, indicating that indeed the neural precursor cells respond to the platelet factor 4 by increasing their proliferation and their neuronal lineage differentiation potential. So to test the effects of PF4 in vivo, we infused platelet factor 4 into the brains of mice using mini-osmotic pumps. This led to a significant increase in the number of newborn neurons generated in the hippocampus of the PF4-treated mice. Conversely, we could show, using PF4 knockout, that in the absence of PF4, there was a significant decrease in the neurogenesis levels, in both the levels of proliferating precursor cells and the levels of newborn neurons. Interestingly, we also found that in the absence of PF4, the mice no longer had the pro-neurogenic response to exercise, indicating that PF4 is indeed required for this exercise-mediated neurogenic response. So as I mentioned before, the adult neurogenesis significantly decreases with age. So we wanted to see whether PF4 treatment could rescue this neurogenic loss during aging. So to do this, we took 20-month-old mice and treated them systemically by a tail vein injection with platelet factor 4. As you can see, PF4 treatment led to a significant increase in the number of proliferating precursor cells and immature neurons. PF4 treatment also led to an enhancement of the dendritic complexity of these newborn neurons, as evidenced by an increase in the dendritic length. Conversely, we saw in the PF4 knockout mice that this was the dendritic length was decreased. So as I mentioned, adult neurogenesis declines with age, and this also leads to a concomitant decline in specific types of spatial learning and memory function. So to determine whether we could reverse this cognitive decline that's associated with aging, we again treated aged mice with PF4 systemically and tested them using a number of spatial learning and memory tasks. Firstly, we used this task called the active place avoidance test, which is basically a spatial learning and memory task in which the mice are placed on a rotating platform and have to use spatial cues throughout the room in order to avoid this shock zone. And basically, we could show that the mice that received the PF4 had significantly enhanced learning ability in this task, as evidenced by a significant decrease in the number of shocks that they received. We confirmed this result in a separate cohort of mice and a number of other spatial learning and memory tasks. As you can see here, we again treated the aged mice with PF4 and tested them in 2 alternative tasks, the novel object location task and the contextual fear conditioning task. As you can see, in the novel object location task, the mice spent significantly more time exploring the novel object, and in the contextual fear conditioning task, the mice spent significantly more time freezing, indicating that PF4 enhances spatial learning and memory function during aging. So finally, we wanted to determine whether this cognitive enhancing effect of PF4 was actually dependent on the PF4-induced increase in neurogenesis. So to test this, we used a transgenic mouse model called the double-cortin diphtheria toxin receptor model. So these mice basically have the diphtheria toxin receptor under the control of the newborn neuron promoter double-cortin. So when we treat these mice with diphtheria toxin, it ablates all the newborn neurons, effectively ablating neurogenesis in these mice. So we used these mice and treated them with PF4, and in the last 10 days of the PF4 treatment, treated them with the diphtheria toxin to ablate neurogenesis. And as you can see here, as expected, the PF4 only treated mice show a significant improvement in their spatial learning and memory function. But this is lost in the mice that were treated with PF4 and diphtheria toxin, in which the double-cortin cells are ablated, indicating that indeed this ongoing neurogenesis is required for the PF4-induced increase in cognition. So overall, our research has shown that platelets are required for the exercise-mediated response, neurogenic response, and that one factor alone, platelet factor 4, can be used to rejuvenate hippocampal neurogenesis and restore cognitive function in aged mice. So excitingly and sort of serendipitously, at the same time that we performed our study, 2 other groups, led by Saul Valida and Dena Dubal, who are both here today, came to the same identification of PF4 as a factor that's involved in rejuvenation in aging. And we were actually very fortunate to be able to publish our research alongside each other at the same time in 3 separate Nature Press journals. So where to from here? Our research is now, we are looking at, given that the exciting findings that we found in the model of physiological, we're now taking this into mouse models of Alzheimer's disease to see if we could rescue some of the cognitive and histopathological hallmarks of Alzheimer's disease. So this is just a little teaser. We're very early in the stages of this research, but basically in the same sort of study, we've taken a transgenic Alzheimer's mouse model and treated these mice with platelet factor 4. And as you can see here, as expected, this is a Y-maze, spatial learning and memory task. As you can see here, the transgenic mice treated with saline have a deficit in this task, and this could be rescued by platelet factor 4. So watch this space for further data on our effects of platelet factor 4 in the AD models. And with that, I'd just like to conclude by thanking the members of my lab. Most of this work was performed by my postdoc, Dr. Odette Leiter, who performed all the aging PF4 studies. My collaborators, particularly Saul Valida, who have been working alongside for the last few years on these PF4 studies, other collaborators, and our funding agencies, including the NIH. So thank you very much.
Great, perfect. Okay, so we're now going to be moving from sort of circulating blood factors more into the borders. So what's at the interface between the blood and the brain? And for that, we have Dr. Andrew Yang, who's a newly minted assistant professor at the Gladstone Institute and the University of California, San Francisco, who will be sharing with us some work on the BBB.
All right, thank you, Saul. I'm excited to be here. I'm an engineer turned neuroscientist, so I thought I'd talk about how some recent technological advances are expanding our understanding of the brain and its relationship with the body. And in particular, how these recent advances are showing an unexpected richness of molecules trafficking from the blood to the brain. And I think understanding what these molecules are, what they do, and how they might differ on an individual basis across lifespan, it may expand our understanding of peripheral regulators of brain health and nominate a whole new set of potential therapeutic targets to promote healthy brain aging. Okay, so in short, we want to understand how does the body speak to the brain across life. But where to begin? We can start with the simple fact that throughout your body, wherever there are nerves, there are blood vessels there to support them. This has been reported since the 16th century from Andreas Vesalius, and nowhere is this more true than in the brain, especially the human brain with its billions of neurons and many more connections dynamically change throughout life, and that make us each who we are. What's fascinating is to support this, the brain's vasculature has adopted these unique molecular properties. One of them, what we call the blood-brain barrier, right? And this was discovered now over a century ago, in 1890s, from Paul Ehrlich and Goldman and Reese and Chromosome and others, who found that if they took a dye, in this case, right here, a textile dye that stains your clothes blue, and injected it into model organisms, that dye would spread throughout the body, but not the brain. Right? So this is our first inkling that there's something special going on in the brain's vasculature. And this has really been the paradigm the field has now used to establish that there is a blood-brain barrier. The way I think of it is the blood-brain barrier forms a special home for the brain. And of course, the walls of your home are incredibly important. It's insulating you from the environment, right? But I think we expect more than that from our homes. We expect there to be windows to let the sunlight in, doors to let the groceries and your friends in to clear the waste out. A thermostat to keep everybody happy, homeostatic temperature. And then nowadays, in the modern home, we couldn't live without Wi-Fi or cellular to keep us in touch with the outside world. Right? And I also think maybe more provocatively, if your home is getting old or if it's been damaged, you don't just fix up the interior of the home. You want to fix the home itself. And I think the same principles may hold true for our blood-brain barrier. So the theme of our lab is understanding these, what we believe are built-in pathways of how the body may be communicating to the brain all the time throughout life. Right? So one of them will be, how do soluble blood proteins enter the brain? What are their neurological impacts, and how can this help us inform new drug delivery strategies across the blood-brain barrier? We're also very interested in our lab on the reverse direction. How do brain antigens get sensed by the peripheral immune system so that the immune system can mobilize and promote brain health, or in certain cases with aging, adverse new inflammation. But today, I just wanted to give a quick proof of concept on one of these avenues. We're trying to build a catalog and kind of an understanding of what are all the proteins in the brain, what are the rules that govern their entry, and then what do they do, right? As I mentioned, the way the field has approached this has been to take one of these dyes and inject them one by one. So here's a recent review from Norm Saunders, kind of summarizing how the field approaches the blood-brain barrier, right? We take these dyes, including Evans blue or a plant protein, and we inject them, and then we ask this binary question, is there a leakage or not? And these have been very important studies, don't get me wrong, right? From these, we've learned hallmark properties of the blood-brain barrier, including these special tight junctions that prevent just free diffusion, right? It's also nominated kind of other hallmarks, at least for proteins. You see number 3 here, low vesicular transport. And this is really the hallmark that we might want to challenge with new technologies that are coming online. And what does that look like? Well, I think it starts with how can we see more, right? Rather than just injecting one thing at a time and then asking a yes-no, we thought the blood-brain barrier has evolved over millions of years alongside a special partner, the blood itself. And the blood contains thousands of proteins. And so with new proteomics and chemical biology techniques, if we could just tag and track and visualize the movement of these thousands of proteins, we might see something new. Because after all, your blood-brain barrier is exposed to a new wave of blood proteins every heartbeat. And so we figured out ways to do this. We can tag them with radioisotopes, fluorophores, and as I'll show, new bioorthogonal chemistry handles, and then transfer them into young, aged, and diseased mice and understand this more comprehensive aspect of blood-brain barrier biology. And so when we do this with fluorophores, the first thing that struck us was instead of an inert passive wall, we saw plenty of uptake of these plasma proteins into the brain's vasculature. And so on the left here is an image of plasma in white along a kind of an arterial down to a capillary tree in the brain. And we can see these characteristic puncta that look like vesicles that mediate transport. In the hippocampus on the right, in green now, you can see neurons that are soaking up all of these plasma proteins, right? So perhaps the blood-brain barrier is less of a barrier and more of a sponge. And we went on to then show how this works. We showed that this works in a really exquisitely sensitive way through receptors, a diverse array of receptors on the brain's vasculature that then shuttle through clathrin-coated pits to then enter neurons. And this is happening constantly. So there's a code here, right? Because prior tracers, prior dyes don't have an evolved receptor on the blood-brain barrier, whereas the plasma proteins in your blood do. And so how do we learn this code, right? What are these proteins? What are their receptors? How does this work? And so what we've been working on recently is developing these new techniques to be able to do so. And this is in collaboration with Al Burlingame, kind of a mass spectrometry expert at UCSF. And the idea here is, well, let's tag all our plasma proteins instead of with a fluorophore, but with one of these new click chemistries, moieties, right? They're very small, they're inert, they don't react with anything else in the body when you inject them. Then wait for the permeable proteins to enter the brain, okay? And then what you can do is you can harvest the brain, you perfuse, get rid of all the blood, and you can do different cell isolation. So you can isolate just the vasculature or just the neurons. And then what you can do is take those cells and incubate them with beads that have a partner click group, right? And so in particular, you can tag all your plasma proteins with one click group called the transcycloroctine. You have your bead that has a partner tetrazine. And what happens is they won't react with anybody else. It's kind of like when you go into a bar and you're looking for your friend. You're not going to just talk to anyone else except your friend. You're going to race straight to them. And this is how this chemistry works. And so that you can fish out those proteins that are permeable, that are entering the brain from the blood. And we can identify what those are using mass spectrometry or antibody arrays. And so this is the particular chemistry after a whole series of optimization that took over 2 years. We figured out conditions to make this work. And just to show you why this was fruitful, you kind of see this blot on the left. And a lot of folks just say, why can't you just tag your proteins with biotin and just do a streptavidin pull down? Well, it turns out the chemistry matters, right? And you can see so much more sensitivity, and you can build such a better map of proteins that are entering the brain using these new technologies. Just to show another proof of concept that we can tag hundreds of proteins in your blood plasma across different classes, they come from different tissues, so that we feel like we are ready to go in understanding what are those proteins that are entering the brain. So first, I mentioned there's this kind of unexpected degree of uptake along our brain's vasculature, how the vasculature may be similar to a sponge. And so when we do this kind of mass spectrometry analysis, I apologize that some of the dots are missing here. What we can see is there are hundreds of proteins, in this case, I guess 387 proteins, that are being taken up by the brain's vasculature all the time, right? And so it suggests that not so much as a barrier is constantly nibbling along with your plasma and sensing what's going on in the blood. Based on our fluorescent imaging, we had shown that these unknown circulating proteins are entering the brain. You can co-stain them for the one protein that a lot of companies are using and that gets in the brain, transferrin. It's the basis of many of these drug delivery shuttles. And what we can find is many neurons contain unknown circulating factors beyond transferrin. And indeed, with the mass spectrometry results, if we now look at the neuronal fraction, we can see almost 200 other proteins that are entering the healthy brain, right? And so I think a fascinating question is, what do they do, right? What are these proteins? Why does our brain need these proteins? And so we've begun doing preliminary analysis, and it seems like there are certain classes of blood proteins that the brain wants. These include carrier proteins. If you think about it, transferrin delivers iron, it carries iron. But many others, like ApoA1, delivers HDL, or good cholesterol. Vitamin D binding protein delivers vitamin D. There seems to also be an enrichment for protease inhibitors that might change enzymatic activity of the brain, and then, of course, growth factors. These proteins come, as you'd expect, mostly from the liver, and the heart, and then the bone marrow. And of course, with every new technology, you want to check, well, is this really working, right? And so I just want to give a couple examples where we've validated that this may be true. So one way to go about this is to say, all right, if we see a protein in our catalog of BB permeable proteins, it's in there, but it's not expressed at the RNA level by any brain cell type, right? If it's protein's there, but the RNA is not there, it likely entered the brain. And so we're going to show this for two examples, ApoA1, which delivers HDL, and then hemopexin, which scavenges heme. Neither are expressed in any brain cell type, and yet when you tag them individually, recombinant proteins with the fluorophore, and inject them into the bloodstream, you can see uptake of these proteins into the brain's vasculature, as well as parenchyma, where the neurons are, at comparable or greater levels than what we currently know with transferrin. You can confirm this with kind of immunostaining. In green, you can see the vasculature is soaked up with these proteins, and you can even see along these neurons in the hippocampus uptake of them. So what do? That remains kind of ongoing work. But I think what's fascinating, what we can say now is that there are clear classes of proteins that are enriched. So there's a meaning to this, right? I mentioned how transferrin can deliver iron. It seems like this is not just one outlier. It seems like it's true that the brain requires many other carrier proteins to deliver the nutrients that it needs. What's really fascinating for us has been prior work showing that knockout of these different proteins has neurological consequences in aged and diseased mice. And that's something that we're following up on now. And finally, though this is still early days, we're very interested in understanding, well, what happens in aging? And that's because in our prior work, we found that there are dramatic changes in their brain's barriers with normal aging, disease-free normal aging. For one, there's a shift from this exquisitely ligand-specific receptor-mediated transport of the right protein entering the right cell in the brain to this more chaotic free-for-all, right? Just kind of more caveolar-mediated transcytosis, where if a protein kind of deforms the membrane the right way, it'll kind of leak through, right? And so we think this is an example of this kind of age-related leakiness. So what these micrographs are showing is, you can see on the top left, plenty of this kind of clathrin vesicles in the young brain vasculature. It diminishes with age. But this kind of leaky caveoli that's actively suppressed in the young vasculature now gets turned back on. And it looks like the brain's vasculature loses its specialization. It looks much more like the peripheral endothelium that you'd see throughout your body. And we can quantify these. And so what does this mean then for the actual composition of proteins that are entering your brain? What we can say now is there seems to be kind of proteins that are more inflammatory. For example, all these different immunoglobulins or that can enter the brain with age, but then a loss of the proteins that we think might be necessary for healthy brain homeostasis. For example, these carrier proteins like transferrin and others, growth factors. We can begin to validate some of this. For example, you can stain for immunoglobulins quite easily and see this increased uptake in the aging brain. Right? And so, I think there's a couple takeaways, but one is kind of pushing the dogma a little bit, right? So it's not just impermeable versus leaky. I think it's a little bit more nuanced than that. I think the brain's vasculature is a really sophisticated, active sponge that's soaking up the right proteins that the brain needs, constantly monitoring what's going on in your blood. And that could be very sensitive to individual lifestyle differences. But with aging, this process becomes dysregulated, and the brain no longer is getting what it needs, and it could be faced with exposure to toxins and pathogens that should not belong. And so, just to summarize, the brain's vasculature is active, it's alive, it's constantly sampling what's going on. We think there are hundreds of proteins that are entering your healthy brain all the time, fall into defined classes. We're beginning to understand their neurological functions. This composition of proteins shifts with age alongside changes in the blood-brain barrier. And I think identifying patterns in our catalog of proteins, for example, It sounds crazy, but is there a molecular motif, right? Just as we know that there's amino acid sequence that tells proteins where to go within a cell, might we find similar patterns that tell proteins to go to different organs, especially the brain, and can that be harnessed for drug delivery avenues? And then, of course, understanding what these proteins do to neurons, the glia, throughout life, I think will be fascinating because it expands the target space. We might not need to have a therapy enter the brain for us to be able to modulate brain health. And so I think, in short, learning the rules of engagement, how the body speaks to the brain, I think will be a really important avenue and open new opportunities to understand and ultimately treat and promote healthy brain aging. And with that, I want to thank the lab. As Saul mentioned, it's been kind of an exhilarating start in our lab at UCSF, and I'm really grateful for our many collaborators, including Saul and others. Thank you for your time, and I'd love to take questions in the discussion.
Awesome. It's always super exciting. All right. Can we go to the next one? Sorry. There we go. Perfect. Okay, here we're going. Great. Okay, so you see kind of the trip that we're taking. We're doing blood. We went to the barriers. But of course, we can't think about that interface between the periphery and the brain without going into other players, such as hormones and the role of metabolism as well. So we're going to start learning a little bit more in that direction. For that, I'll ask Dr. Emily Jacobs from the University of California, Santa Barbara, to talk to us a little bit about that.
Thank you, Saul, for having me. Thank you, Molly, for putting this panel together. So I'm going to be talking about dynamic endocrine modulation of the nervous system, but before I dig into the science, I want to take a step back and I want to consider the status of women's health, because arguably we're 20 years behind where we should be. Most of what we know about health and disease centers on the male body. There's a long history to that, which I could give an entire lecture. I have given entire lectures on, but not today. Take my word for it. And yet 70% of Alzheimer's disease cases are women. 2 out of every 3 people who suffer from depression are women. There are some conditions that only people with a uterus will experience, like postpartum blues or perimenopausal brain fog. And these deserve our attention. And we're not giving it our attention. Women are half of the global population, but only 10% of the NIH budget goes to conditions that primarily affect women. So it is increasingly clear to actually many of us in this room that elevating women's health research is going to require a global shift in science culture. And that shift is starting. I had the distinct honor of joining the President and First Lady and Vice President Monday in the White House, where President Biden signed an executive order pushing for $12 billion in new funding to transform women's health research. This could not come soon enough. The World Economic Forum released a report in January stating that eliminating the gender data gap will unleash $1 trillion in a return on investment. That's $1 trillion annually. Suffice it to say, we can't afford to ignore women's health any longer. Or as Malala Yousafzai said, we cannot succeed when half of us are left behind. We have a lot of work to do, and to put real numbers on that problem, I want to cull from an example that hits close to home. This comes from my own world of human brain imaging. Technology first entered the mainstream in the mid to early 90s. It gave neuroscientists unprecedented access to the living human brain. And since that time, there's been over 50,000 articles that have been published using brain imaging technologies represented by this blue wave. And then if you squint really hard, you can see this sort of thin black line at the bottom bumping around the x-axis. You have to pull out a microscope. That's everything we know about how the brain responds to pregnancy, menopause, the menstrual cycle, hormonal contraception, and so much more. So altogether, of those 50,000 articles, less than half of 1% focus on health factors specific to women. Or to put it another way, neuroscience has overlooked aspects of the human condition that affect half of the world's population, and we have got to correct course for the field to advance. So when we ask about the scientific body of knowledge, I think we need to consider whose body does it serve. And here's the thing, these topics are unknown, but they're knowable. And in the next set of slides, I want to convince you that by overlooking these major life events, these major factors, we are almost certainly missing critical clues that can tell us about brain health over the life course. I'm going to use the lens of precision imaging, which is what my lab has really gotten into in the last several years. And one thing I want you to keep in mind sort of as this field comes into focus is that one thing that we're seeing in both animal models and human models is that the tissues and circuits that are most highly regulated by steroid hormones and modulated across these life events, they overlap with circuits that are dysregulated in both depression and in Alzheimer's disease. So here I'm thinking of tissues within the medial temporal lobe or at a distributed level, things like the default mode network. So a question that's really driving my lab is, what are the physiological properties that make these hubs so prone to damage? I would be remiss not to throw this slide in. This is from a beautiful work from Catherine Woolley from the 90s, and she really sort of shined the spotlight on steroid hormones, not as these regulators of hypothalamic function, which the field was sort of at the time, really focused on its effect on overtly reproductive behaviors like lordosis or mounting. She showed that these are potent neuromodulators within the hippocampus. This is a pyramidal cell in the CA1 region of the hippocampus. She zoomed in to look at apical dendrites and looking at dendritic spines by estrous cycle stage in female rats. And what you can see, it's so beautiful, you don't need statistics, is that during proestrus, this is the day of ovulation when estradiol level spikes, you get this massive proliferation of spines. And then 24 hours later during estrus, when estrogen comes down, you see a retraction of this effect. This was beautiful work, field-defining work, although people did not believe her at the time. She said she was physically and intellectually assaulted, her words. The field were not ready for this I think, kind of science. But one limitation of this study, because of the limitations of the technology at the time, is that it's all cross-sectional, right? So these are camera-lucid drawings. This is based on histology. So one question that I had is, can we use today's technology to see these same pulses in the brain? All right, we have these abilities to peer into the awake behaving animal now at subcellular resolution. So I teamed up with the Gord lab at UCSB to use 2-photon microscopy. Here, Mike's created this little microprism that gets implanted in the mouse brain. It's kind of like a mini periscope flipped upside down, so you can see this beautiful hippocampal circuit at cellular resolution in awake behaving animals. And so in experiments led by Nora Wolcott, she's taking female mice, she's imaging them with 2P every 12 hours for 8 days, which corresponds to 2 complete estrus cycles. She's doing vaginal cytology, so she's staging the animals. There's a really cool machine learning estrus that she put together to try to encourage others in the field to do this. It's all on GitHub, it's open access. What does she find? Sure enough, these endocrine rhythms are driving neural rhythms in the brain. So again, these are from CA1 neurons, and you can see there are these pulses, right, that spike at proestrus and then dip in estrus, and you can see it again. What's beautiful about this technique is that they're looking at the exact same segment of the dendrite every time. So you can see these spines really like ebb and flow in real time. You can categorize what spine types they are. So, 30 years later, it's just sort of justification that Catherine was way ahead of the curve, way ahead of all of us. These rhythms are absolutely happening. Now comes the critical question: why? Why is the brain organized in this way? Why are these rhythms happening? And I don't have time to sort of get into the nuance of this, but I will just tell you, and the preprint will be up soon, we can look at response properties of specialized cell types within the CA1, like place fields. And we know that during proestrus, so I don't have a pointer, but during proestrus, there's something really unique that happens to these cell responses, which these place fields become both more stable. So when you put the animal in environment A or track A and you get these beautiful place cells, these are just aligned by their response properties on this circular track in blue. Another sort of initiation into that track A at a later time, those responses become really high fidelity. the cells also become more flexible. So it's sort of two sides of a different coin. So clearly these morphological changes driven by hormonal regulation are having an effect on the functional properties of these neurons. And in experiments that I'm not going to show here, we're using CRISPR to downregulate certain receptor subtypes to really try to abolish this effect and try to understand mechanism a little bit more. Okay. Now for my bread and butter. What about the human brain? Can we see these endocrine pulses driving changes in the human brain over rapid timescales? This is a study that wouldn't have happened without the pandemic. Our bread and butter of pulling in sort of people from the community couldn't happen. The BIC was shut down, but we could scan ourselves. So Laura Pritchett volunteered to put herself in the scanner. We collected MRI, we did venipuncture every 24 hours for 30 days across the complete menstrual cycle. She did it all again a year later. Why did we pull this off? Because most, sort of a critical feature of the mammalian endocrine system is that there are these ebbs and flows of hormones over time for most women, for most of our lives. These are metronomic, and they drive physiological properties throughout the body, but nobody really knew how they shaped the brain. At best, we had evidence from cross-sectional studies or maybe 2 time point studies, but that really doesn't get at this dynamic change in hormone secretion. So that's what we did. What did we find? So you can see this peak in estradiol right around ovulation in teal. In blue is the progesterone wave that comes in the luteal phase. These are on different axes, otherwise that progesterone would be sort of through the roof. And the first observation that we made was that using sort of time-lagged analyses, that there's real strong evidence that these increases in estradiol over time are driving increases in functional connectivity pretty much across the cortical mantle, with some networks maybe more responsive than others. We turned to graph theory to start to look at sort of the network properties, and one really popped out, which is the default mode network. So at the bottom, these are Laura's hormone levels by day of experiment. And you could see during that peak in estrogen, right around the time of ovulation, it is immediately followed by this increase in global efficiency within the default mode. When estradiol levels plummet, so too does this metric. And this was replicated when she did the experiment again a year later. Okay, at this point, we're sort of knee-deep in the pandemic. We're just staring at this data, and we decided to team up with a group in physics. This is Jean Carlson's lab in work led by her student Josh Mueller and Laura. And we wanted to, so Jean uses complex systems analysis. So Josh was looking at something called dynamic community detection, and he shows that there's this transient reorganization of functional brain networks across the female menstrual cycle. He's looking at these metrics of sort of network flexibility. And in the top row, what you can see, we're just grouping this by kind of gross stage of the menstrual cycle. And there's something different that happens at ovulation, right? These networks become more flexible. You can see this as a time series on the bottom. So again, what I'm highlighting in that bar are these spikes in estradiol around ovulation, and there's this concomitant sort of increase in flexibility in these functional brain networks, and even a secondary peak in the luteal phase when you get that secondary bump in estradiol. This is evident for some networks. Some are pretty stable, like the visual network. So it's not a whole brain phenomenon. There might be some selectivity there. I do want to make a really important point. These hormonal rhythms are not exclusive to women. That is a pernicious myth that has been used to exclude women and female animals from 50 years of biomedical research. That's not true. Men also have these hormonal rhythms. So we followed this up more for political reasons than anything else to do 28andHe. This is a precision imaging study of a densely sampled male who underwent the same brain scans and blood draws every 12 to 24 hours for 40 sessions. And here we're targeting that diurnal cycle. So testosterone, cortisol, estradiol all peak in the morning and dip in the evening. And long story short, we can see the same associations where increases in estradiol and testosterone over time are associated with whole brain functional coherence, in this case in a densely sampled male. And the magnitude of these effects are very similar between the male and the female. If anything, it's just that the periodicity of these effects are different, right? So it's sort of like the accordion is being stretched out, where both males and females have this diurnal rhythm, but females also have this kind of circulunar rhythm. What about brain morphology? Can we actually see an ebb and flow of tissue volume over time? So for this, we zoomed in using a high-resolution sort of T2 imaging and segmentation of the hippocampal subfields and the surrounding cortex. And this is work led by Caitlin Taylor, and what she observed is that in this luteal rise in progesterone in blue, there's this expansion of tissue within the medial temporal lobe. Here I'm just showing two regions, so hippocampal CA2-3 and the parahippocampal cortex. When the participant completed the experiment again a year later, there was one wrinkle to that experiment that I didn't mention. She was on a drug that selectively suppressed progesterone. So notice that that blue wave is absent in the second experiment. And when we removed the effect of progesterone, we could abolish that neuronal rhythm. Okay, what about other major neuroendocrine transitions? We know that the endocrine changes that occur across the menstrual cycle are dwarfed compared to the endocrine changes that observe across pregnancy. This is work done in collaboration with Liz Krassel, who's in the audience. We decided to do a precision imaging study of the maternal brain, and it's wild to me that in the year 2024, we've never developed a map of the brain across pregnancy, but we have now. This is coming out soon. What you can see is that there's this wave of hormone production that occurs across the gestational period and drops precipitously at parturition. In C, you can see just the time course of these scans. We have preconception scans, scans during gestation that occurred roughly every 2 weeks across the complete gestational window, and then scans going up to 2 years postpartum. What did we find? There are these sweeping changes in gray matter volume, cortical thickness, right? That decrease across the gestational window. This is inversely paralleled by this expansion within CSF and lateral ventricles, kind of filling up that cavity, which makes sense. And then there are these really interesting nonlinear changes in quantitative anisotropy, these measures of white matter that seem to peak in the second to third trimester before returning to baseline. In the 4 or 5 studies that have looked at the brain pre and post conception, doing sort of 2 time point analysis, which have told us really a lot. This is work led by Susana Carmona, Magdalena Martinez Garcia, and others in Europe. One thing that's missing, and I think that this precision imaging studies really sort of bring to bear, is that there's all kinds of dynamics that are happening during pregnancy itself that we miss when we just look before and after this a major event. In A, you can see the sort of spatial distribution of these gray matter volume changes. It's pretty ubiquitous, but there's some regions that certainly buck the global trend, and I think that's going to be a thread that we'll want to pull on. In C, we just give some examples looking at sort of specific at the regional level. These gray matter volumes, for example, in the first case in precuneus or posterior cingulate, sort of nodes within the default mode network. You can see these gray matter volume changes unfold week after week. Next to it in the box plots, you can see this grouped by stage, so preconception in pink, and then first, second, and third trimester, just this like staircase decline or decrease. And then in the postpartum period, note that there's no rebound, right? These are almost like permanent etchings into the brain. So these are persisting at least 2 years postpartum. We see the same thing in subcortical structures. So we're really starting to think of matrescence as this extended period of neuroplasticity in the adult brain that could certainly give us clues about how the brain is aging later in life. We know from Lisa Galia's work that previous parity can keep the brain responsive to estrogens, including increased neurogenesis and reduced inflammatory cytokines in middle-aged Paris rats relative to nulliparous mats. And in the 42 seconds I have left, I want to just say a word about the our goal is to take women's brain health prime time. We are driven by a radically simple idea, which is that progress in neuroscience will flourish when the health of men and women are valued equally. We want to, I think, are fueled by a commitment to make neuroscience inclusive, both in terms of who gets to ask the questions and who is served by the answers. So we are integrating data collection across the University of California system to do that. We do 3 things. We generate the missing data, we build the infrastructure to share that data, and we develop AI tools to answer far-reaching questions about women's brain health. We have 3 funded centers under the umbrella of the Institute, and one ethos of the Institute is that once we collect the data, we give it away. It's all open access, so the discoveries never stop. And with that, I will thank the brilliant and passionate scientists who actually do this hard work. I just get the pleasure of being able to speak about it. So, thank you.
Awesome. That was fantastic, and we will definitely be visiting some of those topics during the discussion, so get ready, all of you. I'm giving you a heads up. Okay, so for our last speaker, we're going to continue moving forward within the realm of really the human condition, really looking towards therapeutics and potential interventions that we can do, and in this particular case, really focusing on metabolism. So I'm really excited to hear the work that will be presented by Dr. Roberta Diaz Brinton.
So first, I do want to thank the Dr. Molly Wagster and the nominating committee for the honor of presenting our work to you this morning, and also to give a big shout out to the National Institute on Aging, who has been funding women's health research for decades. Kudos to them, because they were leading the field. Okay, so right, this is pretty simple. So I'll be talking about this midlife aging transitions and our discoveries about how that midlife aging transition in the female brain has implications for brain metabolism and ultimately for brain health and resilience. These are my disclosures. So one of the things that I've learned a great deal about over the several decades of this research is the way that we think about aging. And we typically think about aging as a linear progression from we're alive and then we're dead. What studying the female brain has taught me is that the brain actually undergoes multiple transition states. And so to understand these transition states, it is necessary to really look at systems biology throughout. And the importance of this is understanding these transition states gives us an opportunity to sustain brain health, to delay transitions to disease, and the potential to restore. So let's think about those transition states. So transition states can span years. We know in the case of Alzheimer's disease, that transition state, the preclinical state, can last 10 to 20 years. There's a sequential activation and deactivation of regulatory pathways. And that pre-disease is at the transition of normal. So when we think about what the normal range is a person at the high end or at the low end of that normal curve. And that small perturbations can unmask the existing vulnerabilities, and this is particularly true in the transition state. There's a critical slowing of the system, and this is particularly important for repair. Transition states are unstable, but potentially reversible. They are, however, time limited, and complex systems like that in the brain have critical thresholds, or what I call tipping points. Our study over a number of decades, our analyses over a number of decades, has taught us that there are 3 transition states in this aging process. Likely more, but we have categorized them into 3. The first is early chronological aging, and that's the chronological aging you don't even know is happening. But it is. And that early chronological aging precedes the endocrinological aging. And when we look at the early chronological aging and the transcription differences that are occurring, they're in large part happening in the hypothalamus, very few in the hippocampus. But look what happens in the endocrine, this process that you'd think would be hypothalamically focused. Actually, are very few genes changing in the hypothalamus, and over 2,000 genes changing in this cognitive zone of the hippocampus. And then the late chronological aging returns to the hypothalamus because the brain has changed. And now we go back to the hypothalamus and see the predominance of changes in transcription are happening in the hypothalamus, and that the hippocampus has changed. So when we look at midlife aging in the female and think about the perimenopause to menopausal transition of the female brain, it's a midlife neurological transition. We see that coincident with a decline in brain glucose metabolism, that happens early in this process and during the perimenopausal transition state. There's a rise in the variability of brain function. And when we think about the perimenopause to menopausal transition, we typically think about reproductive senescence. But if you look at the symptoms of this transition, they have nothing to do with reproductive senescence and everything to do with the brain. So we look at many women will complain about cognitive dysfunction, particularly cognitive dysfunction and insomnia. There will be changes in mood and depressive disorders. All of these symptoms of this perimenopausal, menopausal transition are actually neurological symptoms. We have studied the impact of estrogen regulation of outcomes in the brain for many years because of the change that occurs in brain levels of estrogen. We know that estrogen, I call the queen of Darwin. She has left nothing to chance. Estrogen promotes the uptake of glucose into the brain, the metabolism of glucose, ultimately into ATP. So that loss of estrogen during the perimenopausal and menopausal transition leads to about a 20 to 25% decline in glucose metabolism in the female brain. And that activates the release of estrogen suppression of the SCOT enzyme. And that SCOT enzyme is mitochondrially located and is involved in the metabolism, the generation and metabolism of an auxiliary fuel, ketone bodies in the brain. And ultimately, what we find is that this decline in brain glucose metabolism that is coincident with the loss of estrogen in the brain leads to this adaptive response in the metabolic system, so that there is decline in glucose metabolism shown in red, and the first responder is actually to utilize amino acids that can be converted to glucose or to ketone bodies in the brain, followed by this change in phenotype of the mitochondria from very efficient mitochondria to inefficient mitochondria. And that is coincident with a rise in fatty acid beta hydroxylation in the brain. And fatty acid beta oxidation in the brain is the metabolic response of now using lipids as an auxiliary fuel to generate ketone bodies. Good news, bad news. The other component of this whole system actually is the immune system, and there's activation of both the innate and adaptive immune systems that is coincident with these changes in the reprogramming of the metabolic system. So where are those lipids coming from that can now be an auxiliary fuel for the brain and be converted into ketone bodies? The brain is the most lipidated organ of the body. 60% of brain weight is lipid weight. And surprising to us, we discovered that the brain can actually catabolize its white matter for fuel. And this is a schematic of probably 20 years of research that shows that decline in glucose-derived ATP in neurons sets off a starvation response in the brain. And God bless those astrocytes, because those astrocytes are coming to the rescue. And those are the cells that are activated to conduct beta oxidation of fatty acids, to generate bodies, which then they send back to the neurons to generate acetyl-CoA to regulate ATP production. So that starvation response is a coordinated response between neurons and astrocytes. Here's the bad news. The bad news is that catabolism of the white matter for fuel leads to the generation of basically myelin fragments. And those myelin fragments are recognized by the microglia as a foreign body. And that foreign body now activates an adaptive immune system response to present that fragment onto the microglial plasma through MHT2 receptors. And now there is an infiltration of T lymphocytes from the periphery to activate what looks for the world like an autoimmune response in the brain. So this is happening during this midlife transition. So this is work that has all been in the preclinical discovery science space. We then asked the question with our colleague Lisa Mosconi is whether there's evidence for metabolic reprogramming in the human brain. And what outcomes of her work showed that when she paralleled our basic science discovery process design and looked at women who were premenopausal, perimenopausal, and postmenopausal, what she found that is indeed there was a decline in glucose metabolism in the midlife aging female. And that there is, in relative to males, there's a greater decline at this age, in age-matched males, there's a decline in glucose metabolism relative to the age-matched males. And is that leading to a change in gray matter? Not really, not specific to this menopausal transition. It looks much more like an aging change in gray matter. The real change is in the white matter. And consistent with our findings in our discovery science. And that change in white matter volume is coincident with the generation of beta amyloid deposition in the female brain. And that this change in both white matter and the potential catabolism of white matter in the human brain is coincident with a plasma metabolome that is characterized by elevated sphingomyelins and phosphatidylcholines. And guys, we can talk about you later. So could this trajectory of cognitive aging and metabolic aging be modified? And can we identify women at risk for this before that while their brain is still healthy? And this is work that we did in collaboration with Howard Hodis, Wendy Mack at USC in the ELITE trial. And the bottom line is when we looked at healthy midlife aging women who are 60 to 65 and older, what we found is in this healthy population of women, there were actually three groups of women. There were women who were metabolically healthy. There were women who were at risk for developing metabolic syndrome or type 2 diabetes, and a third group of women who had high blood pressure, were at risk for high blood pressure and cardiovascular disease. And when we looked at their cognitive function, what we found is that the healthy women in green performed best on these measures of cognition. The women with high blood pressure were not statistically different, but they were in a transition to lower cognitive function. But it was really the women who had a poor metabolic profile, not diagnosed with type 2 diabetes or metabolic syndrome, who showed significant decline in cognitive function. And then we asked, well, who are those women who are driving that change? And it turns out that they were ApoE4 carriers. So the combination of a at-risk metabolic profile and the ApoE4 gene led to this driving the significant change. But the interesting component of this is that ApoE4 was equally distributed across each of these, suggesting that if we targeted the metabolic health of those women, that we could change the trajectory of brain aging in those women to be consistent with the ApoE4 carriers in the healthy metabolic group and cognitive function. So if estrogen loss initiates vulnerability for cognition and potential for the development of Alzheimer's pathology, does menopausal hormone therapy impact the risk of developing Alzheimer's later in life? And this is work that we conducted, medical informatics, and in a propensity score matched group of over 300,000 women. And the bottom line is women who were on menopausal hormone therapy showed reduced risk of developing Alzheimer's, Parkinson's, all-cause dementia, non-Alzheimer's dementia, multiple sclerosis, and ALS. And the longer women were on menopausal hormone therapy, the greater the benefit for reduced risk. Good news, bad news. The fact that there is some association with hormone therapy, not necessarily estrogen alone, has impacted women's choices to receive hormone therapy, and 80% of them elect not to receive hormone therapy for menopausal symptoms, voting for breast health over brain health. And their principal reason, as I mentioned, was fear of breast cancer for that decision. So to sustain brain health, we had to address breast health. And to do that, we developed a strategy that targets estrogen receptor beta, which in the cancer field, from cancer researchers, showed that activating estrogen receptor beta in breast tissue reduced and inhibits estrogen-stimulated proliferation, inhibits human breast cancer proliferation, inhibits breast cancer cell migration and invasion, and essentially reduces that oncogenic pathway leading to breast cancer. So that was our perfect opportunity to now begin to develop an intervention that targets estrogen receptor beta in the brain because of its potential for breast health. And we did this a number of years ago, again, all supported through the National Institute on Aging, and we developed a formulation of 3 plant-based molecules that target estrogen receptor beta, and conducted a phase 1b/2a clinical trial funded by the NIA, and outcomes of that trial indicated that there were no indicators for this formulation, predictive pharmacokinetics, we established the optimal dose, and that we had biomarkers of responders that were feasible. So we are now conducting 2 clinical trials of the PhytoSerm formulation, or plant-based selective estrogen receptor modulators. The first is a clinical trial targeting hot flashes, and the second is our clinical trial targeting brain glucose metabolism and cognition, and this is being done in collaboration with Lisa Mosconi. So I think there is the potential for altering the trajectory of aging during midlife in the female. I want to emphasize that intervention with either estrogen or PhytoSerm is to sustain brain health. It is not to reverse disease. And I think that has contributed to some of the controversy in the field. But estrogen sustains the utilization of glucose and the activation of glucose metabolism in the brain and all the downstream consequences. Once the brain has undergone metabolic reprogramming, that brain is reprogrammed. So the key concepts is that menopause is a neurological transition that can unmask vulnerability into age-associated neurodegenerative diseases, and that the transition involves both the immune system and the metabolic systems of the brain, that estrogen and menopausal hormone therapy sustains and promotes healthy brain aging. It does not reverse disease. One size will not fit all. We will need precision hormone therapy, and that promoting brain and breast health is feasible through estrogen receptor beta selective formulation. And with that, I will recognize the many individuals in my current group that have contributed to this research and to the generous support from the National Institute of Aging that has supported women's brain health for decades. Thank you so much.
Awesome, that was fantastic. Please join me in thanking all the 4 speakers before we start our discussion. Perfect. So I just wanted to take a little bit of a minute just to give you a little bit of like my historical perspective. I was actually here as a relatively junior assistant professor for the 2017 summit. And just seeing these 4 talks, it really gives you a lot of like hope and energy, because just the techniques that were described in these 4 talks compared to what we were talking about in 2017, you can already see the advancements that have been made. And I'm really looking forward to seeing the advancements we're going to make after this particular summit as well. Before we start the discussion, really one thing that I think we can appreciate, and that I hope that the audience really begins to think is, a lot of times we think about cognitive aging and we think brain, but we have to remember that it's really an aging process, and that's really throughout your entire body. And I think we really need to start thinking about, brain health also as body health. And I think that they're very intimately linked. I don't think we're going to be able to disassociate them. And I think all the work that was shared by the speakers really gets to that interconnection between the body and the brain, and potentially even understanding a lot about the brain without even necessarily only focusing on it. So I think that that's one, I hope, take-home message that you get. So I think what we'll do is I can open it up for a few minutes for questions. I think we already have one there. And then we can also then move on to a discussion topics that I have as well.
Yeah, I'm Diane Bovenkamp from Bright Focus Foundation. I'm over here. Phenomenal talks. I was just blown away. Thank you so much. This is my first time and I'll definitely be back at Roman numeral 5 summit. So the last 2 talks really made me think about, understanding women's brains. And there's about 18 million people in the US who have ER-positive breast cancer, and they're given tamoxifen or aromatase inhibitors. Are there a lot of studies that have looked at them in terms of, seeing what happens? And then also, could they, I guess they can't benefit from this phyto because you're trying to purposely reduce it, and it's not a gradual, it's like immediate before. So how can we help those 18 million women?
So thank you, and great questions. So on the first question around what basically are breast cancer therapies impacting, and this is work, Christine Yaffe has also contributed to this work. We have found that women, tamoxifen and aromatase inhibitors, which was surprising to me, have reduced risk of developing these age-associated neurodegenerative diseases and Alzheimer's in particular. Raloxifene given at double the clinical dose does cross the blood-brain barrier and has some benefits, but at the clinical dose, it does not impact risk for development of these diseases because it's just not crossing the blood-brain barrier very well. But in culture, it activates the same kind of mechanisms. And what we find is that these selective estrogen receptor modulators and aromatase inhibitors activate estrogenic responses in our cultured neurons and in our animal models. But they have so estrogen therapy Hormone therapy reduces risk by anywhere between 50 to 60+% for risk of Alzheimer's, and the selective estrogen receptor modulators are about half that effective. So they're acting, and we have published a number of papers showing that these estrogen receptor modulators are activating estrogenic responses in brain. Now, can women who have a risk for breast cancer use phytoserums as an alternative? I don't know, Dr. Hodis, but we could do that clinical trial. So we don't know that yet, but it's a very important question. It's a very important question and issue. Thank you.
Why don't we go in the middle?
Very, very good talks. I have two questions, one for speaker one. So Roberta, if it's a switch from glucose metabolism to ketosis, would it be possible that intermittent fasting, supplying the ketones from the rest of the body, would supply the brain, that it would not have to break down the myelin? There's a lot of data out there that restricted food intake is very good for longevity. So do you have any thoughts on that?
So another great idea for a clinical trial. My concern is this. Is that if the brain is utilizing its own white matter for fuel, right, it has an abundant source of ketone bodies. So feeding it more ketone bodies may or may not be particularly effective, from the periphery. So the real challenge that I think about is, and coming back to that transition state figure, how can we march the brain backwards, right? So we're not going to be able to take a intervention and take us all the way back to, brain health. I think we're going to have precision therapeutics that target what transition state you're in. Now, part of the Alzheimer's space is, caloric restriction is potentially hazardous in that group because many people are already quite thin and they're losing weight, right? So it's a very good question about how we can change the trajectory through nutritional interventions. And I think that is a key question that remains to be answered.
My thoughts are that, if you look at it from a little bit the opposite direction, that this is the normal change in brain, that it comes from glucose to ketones, and we just need to feed the brain with the correct thing, then maybe it's not going to have to break down its own tissue.
Well, and you make a very good point, because I saw in an earlier slide during the introduction, what about the 87%, right, of people who don't develop Alzheimer's disease, right? And how do you sustain that brain health? I think that's a really important point. And I know that in the Precision Aging Network, led by Carol Barnes, that I have the privilege to make a minor contribution to, is really looking into those questions.
Thank you very much. And then Tara, the other question I had is that we are treating most of the population with antiplatelets, aspirin, Plavix. Is there any data on how it would affect this PF4? Are we doing harm with using those drugs? We are overusing them sometimes. There was primary prevention in diabetes, and they didn't even have to have a vascular event, and we are using it. Is there any data on that?
Yeah, I'm not sure if there's data on that specifically, but yeah, that's a really good point. There's a lot of antiplatelet therapies for a lot of neurodegenerative conditions, and whether we know that PF4 is one of the major factors that's released from platelets when they get activated. So these therapies would be decreasing levels of PF4 systemically. So yeah, I'm not sure if people have specifically looked at the PF4 levels in these populations of people that are being treated with these. So yeah, that's a very, very interesting point.
Thank you.
Closest to the entrance? That's it, yeah.
Okay. Hi, I'm Michelle Kelly from University of Maryland School of Medicine, just down the road. So I was kind of piggybacking actually off the first question from the last individual, but thinking of it more in the context of that increased risk really being in the metabolically challenged group, those about to develop metabolic syndrome or type 2 diabetes, and that interplay with ketones, because many patients are put on ketogenic diets for diabetes, and it super helps the peripheral manifestations of the metabolic syndrome. A1C goes down, they can have more energy. Some people complain of brain fog, but then other people feel like they have super sharp cognitive functioning. So I'm just wondering if beyond the intermittent fasting, which is a little different than the straight up ketogenic diet, have you thought about how that interplays here?
I have not, and I come back to transition state biology. So what you're describing are at least two different groups of people, one who have not made that transition, and the other who, in fact, may have made a transition. And I think this comes back again to the precision aging strategy of how do we actually design and execute interventions that are, precise for this aging population. Where are you, and how can we prevent, advancing or march back to this health, brain health and metabolic health? So I think there's a lot of work to be done. Thank you.
We'll go closest to the door again.
Natalie Rasgan, Stanford. I am delighted and intellectually invigorated with this panel. And of course, I have to compliment all of you on fantastic presentations, but I have a specific question because of course I can take time forever and be thrown out of this audience with a number of questions I have. But I'll ask only one to Dr. Jacobs. Emily, how do you think or envision the future studies of pregnancy specifically, in the connectivity, brain connectivity in pregnancy, in the isolated kind of idealized, right, 28 and us cycle, and then taking it forward through the physiological pregnancy? Do you control for the metabolism, actually, which Robbie so eloquently is defending and we all so support? But the question of the weight, right, the BMI, the weight gain in pregnancy, the metabolism associated with the pregnancy, women who are a priori prior to pregnancy obese, etc., etc. Fernando de Feliz just published a fantastic structural paper on the obesity. I don't know if you've seen it. You can look it up. It's a brain structure changes specifically to obesity. So there's a lot of potential confounds to the very elegant model you're suggesting. We found the leptin specifically, that's kind of a little towards Robbie's presentation, that in the metabolic phenomena on the periphery, the leptin and therefore the hypothalamus, much more than hippocampus, have profound influence on the brain function in structure and connectivity. So there are a lot of confounds to consider when you want to look at pregnancy.
Yeah, and Natalie, there we go. I think that this field of the maternal brain or parental brain is just getting started. There are 1,001 questions that have to be addressed. I don't think this is explained by weight, and I say that because these changes don't go away after pregnancy when rate reduction comes back down. Again, these are more or less permanent etchings in the brain, at least with the gray matter volume changes and also in aspects of white matter. I think that these are this sort of extended neuroplasticity is happening to support behavioral adaptations that we know emerge in pregnancy. We have beautiful animal models. There's a science paper from Amari and colleagues looking at steroid hormones' ability to fine-tune the response properties of galanin neurons in the medial preoptic area of the hypothalamus. And with that fine-tuning, the dams, the rat moms, gain this increased sensitivity to being able to pick up subvocalizations from the rat pups and to literally smell them with higher sensitivity, right? I will say that in addition to the human brain imaging work, like what we're doing, I would love to see more systems, neuroscience, system biology support given, because I think that's going to tell us mechanistically really what's going on. To answer the question about, thinking about peripheral weight gain, we know that women, retain more water, or, often some women do more or less. This paper is really, it's coming out as a resource paper. It's really just a proof of concept to say, guys, we can do this. And in fact, we scan pregnant women all the time. We have beautiful maps of the fetal brain going back to 20 weeks, gestation. Why didn't anybody think to just move the bore of the scanner down 3 feet and look at what's happening to the maternal brain? So this is sort of proof of concept paper, but it comes on the heels of a much larger maternal brain project that's launched at both UC Santa Barbara and UC Irvine. We're hoping to expand to more centers where we're tracking large cohorts of women over this period of time, and that includes measures of arteriospin laboring, so we can start to look at cerebral perfusion, in addition to a whole bunch of other metrics. So great questions. We're just digging into it. I think there's going to be a really cool story coming out about the immune changes that happen during pregnancy and how, there's this sort of neuroimmune dance because the mother has to tolerate the fetus, and there's all these sort of immune changes that occur to make that happen. So in a breath, we're just getting started.
Fantastic. And last comment on that. Obesity is a profoundly inflammatory condition, profoundly immunological condition, and in fact changes estrogen levels in itself. So there's no ovulation in obese women quite often. So there's a lot of things to think through, but great. Thank you. Congratulations.
Over here on the right.
Hi, so my question is to Dr. Brinton. The question is also related to the metabolic shift from glucose to keto, because I wonder, plus like you're doing the functional human imaging, usually our target is like oxygen or glucose metabolism. So do you suggest like to look beyond those metrics, or should we look for like blood flow or other more advanced like markers?
Yeah, so that's a great question, and we're doing MRS imaging to address some of these metabolic questions. But yeah, the more imaging, the better, just letting you know. I think there's a lot of opportunities here. And I think one of the things that, has already been brought up in several instances, I know from Andrew's work and Taro's work, as well as Emily's and mine, is, the dynamic nature of the brain, and to what extent can we bridge this divide from kind of mechanistic discovery science to the human brain and asking exactly the kinds of questions that you are. How can we detect this in the human brain? And in whom and when do we detect this? And so I think the benefit, the real, I think, advantage that we've had in our human brain imaging is the fact that we have a collaborator who matched their kind of imaging protocol to our transition state biology. And I think that's going to be really, really critical. So hopefully you'll jump into the fray here. Thanks.
We're here on the left.
Hi, Steve McCarroll from the Broad Institute. So thank you all four of you for really thought-provoking talks. I have a question for Dr. Jacobs. So loss of thinning of cortical gray matter and loss of dendritic spines are also really prominent recurring findings in many psychiatric disorders, especially schizophrenia and depression. In those disorders, though, at least I'm aware, no one looks at them with the high temporal frequency that you do. And so we actually tend to think of them more as kind of relentless, irreversible, one-way changes. And I kind of feel like implicitly in what you presented is kind of an astonishingly different picture of processes that are actually fluctuating in both directions at high temporal frequencies, and also probably responsive to peripheral events. And it's actually a really different picture than how these things get talked about in a different field. And I just wondered if you could expand on that, because I think, I don't know, sometimes when we talk about these things in the context of psychiatric disorders, it's with a kind of like resignation and hopelessness about like not being able to reverse them and just maybe being able to prevent them. But I feel like such a different picture comes from your work where you're looking at these things at high temporal frequency.
Thank you. That's such a great question. And I think one hope that this work can really tell us is that, as Robbie said, the brain is a highly dynamic organ, and these endocrine, right, the brain's an endocrine organ. It's responsive to these ebbs and flows. And I hope that some of the basic principles that we can take away is that these hormones can drive these changes, and if we can understand that and sort of bottle it up, it could give us really interesting ideas about regenerative medicine and how to use these, extended neuroplasticity that we see, for example, in the maternal brain. Can we apply that to the other conditions where we need that degree of neuroplasticity? And I think, this is why I took 3 minutes to sort of begin this way. I think there are so many nuggets of gold lying just under the surface of our knowledge that have not been addressed, in large part because women's health has been ignored for so long. And there are just these beautiful experiments of nature where we can see this, these endocrine pulses driving these pulses in the brain. They're surely adaptive, and we just need so much more work in this space to realize the clinical utility of it.
We'll do one more here.
Hi, Carol Barnes, University of Arizona. I'm wondering if Tara and Robbie can address this issue for me. Listening to your talk, Tara, and listening about neurogenesis, your mice were 20 months of age, but neurogenesis plateaus at about 15 at mid-age. And it just struck me how, is this a transition like Robbie is talking about, that, many things are changing at this transition point, including neurogenesis?
Yeah, so in the 20-month-old mice, that's sort of relative to about a 80, 85-year-old human, so that's quite end stage for mice. In mice, neurogenesis actually starts off relatively high and drops quite quickly. By around 3 to 4 months, the neurogenesis levels are already declining.
Right.
And also in humans, there's a few human studies that have come out in the last few years looking at neurogenesis in humans, showing that it does exist and does decrease with age and does decrease in neurodegenerative conditions like Alzheimer's disease. So I think our 20-month-old mice is, I wouldn't say necessarily a transition stage. It might be the very last transition stage, but it's more like a close to an endpoint stage. If we wanted to look at a transition stage, I would suggest that we'd look slightly earlier in mice than 20 months.
Well, to dovetail on what Tara has said, it makes me think about our earlier work we were using a mouse model of Alzheimer's disease, and then we always had a normal age-matched control mouse in addition to the non-transgenic mouse. And astoundingly, for neurogenesis, we never saw an impact that we're developing a regenerative therapeutic, allopregnanolone, thanks to NIA, for Alzheimer's disease. And in the normal mice, we never saw neurogenesis being activated by allo until they were 15 months of age, right? And we know that in those really was neurogenesis. We had all the markers for that. So it really, I think, speaks to the regenerative potential of the aging brain. Now, in the data from our phase 1b/2a clinical trial in persons, 60 years and older, we found a set of responders. It was a double-blind, placebo-controlled phase 1b/2a clinical trial, small numbers. But we found that there were individuals who had gained small in, just over 3 months of 12 treatments, had gained hippocampal volume in this aged population, and that they were ApoE4 carriers, right? So those were the responder set. And now we're in a phase 2b clinical trial for efficacy in ApoE4 carriers, all of whom are going to be 65 years and older, right? So I think I was surprised, but in the normal mouse, I was so thrilled because it was just this little blip that finally the mice, the older mice, were showing. And what we find with allopregnanolone is that allo doesn't give you more if you're good. It's only if you have a deficit in neurogenesis. So these aging mice started to show the deficit, but there's hope.
Yeah, I just wanted to add to that. So we see the increase in the neurogenesis in the younger mice with PR4 treatment, but it's only in the older mice that we see the cognitive improvements where there's the obvious deficit. So in the learning and memory tests that we use, we don't see an obvious deficit until about 16 to 17 months of age, and that's why we test them so late.
So can I just say something because I need to? My colleague, Jen Bison, did a heroic study of neurogenesis and spatial cognition way back, it was quite a long time ago, but the old animals that had worse neurogenesis were the animals that were better in spatial cognition. So I'm just wondering about the linearity of the changes in neurogenesis and spatial memory.
Yeah, we've actually seen the opposite to that. So we do the neurogenesis studies in the same mice that we do the cognition, and we generally find that mice that have worse or lower levels of neurogenesis have worse cognition.
So I would just say, Carol, if the rats knew that Carol Barnes was watching, they knew they had to do well. I'm just saying.
Why don't we go to the right here?
Yeah, Chen Chen from Brown University and Rhode Island Hospital. This is a question for Dr. Andrew Yang, since nobody asked you a question yet. So I think I love your talk, and we know the BBB actually shields 80% of small molecule drugs and almost 100% biologic drugs from going to the brain. And it's really fantastic that you found hundreds of proteins crossing into the BBB. I'm just wondering whether you have learned any mechanism by which enable those proteins to get into the brain so that we can use those properties to enable the drugs to reach into the brain.
Yeah, thank you. I think that's it's on, right? Can you hear me? Right. Yeah. Sure. No, I think that's a fantastic question. Back to the analogy of the blood-brain barrier is forming a special home for the brain. I would imagine it, right now we design molecules without taking into account blood-brain barrier biology itself. So it's like trying to run into a wall, right? But if you can now understand the different entryways, the built-in conduits, the keys, molecular keys, then you'll have much better success. At least for biologics, I think we're starting to see that in the industry. There are now over 10 pharmaceutical and biotech companies that are leveraging this built-in receptor-mediated transcytosis pathway with these kind of transferrin antibody-based shuttles. I think that's the start. And I think, just at JPM in January, people are realizing that's the new table stakes. Everyone is going to use this as a new platform for delivering macromolecules into the brain. But there are liabilities for that one target. And I foresee an expanded set. Just like there are now combination therapies for immuno-oncology, I think we need combination therapies for the brain. Different targets for different disease contexts. Because we know, for example, that transferrin receptor expression, abundance, and activity decreases with normal aging, right? And so you'll have a smaller therapeutic window. And so I think we'll just need to complement that and build it out. And I hope, on top of that, that we can have the ability to not just get molecules into the brain, but because we now have all these beautiful single-cell data, RNA-seq data sets of the brain vasculature as well as the brain cell types themselves, you can have cell-type specific therapies, right? A therapy like an antibody that can both enter the brain and then home on, or oligodendrocyte or microglia, where different diseases manifest.
So we're going to have a couple more questions, and then I would like to leave just a few minutes to let me have a little bit of fun and ask a few questions as well. Right here on our right.
Thank you, Elias Maslia from NIA. This question could be for anybody, but Robbie, I was quite fascinated by the data you show on myelin catabolism and microglial interactions, and I was wondering which one, would we consider that that is the main driver of inflammation in aging brain, and that maybe there is some sort of like an autoimmune response to myelin during aging, or are there other drivers that we think are more important for inflammation in the brain in aging? I mean, we know that there is clearly astrogliosis in the aging brain. I mean, this is non-Alzheimer, no plaques, nothing. We know there is definitely astrogliosis, but I was wondering the thoughts of any.
Yeah, so it's a great question, Eliezer, and thank you. So I got dragged kicking and screaming into the immune system, but that's where the brain was taking me. And to your point, it's one of the things that we are about to submit for review is asking the question a bit like you were asking, and we asked, can we change the trajectory of this brain aging by targeting the innate immune system, because that's the first system that's actually activated. It's activated before metabolic reprogramming. So the innate immune system, you don't even know it's happening, and it's activated. So we asked the question, can we actually target the innate immune system and change trajectory of that aging profile in the perimenopausal, menopausal brain? And the answer is yes. It takes combination therapy, multiples, but it's quite effective. So I think there's the potential for actually changing the trajectory of aging with early intervention in this transition state. And then, then the question I think also that you're asking is, how can we bring that back? And what we see is that there's activation of the innate immune system and then activation of the adaptive immune system. So it really depends upon which end of the spectrum you're at, because when we look at that later stage where there is myelin catabolism and the generation of this autoimmune, I think we're going to need therapeutics that target that autoimmunity system. And it's kind of no accident that over 90% of people who develop multiple sclerosis over the age of 50 are women, right? So I think it's a great opportunity for us to really think more deeply around that immune system.
Thank you.
Okay, so just in the interest of time and because I do really want to have fun with a few questions, I'm going to go ahead and start the discussion, but we have a 20-minute break right after this, and please come up to the stage, grab them, and ask your pressing questions. I just have a few of my own as well. Okay, so this is going to go to every single one of you. I really want all of your thoughts, and I'm going to randomly just pick on you all. Okay, so I'm going to piggyback off of Maslia's question on the immune system. So we're talking about periphery to brain. And there's no way we can talk about the periphery in the brain without talking about the immune system, immune cells, immune factors. And we have transition states. We're all on different trajectories. So in aging, oftentimes we think about the immune system or inflammation or inflammaging and detrimental. But then the immune system is adaptive, and it's there for a reason in the first place. So how does each one of you view that sort of like dichotomy between the good role or the beneficial role of the immune system versus the more detrimental? Where do you think it sort of lies in terms of a driver of aging versus a potential target of resilience or to delay kind of aging? And I'll just go in order on this one since, Tara, you study platelets, which is an immune cell.
Yeah, that's a really good question. So basically platelets have always been, until relatively recently, thought to be a detrimental part of the immune system in aging because they're in almost any sort of neurodegenerative condition, there's platelet dysfunction. So it's basically only very recently that platelets have shown to have all these other functions, particularly related to the brain. And this is why we are so interested in them, is because they do seem to contain many, many beneficial proteins, factors that are known to be rejuvenating in the brain. And there's been a number of studies in the last few years showing that platelet-derived factors, or just whole platelet lysates and other platelet-derived products, potent neuroregenerative therapies. So I think this is a topic that's just come up relatively recently in the last few years, and something that's gonna really come to fruition in the very near future as a regenerative therapy.
Andrew, your thoughts?
Right. I think it's such a fantastic question. From the perspective of brain barriers, I think we're beginning to understand they permit and even house a whole milieu of immune cells, right? I think one beautiful embodiment of that is kind of the understanding of the dura and how it can house antigen-presenting cells, T cells, plasma cells, myeloid cells that can shape the brain at a distance, right? I think we're understanding that immune cells don't necessarily even need to be in the parenchyma. They can secrete factors, and your neurons, surprisingly, have antennas for them. They express all the various flavors of cytokine receptors, and this can shape development, but also disease. But I think most interestingly, they can shape different trajectories within the normal range, I think, in midlife. I think what was brought up earlier about how autoimmune diseases, for example, are so sex-biased, or how some of these kind of peripheral inflammation is so sensitive to our lifestyle, I think we're going to realize has a profound influence on brain health through the brain barriers.
Emily?
I think I'm going to pass this on to the true immunologist. I'm only dipping a toe in.
Hormones, I mean, yeah, you have to.
But, the way I'm starting to think about this, and this is with this brilliant woman, Ina Stelzer, is really thinking about these neuroimmune changes across pregnancy and how the song and dance is really sort of changing in a pretty drastic way. So maybe at the next summit, I'll present those data. But I'll pass it on to Robbie, who's far more of an expert in this space than I am.
And I, would rattle in the thimble loudly, but I want to come back to Andrew's point around the blood-brain barrier. It's very interesting. What we've found is that when the brain is under metabolic stress, the blood-brain barrier is much more engageable. So there's this crosstalk between the brain saying, I'm starving, and the peripheral system, metabolic system, and potentially the immune system. And then once that shift has happened, once things have been reorganized, there is no communication, but it was really driven by the stress, the metabolic stress. And then it's like, I'm done up here, you're good, right? So then there is this, less communication between the brain and the periphery through the blood-brain barrier. The other thing that I would say is, this dynamicism of the brain, and I think about Alzheimer's disease. We think about Alzheimer's disease as this, you start here and you end there, and it's not pretty. But when I step back and I look at the Alzheimer's brain, I'm thinking, my God, you're dynamic. You're living for decades. You're living for decades with this, what we call disease. But when I step back and I look at the brain, I'm going, oh my God, you're trying so hard to survive. You're trying so hard to survive and doing everything you can to survive for decades. And so it really has changed my perspective, having studying aging and its dynamicism, and looking at Alzheimer's and the potential of dynamic components within this disease that we see this, progression of, decline in brain function. But boy, it's doing its best to stay alive.
So in the last couple of minutes before the break, my last question is around technology. So, from the 7 years since I was here, technology has really made a huge impact. And in each one of your fields, the way that you think about cognitive aging, what is it, some areas that technological advances or the types of technologies maybe you think we need to really push these questions forward? And I'm going to start with Andrew and just go down the line.
Okay. Well, I think it dovetails what Robbie was saying earlier about how the brain is so dynamic. And I think that technologies that are coming online allow us to study these dynamic processes. For example, I think the last decade has seen a revolution in cell autonomous biology because of things like sequencing, single cell, CRISPR. And I think new tools, like the ones I shared here, are coming online to be able to study cell non-autonomous biology, how cells communicate with each other, how they coordinate to enact the programs that give rise to something as complex as brain and brain health. I think in the context of our conversation here, I think an interesting parallel is we think about the brain oftentimes in terms of anatomy, right? But I think with these new technologies, we can now study things that are not necessarily stable anatomy, but maybe dynamic interactions. So what does a neuroimmune circuit look like, right? For example, do specific neurons need specific cytokines or specific immune cells to be in contact with them? I think that would be fascinating to figure out.
And then the very last answer will go to Emily, just for the sake of time. So you're the last one on tech.
There's so many technologies we can bring to bear. I think, on this question of sort of women's brain health over the life course, I think neuroscientists are so taken by the complexities and unique capabilities of the brain that we forget something really essential about it, which is it's connected to the rest of our body. I think if we could develop real-time monitors, through interstitial fluid, where we can start measuring these steroid hormones and how they ebb and flow in the time to give us over time, and how it could give us insight into dynamics in the brain, would be really cool. I think we need better ligands to start looking at where these receptors are in the human brain. The Allen Brain Atlas has 7 postmortem brains where we have these beautiful, gene expression maps. One of them is from a woman who's 57 years old. So we just need better atlases using the technologies that we have today that are inclusive. And just another plug, I think there needs to be, all of these technologies that we have from a systems neuroscience approach can be leveraged to these new questions. CRISPR being one of them, and, so many others. So, thank you.
Awesome. And with that, I just want to end first by thanking Molly and all of the organizers that invited us for this particular session. And John, I don't know if you're coming or if I should break everyone for a break. Okay, John is coming.
This has been wonderful and almost exactly on time. And so we are going to come back exactly on time at 10:55 a.m. for session 2, the structural and social environment. Thank you then.
Okay, everyone, welcome back from the break. Want to welcome you to session 2. And my name is Jennifer Manley. I'm a professor in the neurology department at Columbia University. So just a quick introduction to our session. I'm excited to have a session dedicated to talk about structural and social environment at the Cognitive Aging Summit. Of course, context is critical for any precision approach to extending brain span, which is what we're talking about at this summit. This session focuses on innovation in measurement, design, and highlights important developments in this area of cognitive aging research. And these issues are important for all dimensions of the environment that we're talking about today and tomorrow, and should be infused in discussion of all of the mechanisms that we're talking about at this Cognitive Aging Summit. So with that, I'm going to introduce the first speaker, Dr. A.J. Adkins-Jackson at Columbia University.
All right. Hey, everyone. Okay, I just wanted to make sure we was all raised to speak. How y'all doing? Okay, put my water right here. All right. You all are assuming I am well-versed enough to know what to do with this. If I just hit the clicker to the right, is that how that works? Okay. I'm an upper millennial, so you can't assume I grew up with technology. I didn't see it until college, so all right, I'm AJ. I'm going to gossip with you about structural racism today. So we're all quite familiar with racialized disparities that we see where we're comparing different groups on a particular outcome. So we're used to seeing this situation where groups who are minoritized in our society have worse cognitive performance, mild cognitive impairment, dementia, incident dementia, whatever the outcome is, than groups who are racialized as white. But I find as I do this work, not enough of us have a robust understanding of history and where we even get these racialized categories from. So if you're not familiar, we have something from the 70s from our beloved Office of the President, the Office of Management and Budget, that created and dictates these racialized categories that we often use in the disparities research that we have. This is largely due to an effort to count equity, to make sure that there's adequate distribution of resources and science for individuals, but it's not meant to imply some of the genetic features we often see in science. So on the first page of this report that I find interesting, it always says, these groups are not to be interpreted as scientific or anthropological in nature. I mean, how many of you have actually really considered that when you think about racialized disparities? Is that we are not supposed to infer the things that we often do about the work that we do. But we are, for equity purposes, mainly historical purposes, supposed to track these differences. And so I like to always inform people on the history of these categories, because this is the context by which these categories really matter to us. That these categories are not the ways individuals has characterized themselves, but these are external forces that have looked at groups of people and said, all of you, y'all go together. And all of you, yeah, y'all go together too. Even though those groups, for historical reasons, would believe they're quite different than the people they are grouped into being with. So I want to give you this historical context of people from the era of European colonization, the most recent colonization we've seen, that have grouped people in these large categories we use. For example, Veneer, who categorized Persians and North Africans as European. So that characterization that we have of thinking of them as European comes from a very particular place in history, not necessarily their own characterizations of themselves. Same thing with John Locke, whose research undergirds the criminal justice system we have in the US, used cognition and logic to differentiate groups, saying that there was different cognition by Negroes and Chinese and American Indians. And then you have Linus and Blumenbach, who are largely responsible for the categories you see today, as they were the major sort of engineers of the classification system that we see. So whenever you think of these racialized groups, think of them in this very specific historical context. It was created for them. This is what we might refer to as the process of racialization, where you look at individuals and you go, I think you're group A, you're group B, you're group C. Now, what's unique about racialization is that it also ranks these groups. So not only does it force you into a category that you might yourself, but then it decides who gets access to resources based on those categories. This is very important to keep in mind when it comes to some of the disparity-happy points we have in the science that we do, because we compare groups without that historical context, so we have no way of really even understanding how they got to the outcomes that we see. Now, if you wanted to shift the energy from just looking at the differences in those outcomes to looking at actual reasons we got there, that would mean to focus on the exposures. That might mean to focus on social determinants of health, which are sort of access to resources, food, income, education that you predominantly see in the literature. I like to, with my colleagues, refer to structural determinants, because we know that there is not a policy in our respective places that don't then dictate that access to education and income and food and other resources. And because of that racialization, it then ranks groups in access to food, to income, to education. And I know my colleague Katrina will talk about access to education in her presentation. A lesser discussed part of this sort of racialization is the intention to do harm to individuals and the violence that some groups experience. I want to highlight this because on the left we have a situation where because the individual is racialized the way they are, they're on their knees, you can see both their hands, but there's 3 officers with weapons pointed at them. Whereas on the image to the right, the individuals are actively assaulting the officers. But because the way they're racialized, there's not a weapon raised. And so without really capturing the deficit that violence gives to our bodies, we're not really getting the full picture. So one of these types of violences I like to focus on in my study of structural racism is the 1994 crime bill. This was a significant bill for a more recent time. I hope everybody in the room has been alive since 1994. Yeah, okay, we was here. Okay, great, sounds great. But what it did was it funded the expansion of prisons throughout the U.S. to house, aka decrease the crime rate by increasing the number of incarcerated people. But because of the scientists like us that create these rates and disparities, it was inferred that crime rates are higher in neighborhoods with predominantly people racialized as Black and Latinx. So then these became the communities targeted for incarceration. So we wanted to see if that disparity in Black-white incarceration in prisons was associated with cognitive performance among adults racialized as Black. So in the top left corner, I have the co-statistical leads for this paper, Ariana Gobad and Bowen Keem. I always like to highlight the statisticians because as a statistician, y'all be forgetting about us, but, without us, the world don't churn, I'm just saying, we deserve awards too. Thank you, So, the Black-white disparity in incarceration for 10 years, 95 through 2005. We gave the crime bill, 1 year for implementation, and then we tracked it across 10 years and then averaged that number. We then looked at overall cognitive performance in the health and retirement study. If you're familiar with it, it's a biannual survey. I'm assuming everyone is. I'm like the newest to aging in this crowd besides my cousin in the first panel. But I'm assuming everyone knows about health and retirement study, and particularly the global cognitive assessment that they use, the modified TICS. But for this sample, predominantly adults racialized as Black, born in the U.S., living in the South, predominantly cisgender women, have a high school diploma, in relationships, and the average age is about 65. Usually when we build our structural racism measures, we do it at a geographic level, and then we link it to the individual data in another data set, like the health and retirement study. So in this example, we're linking zip code at their baseline interview, which would be age 51, or about that age, depending on when they joined the study. But we've also linked data based on their state at birth or their state at age 10. And so what we were able to see is greater incarceration of people racialized as Black in this time period, 95 to 2005, is associated with lower cognitive performance among participants racialized as Black. So that means if you're living in a county that disproportionately incarcerates people racialized as Black, there's an association there with the participants racialized as Black, their cognitive performance. Now, unfortunately, we also ran this study in the participants racialized as white. And so we saw if you had greater incarceration of people racialized as Black, that was associated with higher cognitive performance among participants racialized as white. Now, I illustrate this example for you to give you another way of doing disparities research. It doesn't always have to be a comparison between a minoritized group and a group that has experienced privilege in our society. Maybe it's about separating those two groups and looking at an exposure on these groups separately in different data sets. Now, I base a lot of my understanding of aging and cognition from this paper that got me into aging a couple years ago about life course factors, particularly in individuals racialized as Black, from Dr. Manley and Dr. Gleamore. And so they talk about a lot of these social or more distal factors that may lead to downstream effects that you would see discussed even in the first panel. Like that had my mind going on how we can connect so many different structural factors to what was happening. That paper beautifully talks about the life course and how these exposures are happening, not only in critical periods like the crime bill, but also through trajectories like poverty that people are set on, immediate exposures like an encounter with a racist doctor, or even the accumulation of exposures as you might expect to see in pollution. And so while I know a lot of our literature might focus on exposures in midlife, I know there's grown that early life exposures and adverse childhood experiences are associated with lower cognitive performance. And these last two pictures are of my granny who has dementia, is living with dementia, but also experienced extreme racism growing up in this country. One of the experiences she's talked to me about is living in an environment where people could be lynched. If you're not familiar with this, this was an act that was largely performed from the late 1800s to the mid-1900s, honestly, where people who were racialized as Black, even Filipino, Mexican, Cantonese, were killed largely by people racialized as white. And there was no separation between interpersonal racism and institutional racism, because even individuals committing lynchings were often pardoned, as this next newspaper clipping shows, by the governor. So they were often protected from the criminal consequences of killing people at random or if they believed them to have been a criminal. There were also sort of publications that planned lynching. So the one on the left tells us that they expected a lynching at 5 p.m. that day. They prepped for it, even discussed creating jerky out of the body of the individual that they would kill. So because the criminal justice system that protects individuals who create these murders happens at the state level, we wanted to see if state-level historical lynchings played a role in C-reactive protein or systemic inflammation and cognitive test performance. In the health and retirement study, participants racialized as Black, and the co-statistical lead was double Dr. Cesar Tejera Higgins. So what we saw is just comparing states with one lynching to states with two or more, because we didn't want to use a median or a mean value as if there was some prescribed number of people could be killed that would be appropriate. So we decided just to compare 1 to 2 or more. And we found that just being in a state with 2 or more was associated with 17% increased CRP and 1 lower test score on the cognitive assessment. Just 2 or more. Oh, okay, now I'm doing weird stuff, y'all. You can't trust me. Okay, thank you for fixing it, because you can't trust me. We've also looked at socioeconomic racism across the life course. So what we did was extract a cohort from the health and retirement study, the mid-baby boomers born between 1954 and 1959, after the Brown versus Board of Education ruling. And then we mapped socioeconomic racism around their life course. So we captured socioeconomic racism occurring before their birth, during their childhood or adolescence, during adulthood, and then midlife. And what we saw was greater socioeconomic racism was in childhood, so between 1960 and 1970, was associated with more rapid cognitive decline. Now, I have to note that these individuals entered the health and retirement study in 2010. So this is only 10 years of their cognitive performance that something happening in their childhood is impacting. But it's not all doom and gloom. Let's talk about structural socioeconomic resilience? Is there a possibility that we can create spaces that are protective? And socioeconomic resilience is one of the key ways minoritized communities often fight back against structural racism, by obtaining education, getting higher incomes, owning property. So what does this look like in aggregate? Using those same indicators for socioeconomic racism, we then don't configure a black-white disparity. We configure a proportion. What is the proportion of people racialized as black above the poverty level? The proportion of people racialized as black who own homes. And then we look at sort of cognitive performance across that decade. And what we see is socioeconomic resilience before their births was associated with slower cognitive decline. So that farm ownership, that home ownership, all was protective. I'm now in the understanding of life course racism that it might not just be about the exposures an individual has, it might be about the history of a place. So a place, irrespective of when you've been there, how long you've been there in your life course, might be contributing to the cognitive performance you see over your life course. And so I've been building this new model with my colleague here, Dr. Maribel Robledo Gonzalez, at The Ohio State, to build an anti-Latinx structural racism measure where we actually start at the moment the US-Mexico border moves, and then we track racism ownership over time. So next time you're with your colleagues and you're looking at disparities or you're talking about differential impact and you're encouraged to think about someone being born with something, I encourage you to think, maybe it's racism. Maybe racism is playing a role in what has happened to individuals and the disproportionate impact to their bodies. So I'd like to thank this huge collaborative team that we have, the Marvin Gray Matter team. You get that, Marvin Gray Matter? I like that so much. Okay. We collaborate on measures of racism, share data. We're practically a cult. And Ariana Gobald here is defending her dissertation tomorrow, so she'll be the latest one to get her PhD. I also want to thank my elder authors like Dr. Manley, Dr. Barnes, Dr. Thorpe, Dr. Lang, who've been writing this into the fabric of our science, and I'm just happy to be a part of illustrating that through my little bougie mathematical models. Thank you to the funding I have, but mostly thank you to my granny who has, through her lived experience, taught me the most about this science. So, thank you.
Thank you so much, AJ. And now I'm going to introduce Laura Zahodne, who is in the psychology department at University of Michigan.
All right, thank you. Thank you so much for the opportunity to be here. Thank you for the opportunity to be a part of this panel. This is a huge honor. My lab is interested in multiple aspects of the social and structural environment. I'm going to start my talk today on discrimination, which may influence inequalities in cognitive aging alongside other social determinants of health. Our work in this area has been grounded in this theoretical framework from Rodney Clark, which characterizes the day-to-day experience of racism as a potent form of stress that can influence health outcomes through psychological and physiological pathways. So I'm going to show you now some empirical evidence supporting this model as it relates to brain and cognitive aging. So starting with psychological pathways, using data from the health and retirement study, which AJ thankfully introduced us to already, here we were interested in psychological constructs that qualitative work had shown to be influenced by the experience of day-to-day discrimination, including changes in mood and changes in external locus of control, or the perception that you're not really in control of important life outcomes. And what we found was that those Black older adults in the HRS reported more experiences of discrimination, which predicted both more depressive symptoms and more external locus of control. And both of these psychological consequences of those discriminatory experiences were related to worse cognitive health. So some evidence that discrimination may influence cognitive aging in part through psychological stress pathways. We're also interested in physiological mechanisms by which discrimination might get under the skin to affect health. Here we incorporated some longitudinal biomarker data from the HRS, specifically C-reactive protein, which is a marker of systemic inflammation. We know from both animal and human research that CRP and other markers of inflammation are elevated in the context of chronic stress. So we wanted to test whether increases in CRP mediated longitudinal associations between discrimination and cognitive health. Now, we did not see a whole lot of intra-individual change in CRP over this 4-year period, so there was no longitudinal mediation in these data, but there was evidence for mediation in the baseline data, where we had a lot of inter-individual variability. So specifically, those participants who reported more frequent experiences of discrimination also had higher CRP, which in turn was related to lower initial memory performance. So some preliminary evidence that inflammation may be one physiological mechanism by which racially patterned social stress might get under the skin. But the time scale of these processes is not yet clear. In addition to how discrimination might get under the skin, we're interested in how it might get into the skull. Here I'm showing you a within group study of 221 non-Hispanic Black older adults in New York. And I'm showing a longitudinal association between the day-to-day experience of discrimination and white matter hyperintensities. White matter hyperintensities are an MRI indicator of cerebrovascular disease that's related to inflammation. And as you can see, those older Black adults who reported more frequent discrimination showed a faster accumulation of this pathology over a 4-year period. Independent of white matter hyperintensities, this study by a graduate student at Michigan, Jordan Palms, showed that the day-to-day experience of racist events is also associated with lower white matter integrity. This was found for multiple white matter tracts that are critical for cognitive function. Here I'm just showing the adjusted association for the cingulum. So together, these studies point to physiological mechanisms by which social stress might influence cognitive health, involving inflammation and inflammation-related brain changes, which we know are critical for cognitive function, particularly among Black older adults. I want to pause here for a minute because so much of the work on racial inequalities in cognitive aging focuses on risk pathways. But we know that this relationship between race, this complex sociocultural construct, and cognitive aging really reflects the sum of both risk and resilience pathways. And this is because social groups also cultivate norms and behaviors that can be health promoting. So by using race as a simple proxy for racism, or by focusing only on risk pathways, we won't be able to see the whole picture. So I'll show you just one example now of considering both risk and resilience in a model of cognitive disparities. This study was led by another graduate student, Emily Morris, and a former postdoc at Michigan, Dr. Lauren Brown. And here Emily was interested in extending Dr. Brown's work on stress appraisal and mental health to cognitive health. So together, using data from the HRS, they showed that older Black Americans report being exposed to more chronic stressors than whites, and in turn, being exposed to more chronic stressors is related to lower cognitive health. But those same Black older adults also appraised stressors as being less upsetting, showing evidence of adaptation and coping related to historical and contemporary lived experiences. And appraising stressors as less upsetting was in and of itself a unique predictor of better memory, independent of stress exposure. So I think this provides some nuanced evidence of both risk and resilience for cognitive aging among Black older adults. Now, I want to talk about some limitations to this work on discrimination. One of which is that Everyday discrimination measures might not be measuring the same construct across groups. So we've seen quantitative evidence of measurement non-invariance from Tene Lewis and others. This more recent qualitative study interviewed people as they completed the everyday discrimination scale, which is probably the most common measure of discrimination used in this work. And they showed that people use different cognitive schemas or different frameworks for interpreting and responding to the items on this scale. So some people approach the scale through a more general lens of negative social interactions, while others approach it through a more specific lens of social inequality or racism. And these differences in use of schemas corresponded to socio-demographic differences. So specifically, more white participants in this qualitative study described the use of a more general negative social interaction schema, whereas individuals with marginalized identities were more likely to describe one of these more specific schemas related to social inequality or racism. Now, this presents a problem to quantitative research on group differences. These between-group studies might not be measuring what we think we're measuring when we're querying discrimination experiences. And this is one reason why a lot of our work has really moved more toward within-group studies to understand how discrimination might influence cognitive aging. Another limitation is that these everyday discrimination measures focus on overt experiences at the interpersonal level. So they don't capture all types of discrimination, including institutional discrimination that might get transmitted through policies, vicarious discrimination experienced through social networks or through the media, or other insidious forms of discrimination that all could influence cognitive aging through stress pathways or through other pathways, like access to resources. So to address some of these limitations, I'll talk about some of the directions we've taken. One direction is to seek out alternative individual level measures that might provide a broader picture of how an individual is impacted by a broader array of environmental exposures. So perceived control is a psychological construct. It comes out of the social learning theory literature. To which people feel they have control over important life outcomes. It includes separable dimensions of internal control, which is akin to mastery or self-efficacy, and external control, which reflects the perception of environmental constraints that limit one's instrumentality. We have decades of social science research showing that these control beliefs are shaped by the environment. So even though it's an individual level measure, if we want to influence it, we've got to influence it through the environment. And it shows clear social gradients. So in the case of racial differences, we typically don't see racial differences in internal control. That seems to be more shaped by the microenvironment, family and friends. But we do see pretty large racial differences on external control, which appears to be shaped more by the macro environment, these larger social structures. So measures of external perceived control in particular might be helpful for gauging that individual level impact of a broader set of environmental exposures, including interpersonal discrimination. Remember, external control was a mediator of some of the effects of interpersonal discrimination in the first HRS study that I showed you, but also experiences across different ecological levels. Moving beyond the individual level now, we've also sought out measures that directly quantify exposures at other ecological levels, including the neighborhood level. This is exciting because neighborhood level exposures might be more actionable and more amenable to policy changes and interventions. So I'll show you some examples of studies using these different directions for this research on racial inequality. So this study was interested in characterizing the social determinants of cognitive disparities in a decomposition framework. So specifically, we were interested in the extent to which Black, white, and Hispanic white disparities can be attributed to specific socioeconomic and psychosocial factors. Not surprisingly, the socioeconomic factors explained the majority of these disparities, so up to 60%. But above and beyond the socioeconomic factors, perceived control explained an additional 7 to 8% of these disparities, while these two commonly used measures of discrimination were not contributors. And that's because In this sample, neither of those discrimination measures was related to cognitive health. And in the case of Hispanic white disparities, Hispanic participants in this sample did not report higher levels of discrimination, though they did report higher levels of external perceived control. So some evidence that these measures of external perceived control might capture those psychosocial experiences of different marginalized groups that might be relevant for cognitive aging. Moving beyond the individual level now, here we operationalized neighborhood racial income inequality as the ratio of median income for Black white census respondents versus Black census respondents. This gives us one measure of structural racism at the census tract level. In this particular study, which takes place in and around Detroit, we saw that white families were reporting on average 73% higher income than Black families. This is just a little national average, which has not changed since the 1960s. But importantly, above and beyond individual SES, overall neighborhood SES, neighborhood population density, the racial composition of the neighborhood, geographic clustering, we saw that greater neighborhood racial income inequality was related to poorer cognitive health. And interestingly, this association was not moderated by individual race. So this cohort is racially balanced. It's about 50% Black, 50% white, and we saw the same negative association in both Black and white participants. So this kind of universal effect of neighborhood racial income inequality suggests that inequality might be bad for everybody in the neighborhood, and we're really interested in interrogating some of the social and behavioral mechanisms underlying these associations. One more example of the importance of considering neighborhood conditions. This comes from Dr. Kathleen Soul, who was interested in extending the literature on cross-sectional associations between neighborhood socioeconomic disadvantage and cognitive health by incorporating longitudinal trajectories of neighborhood disinvestment. So using a latent class analysis of 17 years of census data, Dr. Soul showed evidence for 3 categories of neighborhoods, neighborhoods with low but increasing socioeconomic disadvantage, shown in green, neighborhoods with moderate and increasing socioeconomic disadvantage in blue, and neighborhoods with persistently high socioeconomic disadvantage in red. And she then looked at whether residing in these different types of neighborhoods helped to understand racial differences in cognitive health. So here she found that Black participants in this cohort study were more likely to live in neighborhoods characterized by persistently high or moderate and increasing socioeconomic disadvantage, but only living in neighborhoods with moderate and increasing socioeconomic disadvantage was related to poorer cognitive health. So this tells us that taking a snapshot of a neighborhood at one point in time might not tell us the whole story. Dynamic changes in neighborhoods related to disinvestment, displacement, gentrification are all going to be important for understanding how neighborhood social conditions affect cognitive and brain aging. So just to give you some summary points from the talk today, I showed some evidence that discrimination may be linked to cognitive aging through psychological and physiological stress pathways in line with Rodney Clark's biopsychosocial model. But measures of interpersonal discrimination can't tell the whole story about how racially patterned environmental exposures get under the skin and into the skull to affect brain and cognitive aging. We also need to consider resilience factors, which might also be racially patterned. With regard to alternative individual level measures, we saw that external perceived control might be a helpful measure of the impact of a broader array of environmental exposures, including but not limited to interpersonal discrimination. Moving beyond the individual level, we saw that neighborhood conditions appear to be important for cognitive aging and might be more actionable and relevant for policies and interventions. And if we really want to improve population health and advance health equity, we need to look at even higher levels, consider larger social structures. I want to take time to acknowledge the thousands of participants who take time to participate in the studies that I presented today. This work could be done without me, but it can't be done without them. I also want to acknowledge some key collaborators on these studies, as well as generous funding, particularly from the National Institute on Aging. So thanks again for this opportunity.
Thank you, Laura. And next up, we have Dr. Miguel Arce Rentería to speak to us about bilingualism.
All right, hello everybody, good morning. All right, I'm very excited to be here. Thank you to the organizers for the invitation. So I'll be talking about the work that we do in my lab. We're trying to understand how the social and environmental factors associated with bilingualism impact cognitive aging, and as well trying to disentangle that from the potential cognitive mechanisms of bilingualism itself. And so first, diving into it, right, like we know that there's plenty of research that supports that the engagement in cognitively enriching activities is associated with a reduced risk of dementia, better cognitive aging, better cognitive outcomes overall. For that, we can think about like what we know about education, quality of education, literacy, occupational complexity. But you can think of bilingualism as well as a cognitively enriching activity. And some studies have been reporting this quote-unquote bilingual advantage on cognition, meaning that bilinguals outperform monolinguals on aspects of executive functioning, episodic abilities. These results have been found in children, young adults, older adults, but obviously with everything in science, there's plenty of inconsistent findings. The idea, right, is that the action of like learning the language and then using the language, of the inhibition and switching between languages, can potentially strengthen attentional and executive control neural networks that may lead to domain-specific and domain-general benefits. However, in terms of what can potentially be explaining some of these discrepancies, there's plenty of methodological differences. First of all, you would be surprised with just how inconsistent the definition of bilingualism is. And then for those of you who are in the room who are bilingual yourself, you'll know it's not just cut and dry as yes or no, right? Some studies are very strict in their definitions, for some others are very loose. And a key thing about that too, it's also what about bilingualism may potentially about it itself can provide a benefit, right? Is it the age of acquisition? Is it how you're using it, how you're proficient you are in the language? In addition to that, there's plenty of also inconsistent control for potential confounders. But what's really important to know, right, that in terms of the context, bilingualism isn't randomly assigned, right? Doesn't happen in a vacuum. There are certain lived experiences that increase the person's ability or opportunity to become bilingual, which are associated with late life brain health. So these differences could explain inconsistent results across studies. So I really want to walk us through first through my conceptual kind of framework, how I approach this research in terms of these factors that complicate or enrich the study of bilingualism and cognitive aging. Right, we know that we have aging, right? Stuff happens to the brain and then it impacts the outcome that we're interested in, right? Dementia, cognitive decline, cognitive functioning. And bilingualism happens somewhere in a person's life, right? And then it can provide some benefit from either limiting the impact of aging on actual brain structure or impacting the effects of actual brain health on your cognitive outcomes. You're thinking brain reserve, brain maintenance versus the cognitive reserve. But then in addition, there's plenty of childhood contextual factors, adulthood contextual factors, like some of the ones that my colleagues have been presenting today, that are associated with, well, becoming bilingual and with migration history too, which would happen at any point in a person's life. And like I was mentioning, all of these factors have been associated with both brain and cognitive outcomes in late life. So we really need to keep all of these in mind with the bilingual population you're interested in working with. And then lastly, right, what about bilingualism? We need to get more detailed characterization of that. And so I'll be highlighting some of these key factors across different studies. Specifically, also what's important that seeing how then this relates across bilingual populations from diverse parts of the world. A lot of the research primarily really comes from the United States or just high-income countries. So I'll be showing about work from the Offspring study in New York, the Mexican Health and Aging Study, the MEXCOG study in Mexico, and then the Longitudinal Aging Study in India, Diagnostic Assessment of Dementia, LACIDAD, based in India. So first talking about Offspring study. So this is a community-based study of participants who are middle-aged, residing, they came from the northern part of Manhattan. You're thinking Inwood, Washington Heights. PIs are Dr. Jennifer Manley and Adam Brickman. For this study, participants are middle-aged, but I'm focusing on the subsample of participants who are Latino, Latinx, so they're age 30 and above. And for that, to try to account for the issues of just what languages they know. So these are going to be English, Spanish bilinguals. and then who completed a language history questionnaire where we're able to ascertain their bilingualism in different aspects, such as the language, how proficient they are in their second language, which in this case it would be English, in terms of speaking, reading, writing, understanding, and what age they had when they started to learn it. And all participants undergo a comprehensive cognitive assessment. What I want to show here already, like seeing how important it is to consider the context, right? We're working with around 980 participants, and you're seeing already that I'm grouping them into 4 groups, right? We have 1 monolingual group and 3 bilingual groups, really separated by whether they were tested in Spanish or English, and whether they were immigrant or US born. Some of the key things to point out is that we only have 1 monolingual group. And if you're monolingual among middle-aged Latinos in northern Manhattan, you're tested in Spanish and then you're an immigrant. You were born outside of the US. For something I forgot to mention, most of the Latinos here are Dominican. And then the other group to mention is that among those that are Latinos who are US born, they're all bilingual and all prefer to be tested in English. So the immigrant group, they're the ones that flipped between some prefer tested in Spanish, some in English. As you can see in terms of just some of the socio-demographic factors, it actually goes from like left to right, where left is the most disadvantaged group, the monolingual group, to the US born being the more advantaged group in terms of age, from older to younger, education, lower to higher, and even for parent years of education, which we're using a proxy for childhood SES, which we see lower in the monolingual group and higher in the groups towards the right. And in terms of their linguistic characteristics, it makes sense. Those that were tested in English have the highest proficiency in English compared to those tested in Spanish. And what's interesting too is the age of acquisition of their second language. So the group that were immigrant group tested in Spanish, they really started learning English in the 20s. Those that were tested in English, they reported learning English more when they were around 10 years of age. Then those that were US born, well, obviously when they were kids, around 3. So what I'm going to show first here, I'm showing the estimates and confidence intervals of the association between being bilingual compared to monolinguals. So grouping across all these 3 different categories, adjusting for the variables below, across all the different the tests in our battery, which are largely grouped between like memory tests, attention working memory, language and the semantic letter fluencies, aspects of executive functioning, and a processing speed test. As you can see here, bilinguals are performing better than monolinguals across several of the attention working memory tests, language tests, and different tests for executive functioning, like visual working memory and list sort, and set shifting and card sort. But then you have to wonder then, what about the groups that I was just describing? Is the association of bilingualism to cognition the same across all the groups, or certain groups benefiting, quote unquote, more than the others? And here I'm showing the different groups, again, how they compare to monolingual performance, and you can see between all of them too. The key things to mention first is the orange group. Those are the ones tested, the US-born group, which were tested in English. As you can see, they're the ones that are performing all the groups in several of the domains that we just talked about. The next group to mention is the blue group. So those are the immigrants tested in English. They're the next group that are performing better than monolinguals on several of domains that we talked about, mentioned before, but they're not different from the immigrant group tested in Spanish. And then the immigrant group tested in Spanish really only differ from the monolingual group on a test of semantic fluency and card sort, a set shifting task. And then when we go back into, okay, what about bilingualism? Looking only within the bilingual sample, let's look at these characteristics such as age of acquisition. Here what I'm showing is that a later age of acquisition is associated with worse performance. So we were mentioning from before, but as you're noticing here, we see also memory is starting to pop up. On the right, now we have basically like the mirror image, right, of like proficiency in English, they're L2, where we're seeing greater proficiency in English is associated with better performance across several of the tests, which makes sense, right, because a lot of the times proficiency is associated with age of acquisition. The younger the age of acquisition, the more likely to have higher proficiency. But then let's break it down between the groups. Within all the groups, you'll notice that for age of acquisition, it's really primarily driven among the group that was born in the US, which is also not terribly surprising because they are the ones that had the earliest age of acquisition. And we see some association with the immigrants tested in English, but really age of acquisition didn't play a role among those that were tested in Spanish. However, for English proficiency, basically all the groups are performing better compared to monolinguals with greater degree of proficiency in English. So we're seeing English proficiency is more associated with cognitive performance than age of acquisition in this group. And so just to wrap up first the results of the bilinguals in the US, so right, it was associated with better cognitive performance across several domains, and characteristics such as age of acquisition, the proficiency, was associated with performance across different domains, but obviously these differ by the immigrant status and language testing, right? Getting back to the social and environmental factors associated with bilingualism, where the greater quote-unquote benefit, it was among those that were US born, followed by those that were tested in immigrants tested in English, and then lastly, those that were tested in Spanish really outperformed monolinguals in fewer domains. And now, this is a group of Latinos living in the United States. You know, a good chunk of them are primarily immigrants. So now, how does this relate to bilingualism in a country primarily of Latinos, like Mexico? And so for that, we're taking advantage of the Mexican Health and Aging Study, which is a sister study to the HRS that was just presented about from my colleagues. And in 2015, they launched a subsample of a comprehensive cognitive assessment study of aging, a MEXCOG. And so I'm leveraging the MEXCOG study, Wave 1, at 2015. And what's exciting about Mexico that I don't think a lot of people know, that we do have around 7 million speakers of indigenous languages, and over 15 million people report speaking English. And so what we're doing here, it doesn't look that good, I think, but we're categorizing the groups here between urban and rural regions because we really had two types of bilinguals in Mexico. Those that acquired English as another language, or those that acquired who were indigenous language bilinguals. Right, so either Spanish in an indigenous language or Spanish in English. And the Spanish-English bilinguals were really predominantly found in urban areas, where in rural areas, which wouldn't be surprising, that's where we found the indigenous Spanish language bilinguals. And so we're stratifying the analyses by both groups, adjusting for relevant covariates, and then looking at their performance on the cognitive battery. So in the urban area, among comparing Spanish-English bilinguals to Spanish monolinguals in Mexico, there was no association. There was no difference between they were bilinguals or monolinguals in that area. However, when looking at the rural group, looking at the Spanish indigenous bilinguals compared to Spanish monolinguals, we see actually a disadvantage. We're seeing that they're performing worse compared to all other, compared to the monolingual groups in a Spanish monolingual group in a rural area, right? So I think what this is highlighting really, right, it's not necessarily that you're thinking that, there's something bad about an indigenous language, right? Is that we're thinking, what about the environmental factors that are associated with these individuals that maybe we're not completely accounting for in this study right now? And as well in terms of thinking about the validity of the evaluation too, right? They're all tested in Spanish. There's no opportunity to be tested in an indigenous language like Mayan or Nahuatl. And we're thinking really how valid are the tests among these individuals. Like for instance, we don't know their degree of proficiency in Spanish, which these more detailed characteristics of bilingualism, we're going to be now collecting that in this year and next year in the next waves of MHAS, which I'm excited about, to really trying to now also look at these different linguistic profiles among both groups as we were doing it in the Offspring study. And so something also then got me thinking about, for instance, for that study, right, is some of the differences, right, from the Latinos in the United States, which were Dominican, primarily immigrants, and the Latinos in Mexico, Mexicans, right, I'm Mexican myself, is that, well, in Mexico, right, they were learning a minority language. Those that were acquiring English, that's a minority language, versus the ones that acquired English in the US, that's a majority language, right? So it got me thinking, like, does that have anything to do in terms of how it relates to cognitive functioning? And luckily enough, in India, that provides us a pretty good opportunity to try to evaluate that, given that in India, it's really linguistically diverse, and really what's cool about that too is that multilingualism is kind of more equally distributed across different socioeconomic gradients in India as opposed to in different countries. And so for that, I'm leveraging the LACIDAD study, which is also a sister study to the MEXCOG study, sister study to the HRS HCAP study in the United States. So they have very similar methodological designs. What I'm going to show here is work that's being led by my postdoc, Dr. Iris Strangman, in terms of categorizing the participants in terms of whether they're monolingual or they're multilingual and their first language was a majority language or their first language was a minority language. So then their second language one. And so here we're just showing in terms of highlighting the linguistic diversity, right, is that for the monolingual groups, also all participants were speaking the majority language for monolinguals of the region. Then we're seeing the different languages that it's their L1. For the multilinguals, who their majority language was the first one, these are the languages that are popping up, pretty similar to the monolingual. And then for the multilinguals who their first language was a minority language, you're thinking here those are the Latinos in the US, for instance, right, that they're learning. Their Spanish is their first language, a minority. And versus the previous group, that's the Mexicans in Mexico, right, who are learning English. We're seeing all these different languages pop up. And actually, we're seeing some languages popping up that are not actually a majority language anywhere in India, like Rajasthani and Urdu, And so what's also cool about this work is we're looking at it across SES as well, across stratus of SES. So what I'm going to show you here, we're seeing the different cognitive domains across all the figures, and we're seeing the three groups, monolingual, the minority multilingual, majority multilingual group, and then across SES strata. So like in red, we have the low SES, in green, we have the high, medium SES, and in blue, we have the high SES. And these are model strata adjusted for the other relevant covariates. I think what's really interesting to start showing from the beginning is that it didn't matter, at least in this sample, whether they were a minority, majority language, first language speaker, multilingual, it didn't really differ between them and between those groups across all the domains. However, what you can clearly see that there was a strong association of SES, right? As SES increased, we see better performance across all the groups. However, really, in terms of the multilingual type of difference, we really only see that among people who have medium SES to high SES. If you're seeing among those that had low SES, there's really no difference, whether they're multilingual or not, compared to monolingual participants in terms of performance on executive functioning. So really, we're getting no real strong association with bilingualism with cognitive performance on low SES. But also, I think what's really cool to then highlight is really the important role of SES, because even though, however, right, in medium SES, the medium SES multilinguals are performing better than their medium monolinguals, SES, and then the same way in the high SES compared to the high SES monolinguals, still the high SES monolinguals are outperforming medium SES bilinguals. Right, so really showing that it's not necessarily just that clear cut of the association between multilingualism and these environmental factors on cognition and how the role the SES plays on cognitive functioning. And so, to conclude, the association between bilingualism and cognition seems to differ across nationally, right? It's not really that clear cut that it definitely provides a quote-unquote advantage. In the United States, bilingualism was associated with better cognitive functioning across several domains, but we did see that differ across these different socioeconomic, the social groups that we characterize, right, between being born in the US and whether they were tested or not in English or Spanish. But we also started to see how aspects of bilingualism are associated with cognitive functioning, such as greater bilingual proficiency, age of L2 acquisition. For the sake of time, I didn't show the results of active bilingualism, whether they were using two languages every day compared to only one language, and we also found an association of that on cognitive functioning, but in fewer domains, really only on episodic memory and processing speed. In India, multilingualism was associated with cognition, right, regardless of what, where they were a minority language speaker or majority language speaker, but really we only saw that among those with medium to high SES. And then there was no evidence of a bilingual cognitive advantage among older adults residing in urban and rural regions in Mexico, right? So really indicating how these social and environmental factors associated with bilingualism are associated with cognitive functioning, and really trying to see, well, some aspects of bilingualism do seem to be associated with cognitive functioning by themselves, like the linguistic aspects. But we really need to do more work to see separating these things further and then following them over time, to really seeing how this relates to cognitive decline. So with that, I'd just like to acknowledge the different teams that I work with, the people are the co-mentors, my co-mentors at Columbia and my lab at Columbia, and our La Ciudad team, XHOC team, and all the studies. Thank you.
Okay, and thank you, Miguel. And in the last talk from our session, we have Katrina Walsemann, who's joining us remotely, to speak about educational inequalities.
Good morning. Today I'm going to share some of my work looking at the relationship between school inequities and cognitive function in the pre- and post-Brown U.S. South. Before I begin, I want to acknowledge the Alzheimer's Association and the National Institute on Aging for supporting this work, and of course my collaborators and research assistants for the valuable expertise and work that they provided to this project. In the United States, large and persistent Black-white inequities on tests of cognitive function, memory, and executive function are well documented. Research also consistently finds that educational attainment is one of the most important protective factors against poor cognitive performance, but is often not related to the rate of cognitive decline documented at older ages. Because Black older adults on average completed fewer years of school than their white counterparts, educational attainment is one of the most common explanations for why Black older adults report worse cognitive function than their white counterparts. However, prior work typically finds that educational attainment explains only about a quarter of the Black-white inequity. One reason why education may not do a great job explaining Black-white inequities in cognitive function is because the school experiences of Black and white older adults were vastly different. Nowhere are these differences more evident than in the Jim Crow South. Before 1954, racially segregated schools were mandatory in all southern states, and you can see that on this map in the dark purple that's outlined in white. This de jure school segregation led to large racial inequities in educational resources, particularly as it relates to school term length and how many days of instruction students receive when they attended Black or white segregated schools. Importantly, almost 80% of Black older adults who attended school before 1954 were actually educated in the U.S. South. Thus, traditional measures of education, like years of education completed, may not accurately measure the amount of time spent in school where one could actually build cognitive reserve, especially for Black older adults who grew up in the South during Jim Crow. So these two box plots show variation in school term length by state and census division from 1920 to 1954 for white and Black segregated schools. And you can see here that there is significant state variation in school term length for both white and Black segregated schools, but that the variation is much larger among Black segregated schools. So, for example, variation in school term length during this period was quite large for South Carolina. It had a low of 80 days in 1920, and it was near a high of 188 days by the end of the period. Whereas Delaware always had quite long school terms for both the Black and white segregated school systems. So although the box plots help us understand the state variation in school term length during the period, they don't explicitly show how school term length actually changed over this time period. So these line plots provide that information for the South Atlantic Division to illustrate the general trend that school term lengths were shorter earlier in the period and lengthened over time. So by 1954, southern states had school term lengths that were about 160 to 180 days, and that's a length that's generally on par with the average school term length of 180 days that we have today in the United States. So just because states have a set term length doesn't mean that students are going to actually attend for the full term. So this multiple line graph shows the proportion of the school term that Black students, who are shown in aqua color, and white students, who are shown in purple, on average attended by state from 1920 to 1954. So you can see that there's significant state and time variation in this variable for both white and Black segregated school systems, but that Black students typically attended fewer days of school than white students. So again, we're going to pick on South Carolina, and you can see that there are large inequities between Black and white segregated school systems, very large in 1920. They narrowed by 1954, but were still present. But you compare that to Delaware, that had very small inequities during this entire time period. So for this first study, our main aim was to determine if accounting for state and racial variation in school attendance better explained Black-white disparities in cognitive and trajectories of cognitive function than using a measure of self-reported years of schooling. And we did this in a representative sample of Black and white older adults who attended school in the Jim Crow South. Our first step was, to address the theme was to extract historical data on Black and white segregated school systems in the 16 southern states and the District of Columbia that were under Jim Crow, as shown previously in the map of de jure school segregation. This data was reported in the 1920 to 1954 biennial surveys of education of the United States, and you can see kind of a picture of this historical report here on the slide. So we extracted two measures, the measure of school term length and the average days attended by students. These were reported separately by segregated school systems, and they include all public primary and secondary schools in each of the states. The individual level data comes from the Health and Retirement Study, which is a nationally representative prospective study of U.S. adults over age 50. And we linked this historical data to the health and retirement study using a single measure from the HRS that asked respondents the state where they lived when they were in school or around age 10. So once we linked our data, we restricted our sample to black and white HRS respondents who reported residing in the U.S. South when they were school-aged. They started primary school in or after 1919. They completed their primary or secondary schooling by 1954, and they provided at least one observation on cognitive function from 1996 to 2016. So our final analytic sample included about 4,300 older adults with almost 26,000 person period observations, so almost 3,000 white adults and almost 1,400 black adults. So to address our aim, we used two measures of education and compared how much of the black-white inequity in trajectories of cognitive function was explained by each measure. So the first measure is a traditional measure of self-reported years of schooling that we then top-coded at 12 because the historical data only reported school term length and attendance for primary and secondary schooling. But then to account for any attendance in post-secondary schooling, we did have a indicator variable, a dummy variable that we included in our models. The second measure is a measure of the duration of time that HRS respondents spent in school. And that measure modifies the self-reported years of schooling measure by taking into account variation in the proportion of the school term attended across southern states and segregated school systems during the years when our respondents were attending school. And we figured this out based on how many years of schooling they completed and their birth year and the state that they were born in and their self-reported race. So when using the historically informed duration in school measure, both black and white older adults spent fewer years in school than suggested by their self-reports. So this was about 2.5 to 2.8 fewer years in school for white and black adults respectively when using this duration of school measure. Okay, so then once we did the linkage and we created our measures, we then estimated cognitive trajectories. We adjusted for a number of covariates that you can see listed there on the bottom of the slide. This figure plots the average adjusted predicted values of cognitive function across age for white and black older adults. And what I'm going to do is show the difference in cognitive function between these two groups at age 95, just for the ease of presentation. But it's important to note that the size of the gap is pretty stable across age because the rate of decline didn't vary by race. So here we see that the black-white disparity is 3.5 points on this cognitive function scale. But once we include in our models the traditional years of schooling measure and that degree attainment indicator variable, the disparity reduces to 2.3 points. So a traditional measure of education explained about 34% of the race disparity. Note also that the adjustment almost entirely occurs because black respondents' level of cognitive function is higher after education is included in the model. So as expected, part of the reason why black older adults have lower levels of cognitive function than white older adults is because of lower educational attainment. Okay, so then we removed that traditional measure years of schooling from the model and included our new duration in school measure that accounts for these differences in school term length across states and time and segregated school systems. Now, in these models, the disparity is 1.76 points. So duration in school explains about 49% of the black-white inequity in level of cognitive function. So overall, the same pattern that we found for cognitive function, we also found when we looked at episodic memory and mental status. So years of schooling explained about 39% and 35% of the black-white inequity in episodic memory and mental status. But in comparison, our duration school measure explained 57% and 48% of that black-white inequity in those two measures, respectively. So consistent with prior research, we found that a traditional measure of education only explained about a third of the black-white disparity in level of cognitive health. And this seems to be due in part to the fact that traditional measures of education do not account for the severe racial inequities in the education system that were in place during the Jim Crow South, which resulted in black students spending fewer days in the classroom being educated. Our measure, duration in school, better aligns with the amount of instruction students likely received and may tap into those skills that build cognitive reserve early in life and preserve cognitive performance later in life. It also accounts for institutionalized racial inequity in the amount of instruction provided to Black and white students during the Jim Crow era, and in so doing allows us to more accurately adjust for race differences in the quantity of education. So what happened to schools after 1954? The year the Supreme Court declared in Brown versus Board of Education that separate but equal schools were unconstitutional and ordered states to desegregate their schools with all deliberate speed. Because the Supreme Court did not define all deliberate speed, this resulted in delayed school desegregation across many of the southern states. So to show you how states varied in their timing and level of school desegregation during this time period, I plotted in this multiple line graph the percent of Black students attending school with any white students by state for the years 1955 to 1974. And as you can see, the well-known delay in desegregation is evident in most of these state graphs. Only a few states began integrating schools in the late 1950s. Most waited until after the 1964 Civil Rights Act prohibited federally funded programs, including schools, from discriminating on the basis of race. So our second study aimed to exploit that state and period variation in school desegregation efforts in the U.S. South in the 2 decades after Brown to determine if one's expected exposure to desegregated primary schooling was associated with midlife cognitive health among Black and white Americans who grew up in the South during this particular period. Our study builds on a small but growing body of evidence that generally finds attending desegregated schools is positively associated with measures of cognitive function among Black older adults. This work has mostly been conducted on small community samples that have generally limited generalizability, and all of the studies have relied on self-reported measures of school desegregation. So, for example, one of my recent papers used the Life History Mail Survey in the Health and Retirement Study and found that Black adults who ever attended a race discordant school, which we define as mostly white, had higher scores on cognitive function, episodic memory, and mental status at age 65 than Black adults who never attended a race discordant school. And we found no association between attending a race discordant school and cognitive outcomes for white adults. And this is generally consistent with the social and econ literature that looks at some of the social and economic outcomes of school desegregation for these cohorts of Black and white adults. Another recent study also suggests that the timing of exposure to integrated schools may matter for cognitive cohort of Kaiser beneficiaries. The authors found that Black adults who attended integrated elementary schools had higher Z-scores on executive function and semantic memory than Black adults who had never attended integrated schools. But they only found this relationship for attendance at integrated elementary schools, so they didn't find this for middle schools or high schools. So to answer our questions, we extracted historical administrative data on the extent of school desegregation in the U.S. South from the Southern Education Reporting Service and U.S. Census abstracts. To assess state-level desegregation during this period, we use a measure of the percent of Black students in the state that were attending schools with white students. To try to capture what was going on in these states during this period, we created an estimate of students' expected exposure to desegregated primary schooling by summing the proportion of Black students who were attending schools with white students in each year across their 6 years of primary school. So we did this for each school entry cohort and for each state. So this state-level estimate can loosely be thought of as the expected years of exposure to desegregated primary school, which ranged from 0 in a state that did not desegregate during the 6 years of primary school, to a state to 6 in a state where 100% of Black students were attending desegregated schools throughout their primary school. Here we show examples from 3 southern states, Maryland, Alabama, and North Carolina. The lines represent the baby boom cohort, subdivided into early, mid, and late, and the x-axis is the year a hypothetical student entered first grade, and the y-axis denotes the expected number of primary schools that hypothetical student would have attended desegregated schools during primary school. And you can see that students in that early boomer cohort in Maryland could expect to attend about 1 to 2 years in desegregated schools. But in Alabama and North Carolina, students in that same cohort would be basically expected to attend no years of schooling in desegregated schools. But that students in the late boomer cohort were much more likely to attend some of their primary school in desegregated schools. But of course, this also varied by state. So next, we linked that administrative data again to the health and retirement study. We restricted our sample to Black and white adults who were attending primary school in the U.S. South between 1954 to 1974. And then we estimated the level of cognitive function at midlife using state fixed effects models and adjustment for several childhood covariates, state education quality, and respondents' educational attainment. And we found that an additional expected year of exposure to desegregated primary school was associated with higher scores on cognitive function and episodic memory for Black adults, which translated to about 2.5 points of higher cognitive function and episodic memory scores for Black adults who had 5 years of expected exposures to desegregated primary school compared to 0 years. But we didn't find any association for this group for mental status, and we didn't find any relationship for white adults. So our results suggest that the pathways to cognitive health vary for key U.S. populations, and that within race analysis is critical for understanding these pathways. Years of school desegregation data and the use of a large representative sample of Black and white adults who grew up in the U.S. South in the 2 decades following Brown may also provide a more complete examination of the role of school desegregation for cognitive function, given our use of administrative data and also our use of a generalizable sample of older adults. Thus, our findings from both studies highlight not only the importance of historically situating Black older adults' lived experiences, but also measuring these experiences to determine how they shape their cognitive health. And then just to close, if you're interested in some of these questions related to education and dementia, the NIA funds our network on education, biosocial pathways, and dementia in diverse populations. And if you want to get involved in that, it's an interdisciplinary group of scholars, and we welcome your involvement. You can click the QR code, you can go to our website and just fill out our Get Involved form. So we'd love to see you there. Thank you.
All right. Thank you, Katrina. I know you're joining us via Zoom. So I am so excited about this discussion session. I hope all of you have been writing down your questions and your comments. It really is historical to have a session like this at the Cognitive Aging Summit. And I just want to take a minute to thank all the speakers for all of your work. We have been on Zoom a few times before the meeting preparing for this. And so I thought your talks were incredible and really just inspiring for me personally as well. So, John, are you at the mic? Okay, do you want to,
Mic is even on. The mic is even on. It's on, yeah. Just one brief comment. I hate to disappoint AJ, but not everybody knows about the health and retirement study. More people probably know more about it after this session than before by a lot. But I just wanted to point out that in addition to the health and retirement study and the partner studies that actually Miguel also mentioned, I can tell you a lot more about those because I'm the project scientist for the HRS and for the network of those studies. So a lot more there than you heard about today. I'd be happy to talk with you. And this is my free commercial, and talk to you later.
And let me take credit and blame for AJ not going in more detail about the HRS. I gave her feedback. Everyone in the audience is going to know about HR, and more people are going to. So, okay, I'm going to call on people that I see, as they come to the mic. Go ahead.
Jonas Geda, can you hear me?
Yeah.
Jonas Geda from Barrow Neurological Institute. A great panel, a great discussion, and I just wanted to, particularly for AJ, When I was at Mayo Clinic, the dean and me, we wrote a paper on how to improve the admission of underrepresented minority students. And one of the key papers we stumbled upon was a review, I think, on American Sociological Society paper by Duster. And it was quite striking that at one point, for a long time in the United States, it was illegal to educate, basic literacy to a black person. So I don't remember for how long that duration was. So I think probably looking at that, some of the differences, in fact, in the presentation from the Michigan between minority groups, could it be traced to that type of exposure? Certainly it had a key role in our paper when we wrote our paper. So can you comment on that?
Face. Push the face. Thank you. As you know, I didn't know. So I have to disclose my bias that I tend to not think it's education, and this is where I tend to differ from a lot of cognitive researchers. I am of the belief that what education provided over history was access to good-paying jobs and access to resources through those good-paying jobs like healthcare, health insurance, retirement, so on and so forth. So I actually do not have the belief that however long you were in school or what kind of training you provided is protective against dementia or cognitive decline. But Katrina would be someone who does believe that and could speak to how a history of not having access to some of this content could then increase risk for cognitive decline. But I would argue that it's really lack of access to completion of high school diplomas that is really detrimental to cognitive health.
Katrina, do you want to add?
Sure. I think that one of the things to consider around education is timing in some ways. Well, I also agree with AJ that access to resources is probably one of the key pathways linking educational attainment to cognitive function and dementia later in life. I also think that exposure to high quality education matters early in life for brain development and cognitive development that can set the stage for all of those other things like during midlife and later life. So I would say that, yes, some of the work that I have done has shown that some of these inequities, these institutionalized inequities, do seem to matter and could help to explain why we are seeing some of the black-white inequities in cognitive function that we do document among different cohorts of older adults.
Okay.
Hi, thanks. This was a very interesting session. One thing I'm kind of curious about that, maybe Miguel alluded to, but it didn't come up much, is the validity of a lot of the cognitive measures that we use, because a lot of these are made, with certain populations in mind, might not be valid to the diverse populations we're looking at. And I'm just kind of curious some of your thoughts of both the issue and maybe also some of the solutions to the problem.
That's a wonderful question. Thank you for bringing that up. And I do think, because for instance, from the work that I was showing from the MEXCOG study, right, I think that's getting at that, right? In addition that we saw the such a lower performance among the indigenous population that was tested in Spanish, that how much is that being driven by that, right? Because at least I know, for instance, among the Offspring study and the study in La Ciudad, both of those studies, they've undergone rigorous, evaluation of the validity of the test in English and in Spanish and Offspring, evaluating it in terms of, the measurement in variance for language, and as well, and in India as well. In India, in the La Ciudad study, participants can be evaluated in 11 or 12 different languages, and the battery was carefully constructed to be used in these different languages, and we show measurement in variance across those different languages. So I'm a little bit less concerned there, but yes, but it's still, it doesn't hold the same for all the domains. domains, right? Certain domains are going to be much more culturally impacted than others, although all of them are. But I do think it's really important, especially when we're working with bilingual populations, right? What language should they be tested in? What's their most comfortable language? And how are our tools valid in terms of linguistic and cultural appropriateness for measuring what we want to measure in this population? Thank you.
I think you were in line before, and then I'm just going to go around. So go right ahead with your question.
Thank you, and thank you so much for an amazing panel. I'm a medical anthropologist, so it's very inspiring to hear social and structural inequalities and social and structural factors being mentioned, and with such rigorous research behind them. At the same time, I can't help but reflect from how many decades we've been talking in the social sciences about social and structural factors in health. So it's not actually surprising to hear them in the area of cognitive aging. I think what it makes me reflect on is deeper questions about the role of interventions and what are considered to be valid interventions, why we don't have more structural interventions, and why it is that certain intervention development models, such as just one of them, intervention mapping, and others that so many public health students are learning in their intervention design classes, say that at the time of developing your interventions, structural factors are usually not changeable. And so as people like Paul Farmer always said, is what is changeable is also a social construction. So how do we get to the point where we're discussing new intervention development models at the structural level? What role does NIH and other funding agencies have in creating funding lines for us to be able to intervene at the structural level without having to say we are also moderating or primarily moderating at the behavioral or micro level of interventions when we have over and over decades and decades of science showing that structural factors are fundamental causes of health. So again, not surprising that we would see it in the area of cognitive health. So any of you, I was most inspired by Dr. Adkins-Jackson's talk when I was reflecting on this, but any of you really could address that question if you so desire. Thank you.
Sure. So my mentor, Dr. Derek Griffith, talks a lot about expanding how we see precision medicine, because often the role is to minimize the noise, the social factors, into looking underneath the skin of the individual and tailoring an intervention. But what if you considered those structural factors to be a part of that precision medicine? What if you knew that this is a county that disproportionately incarcerated people racialized as Black? So what kind of public health interventions would you target for a place like that? How would you talk to people when they're in clinical settings about that, instead of saying their risk factor is that they're Black? What if it's their risk factor that they lived in a context at which they might have had to flee because a family member was lynched? How can we reconfigure our assessments to capture these variables? How we can reconfigure precision medicine to then target structural interventions? So then we can get to the fun of the individualized stuff, but we have to tackle the first thing first.
Others want to weigh in? I'll say that, we have had a number of natural experiments that, you've seen some of those data presented just now, and there's more out there that, weren't presented. These are ways of, modeling what an intervention would look like. There's a really great paper. The first author, I'm forgetting their name, but it presents what would happen using HRS data if patients to mortality, Black-white mortality differences. And so, who is it?
Himmelstein?
Himmelstein. Look it up. Really good paper. And so, we have the opportunity, I think, to take advantage of some of these policies that have changed across states, across time. But I agree, and thank you for that sort of rallying comment.
Thank you so much to this panel, which absolutely blows my mind the science is clearly developing, but there are so many things that you're talking about that we understand from culture relating to economic issues, relating to cultural factors. And so even though you can't fix everything right now, if I ask each of you, what are the public health recommendations you would pick? One, could you say what you think we should do?
Let's start with, well,
you have 30 seconds each.
Anyone can start. Should I pick on Katrina since you were last to speak?
Sure. I mean, I'm in a school of public policy, so I always think now about policy, what policies could change. I'm doing other work in addition to the work that I presented here on educational quality and investment in schools. And I would say that one thing that I would kind of advocate for is equal access to high quality education as a key kind of way to prevent some of these later life outcomes. It's not just cognitive. I mean, we're talking about cognitive health here, but it also would tap into a lot of other health outcomes that we're all concerned about too when we're thinking about, population health. So that would be my recommendation.
Miguel?
Yeah, in terms of my recommendation, I would think from my work is the socioeconomic opportunities, the socioeconomic benefit. Because at least I think, from the theme across the different studies that I was presenting on, right, that we see that those that are most socioeconomic disadvantaged are doing the worst, right? Like our reference group in Offspring are the monolingual immigrant Spanish group, have more socioeconomic disadvantage. In Mexico, the indigenous population do the worst. And in India, the ones with the lowest SES, they were the ones doing the worst compared to everybody, right? And we're seeing this steady increasing gradient. So I think improving everyone's socioeconomic status and socioeconomic gradient, getting to what AJ was mentioning about the opportunities that can come with that too. I think that would help bring everyone's brain health up.
Yeah, I think to answer this question, we have to think both about what's the biggest contributor to these disparities, and then also what is most tractable as an intervention target. And so I think we have seen that education and income, these socioeconomic factors, are the biggest contributors. And as far as what's most actionable, finding policy levers, which I think most naturally exist at the school level and relatedly at the neighborhood level in terms of investing in communities rather than individuals, which might be more difficult to legislate.
I completely disagree with all the socioeconomic stuff. And here is why. Because there's robust literature from within group studies that show that Black middle class communities experience some of the worst outcomes. And so you would think that they're going to perform well on a lot of health outcomes because they've quote unquote made it. But the pressure to even get to that level or stay on that level is so extreme that it's deleterious to the body. So I'm not sure I want people to do that. I would actually advocate for restorative justice interventions because in a lot of the factors I look at, we don't know if historical lynchings is picking up on fear, anxiety, trauma. Like it's picking up on so much that imagine what a restorative justice intervention will give someone who survived this. Not only would it may provide reparations that could economically help, but also the healing of that apology and recognition that you're not crazy, that you really were taunted and traumatized. I would advocate for that.
Mm-hmm. I love that you brought up that literature about middle and upper class Black older adults. And there's such a rich literature on higher levels of discrimination experienced among that group. And, a lot of that does seem to be linked to work environments and neighborhood environments. So, I think one of the challenges to neighborhood research has really been to identify the counterfactual. Are we taking people out of neighborhoods and putting them in different neighborhoods? And I think that can create more of those stress. And I think in terms of intervening, I think it's more about putting resources into neighborhoods rather than moving people out of those neighborhoods, which might be protective enclaves. I mean, you know, Dr. Kathleen Soul's study I showed, there was no difference in current neighborhood socioeconomic status between the groups who had persistently high neighborhood SES versus moderate and increasing, but it was the ones who were moderate increasing that were doing the worst. And it may be that those who were living in persistently high neighborhoods had naturally developed resilience factors, and plucking them out of those neighborhoods is probably not the answer. So I think investing in the neighborhoods, meeting people where they are is probably the best way if we did want to intervene on socioeconomic status. Just wanted to add that.
Thank you.
Yeah, thank you for such a spectacular panel. So my question is for Miguel, out of curiosity, and so I understand that you in the U.S. study, you let them take the test in whichever language they prefer, right? So my question is, do you think there is value on making them take the test on both languages? And if so, would you expect a difference? Because, for example, the group of obviously for the immigrant, maybe for the U.S. one as well, but for the immigrants, in my personal experience, I think if they arrive to the country like in the first 10 years of life, probably the first language would switch. I don't know if you agree with that, but at least that. So anyways, do you think it's value on taking that? And if so, would you expect differences?
Thank you for that question. Yes, I think it depends, right? I think, in terms of, because you're making a good point towards the end about, right, that there is studies do mention that there can be change in language dominance, where exactly somebody, immigrates to the U.S., Spanish dominant, but then stays for different reasons, environments only English, they may become English dominant. Like I've experienced that myself, going from Mexico being the U.S., I go back and forth. So there might be, I think it's important for that to really characterize their linguistic background, to really understand that. What is their true dominance and how does that change over time? In terms of being evaluated in both, let's say in the offspring study, in English and in Spanish together, I think it'll depend, right? I think it'll depend on what we're trying to get at, because we don't want to necessarily test them in a language that's harder for them, then we're not really measuring memory, perhaps, right? We're getting more at now their proficiency in that other language that they're getting tested in. But there may be some value, though, to understanding some linguistic aspects, how they relate to both languages, because given that some studies have shown that individuals who may be at a greater risk of developing dementia, Alzheimer's disease, we see greater decline in their second language, in their least dominant language, and so it might be good to figure out how to best keep those markers over time. It may not be ideal both, like the whole battery in two languages, but maybe aspects of them or other aspects of linguistic actual abilities other than self-report that may be important to document over time to see how they change. Thank you.
Hi, Diane Bevenkamp, Bright Focus Foundation. So I would love for each of you to kind of maybe parse out some sex-based differences, because I know that structural and societal differences between men and women. In the 1980s, I remember in my math class, my algebra teacher telling me that none of the women in class could understand vectors because they couldn't think in 3D. And he didn't believe me when I argued I could understand it. So, that's 1980. So, could you give some more information to parse out? Because it's not all one society.
Sure. And you're thinking with me on this, because I don't know what to make of it. I really don't know. I collaborate with people like Tanisha Hill-Jarrett and Justina Avila-Rieger, who look at intersectionality in this work and show that often women racialized as Black have worse cognitive outcomes, particularly in memory trajectories, than women racialized as white. But in the studies I've done with HRS and with Monitoring the Future, which is an adolescent survey, when structural racism is in the equation, boys and older adults racialized, men racialized as Black do worse. And I don't know what to make of that. I don't understand it for the life of me. And quite frankly, I think I'm biased because I just refuse to accept it. I just keep looking at it. But we published both of those findings. I don't know what to think of it. So the next stage in the research is to specifically look at the racialized experiences of Black men and boys to figure out what's going on there, because it's weird.
I look forward to that research.
I'll take the opportunity to highlight some work of a graduate student in my lab, Kiana Scambray, who's very interested in intersectionality between race and gender identity. And she has some really interesting social resilience in cognitive aging. And, we didn't always find what we expected to find. I think some of the interesting things that came out of that work were that it seemed like Black women benefited less from psychosocial resilience, and that makes us think a lot about high effort coping, the strong Black woman schema and the type of coping that people are having to engage in order to manage stress. It may protect them in some ways, but it may be deleterious in other ways. And another finding that came out of that was that those who were doing worse were Black men in our Michigan cohort. And we really thought about what types of stressors were both racially and patterned by gender. I think bringing up AJ's great work about mass incarceration comes into play here, that, what we see is much lower socioeconomic outcomes among the Black men in our sample compared to the Black women, which is the opposite of what we see in the white sample. And I think that might be related to a lot of AJ's work in that space. So, I think that when we find unexpected findings, that's our best opportunity to really understand what's going on. Is it different stressors, or is it different coping skills?
I just wanted to kind of say a shout out, I guess, to one of my co-authors as well, Andrea Henderson. We had a paper published, I want to say, in JGSS a few years ago, but we were looking at the question of intersectionality, really, and religious spirituality, religious participation, and that we found, if I recall correctly, that that was protective, we found, for Black midlife and older adult women, but we, in terms of their cognitive function, we didn't really find an effect for Black men, and we found the opposite for white men and women. So more participation and more religiosity was associated actually with poor cognitive function among white men and women. So I think that might kind of tap into what Laura was just saying in terms of resilience, what aspects make certain people more resilient or not resilient. So.
Can I just add to this? I think this might be a prime opportunity for looking at critical periods of exposure, because thinking about the first panel and the topic on menopause and pregnancy, NAFTA in the 1990s that sort of took away the socioeconomic opportunity of indigenous farmers in Mexico and forced them into a situation where they had to migrate. These are individuals who might have been pregnant, having children. What was the impact of that exposure at that critical time on those women? And what are sort of the cognitive trajectories later? So this might be a ripe opportunity.
And it's very interesting. I think in the Netherlands, they have that Dutch winter hunger from World War II. That's another one.
Thank you for that question. A really call for all of us to consider intersectionality in our work. We should all know what that is. We should all know the principles of it. Joel.
Hi, Joel Salinas. I'm with the American Academy of Neurology's Brain Health Initiative. Thank you again for the session. I echo the excitement. I have so many questions, but I'll give my time to maybe bring the spotlight back to this point about interventions. I think from the group, if you had to pick a single barrier to be addressed to develop more interventions, what would that be? Is it harmonization of measures? Is it funding? Is it getting more people in the pipeline to do this kind of work?
I would probably like to see tailored interventions to spotlighted places that can be representative. For example, if there's a city that has a historic relationship to structural racism, then we might want to see a targeted intervention in a city like that other cities could then model themselves after. So then we would need to have the kind of funding that would allow for multiple community partnerships and multiple sectors to illustrate that kind of stuff. I'm a psychometrician, and I am largely not a fan of harmonizing instruments, because it just takes so much time, and I think it's a distraction, but I know y'all on the panel, so I'm gonna shut up.
I mean, I think one barrier is reaching out to communities and doing more community-engaged research. And I think most of the participants in these intervention studies are not representative of the population, and I think it can be hard to do community-engaged work. It's often more expensive and more time-consuming. So I think to address those problems, we do need more funding. And if you kind of look at where funding goes, often these studies are underfunded compared to more clinic-based clinical trial studies. So, I think we need funding in order to do specialty kind of research that will allow us to have representative samples in intervention studies.
Thank you. We plan on, highlighting many of the things that you mentioned, in our sum up of this conference.
So Luke Stokel from NIA, is this on?
Yeah.
Right, great. So I wrote mine down because I don't want to go off script here. So I also wear a hat as a clinical neuropsychologist, as Jen knows. And so my question is going to come from the traditional clinical neuropsych clinic, where people are evaluated at whatever point they're evaluated at right now. So we often don't know a person's baseline or so-called premorbid cognitive function. And in addition to things like education, we often use language-based estimates using vocabulary or word reading measures. Sometimes we'll use alternative nonverbal measures, like a matrix reasoning task or something like that, that are thought to have less sensitivity to variability in sociocultural influences. We know that's not true, but that's historically been what people have thought. So in light of these talks and based on information like what Jonas mentioned earlier, how should we rethink how we assess premorbid function in the context of clinical assessment? That's a big question, so if you want to say, this is the one thing I want to do. And my follow-up was going to be, how do we get to the 10-minute assessment, which everybody wants to include in the primary care clinic now. But I don't want you to get there. Thank you.
Miguel, do you want to try that?
Yeah, I can try. That's a big question. As a clinical neuropsychologist, I understand the struggle with that. I think, because of what you were mentioning exactly, especially working for me personally, working with Spanish-speaking communities and bilingual communities, there's no real good measure of premorbid functioning, right? And I think it really depends on what we're trying to get at with that. I personally think for me, what I would advocate is also with the continued improvement of the instruments that we're using, right? Better efforts to create normative standards, normative references for the population that we're working with, right? Because I think really we need the premorbid estimate also to use it, how we interpret normative data in a clinical context to diagnose somebody or not. All of those things are biased, and a lot of normative samples that we use in a clinic sample are outdated at this point too. They're very old, primarily representing English-speaking or monolingual individuals. I think we still need a lot of efforts, to really, people should value that in terms of the clinical setting, and how can we leverage other studies to be able to help with that and use the instruments we use in research studies in a clinical setting to help with the diagnosis. Because then the premorbid functioning, if we have better norms, we might not care too much about that as long as we got a good clinical history. That's at least my take, I think, my gut reaction from that.
I'm going to move forward because we have people who've been standing for quite some time. So Roberta, you're next.
Oh, thank you. Quick question. So Dr. Adkins-Jackson, you brought up the issue of, kind of this historical impact of race, and lynching. And I'm thinking, well, how could you test that? And it turns out that, maybe epigenetics, because I think about the mechanisms, right? So the epigenetic potential to track that impact. Your thoughts?
Yeah, no, when we did the SES paper, for example, what was interesting about that is we had 3 different predominant sort of thoughts on this. Some people were like, oh no, this is in utero exposure. And then other people were like, no, it's epigenetic. And I had the perspective that no, the policy still exists. I would love to test things epigenetically, but I think it's probably more cost effective to determine whether or not a policy is still re-injuring future generations. And then if we see that that's not the case, then we can invest efforts in whether or not this was actually epigenetic. But teasing apart that difference is difficult.
Well, you don't have to do either or. Just
yep, we can do both. Well, if we do, you holla at me and we can make this happen. I'm very collaborative.
Hello. Thank you so much for your talks. I'm Magdalena Misiak. I'm assistant professor at Howard University. And I actually like a lot what you said about when you mentioned trauma in Black population, despite their socioeconomical status. I'm particularly interested in participant recruitment for minority groups, and I know how difficult it is to recruit those individuals, I know about their distrust. And my question is that how would you advise to approach or develop the structures for efficient participant recruitment for my study, your study, build the collaborative efforts, because this is a big thing. The distrust in healthcare and recruitment in minority population is the big problem for us.
Did you want to jump in?
Yeah, I was just going to say, don't recruit from a clinic.
No, we recruit from the street.
Yeah. So, go into the communities, is the best advice there. For these between group studies, it's not always just that it's hard to recruit the minoritized population. It's that we're kind of lazy in recruiting the white population. So we'll make the effort to go into the community and recruit Black participants, but then we compare them to white participants who come to us in the clinic. So I want to point that out too, that if we want better recruitment and more representative samples for causal inference, we have to think both about both comparison groups. So I hope that is helpful.
Can I add to that, so if you think about the clinical model where a person has a problem, they come in, they get intervention, clinical intervention, and you also offer them the opportunity to participate in a study, what if you magnify that to the structural level? That would tackle some of the distrust because people have greater demands going on. So if you come in and you tackle economic disenfranchisement in the community through a structural intervention that also invites them to be a part of some other sort of epidemiological larger study, then you're killing 2 birds. Oh, that's such a horrible expression. But you get my point that there's more ways you can do this than just going, I need stuff from you. Maybe it's, I want to fix something for you, and also can you participate in this study?
We have to end. Thank you so much to everyone. Thank you, Molly. Thank you, John, for having this session. And we're around. We're here during lunch. So please, if you didn't get to ask your question, come find us.
All right. I think we're ready to start. Everyone ready? Okay. I'm not Catherine Kaczorowski. Although I aspire to be. I'm Molly Wagster with the National Institute on Aging, and I'm going to be serve as the session chair. Unfortunately, Catherine couldn't join us this afternoon, but she is going to be participating virtually and will be with us hopefully through most, if not all, of the discussion session virtually. So it's my pleasure to start off this first session of the afternoon. This session is entitled Genetic Environment, and our first speaker is Dr. Dena Dubal from the University of California, San Francisco, And the title of her talk is X Chromosome-Derived Mechanisms in Cognitive Aging. Dena.
Thank you to the NIA, to the McKnight Foundation, Molly, Jonathan, the organizers, the speakers, the audience. This has been an amazing meeting so far, and I'm so honored to be among you and to share our work on X chromosome and cognitive aging. Why is studying sex biology important? Because every cell has a sex, and sex influences fundamental biology through hormones, as we heard about earlier today, through the dose of the X chromosome. Female mammals have 2 X chromosomes. Male mammals have an X and a Y biology, and more. And true sex differences exist in aging. For example, females live longer. We heard this morning a very compelling rationale about why it's important to study women's health. It's essential to our knowledge of sex biology. It is essential to precision medicine. It'll be the first filter, male, female, gender spectrum. How does this affect risk for disease, response to treatment, and fundamental biology? And importantly, more than being just interesting, sex differences will make us understand if one sex is more resilient or vulnerable. This unravels new pathways to target with novel treatments that could benefit men, women, or both. This graph is astounding. From the United Nations a couple of years ago, this records mortality all across the world. In every society that records mortality, with life expectancy of women on the y-axis and of men on the x-axis. And what you can see from Somalia to Japan is that women live longer all across the world, regardless of socioeconomic status. Even in times of famine, girls will live longer than boys. And there is a biologic component to this. There's also behavioral, environmental, genetic, structural components to this. But there's also something very biologic. And in previous studies, we showed that mice that grew up with XX chromosomes, regardless of having ovaries or testes, live longer than mice that grew up with an XY chromosome complement. What about the brain? Women will show less cognitive decline or increased baseline functions in many populations of clinically normal adults. I will make the acknowledgement that the limitation of many of these studies, that they're predominantly white. And these studies listed approximately 25% of the populations collectively were Black individuals. This is an area that really needs to be worked on and advanced. So women will show less cognitive aging, not talking about dementia, but in normal cognitive aging. That women will show less cognitive aging is shown by the orange line there compared to men. So there is some resilience in the brain in aging. In contrast, and you'll hear from Rachel Buckley soon, women are also vulnerable, and we heard this from Bobby Brinton earlier, to effects of the genetic ApoE4 allele, to gonadal hormone depletion, and to more rapid cognitive decline, even though they start at a higher baseline in some populations. What about mice? Do mice capitulate sex differences in human cognitive aging? My graduate student, Francesca Marino, did an elegant study in which she used the large Y maze to test spatial and working memory, and found in young mice, XX compared to XY, there was equivalent cognitive functions. Above 1 means memory retention. In aging, as expected, males and females declined. But she saw something very interesting in that the XX mice showed resilience to the cognitive aging compared to XY mice, similar to what we see in some human populations. So in a subsequent study that I won't show you, we dissected out the effects of gonads and sex chromosomes and found that female resilience in cognitive aging is mediated by both gonads and sex chromosomes, but we were so struck by the effect of sex chromosomes, which was very large and very significant, that we dove right in and asked a question: if sex chromosomes are contributing to resilience, the XX complement, is it the second X that's helpful, or is it the Y that's harmful? She used an XY star model of sex biology, which varies doses of the X and Y chromosome. And what she found is that mice with 2 X's showed better cognition and aging on the left compared to mice with 1 X. Didn't matter whether the mouse was male or female, whether there was the presence or absence of a Y, that 2 X's improved cognition in aging male and female mice, giving us data that the second X is important. Another graduate student, Casey Shaw, examined whether there were changes on the X chromosome in aging mice, and found that compared to all the autosomes, that there were more differentially expressed genes in aging on the X chromosome than any other chromosome. Now, what is the big deal about this second X? As you remember, in our development, in female mammals, one of the X's is shut down. So males have 1 X and females have 1 active X. Why would that second X matter? Well, as a whole, when we step back, the X chromosome contains 5% of our genome. It's enriched for cognition-related genes. There's more brain-related genes on the X chromosome than any other autosome. And historically, the X chromosome has been, as well as the Y, been siphoned away in informatics studies because they pose technical difficulties with different dosages between males and females. But as we enter this new era where it's an exciting time to really dig into the X. And that second X inactivates in females. So what is the big deal? To come back to the question, why would a second X matter? There are many reasons why a second X could matter. One, for example, is baseline X escape that we published on and that Rachel Buckley is working on in Alzheimer's disease. And that is that silent X isn't so silent, that some genes escape that inactivation. And so females will have more of an X dose. But today I'm going to talk to you about mixed parent of X origin. And this is, if you haven't done your Sudoku or your Wordle today, this is your chance to really put your puzzle hat on and think through this. So, why examine parent of X origin? Meaning, does my X come from my mom or does my X come from my dad? That's parent of X origin. This work is done by Samira Abdullahi Saiku, a very talented postdoctoral fellow who's on the job market. If you see her application, this is her work. Parent of X origin confers genetic diversity and epigenetic diversity and is a major cause for sex difference. And it's a major cause for heterogeneity between women. Okay, so again, one of the X's inactivates in development. Because it's random, one X will be active from mom in a cell that's on the left, but in a neighboring cell, because of random X inactivation, the X will be active from dad, the paternal X. So side by side, you may have a female cell with mom's X active, and then the neighboring cell will have dad's X active. And this is a picture of the CA1 region of a mouse hippocampus where there's a fluorescence labeling of neurons that show mom's X in green and show dad's X in pink. And look at the mosaicism here. Absolutely beautiful and such diversity. In this case, the mice are inbred. It's not genetic diversity, but let's see if there's epigenetic diversity here. Jeremy Nathans published now a decade ago that in the same litter, this is to highlight the heterogeneity between women, in the same litter, Sisters can have a very different ratio of mom's X active or dad's X active. So on the left, you see a green female. She's mostly mom's X active, and on the left, I think I've been confusing that. On the left, you see mom's X active, mainly in this in the red sister. So just to say, there's incredible differences between women and even between sisters in which X can be active. So Samira asked, well, does this mosaicism matter? Could it buffer age-induced decline? And she first had to ask, are neurons that express mom's X actually different than neurons that express dad's X? And to do that, she used a very elegant genetic system with a synapsin-driven Cree labeling neurons specifically in the hippocampus that are showing dad's X red, mom's X green. She fact sorted them out, had a rigorous experimental design, and found something that was like unbelievable, was so exciting. I've long been excited by even 20% of difference in the brain, but this is on-off gene expression changes. So neurons that express mom's X in the graph in green on the right have many genes that are completely silenced. And so mom's X will silence specific genes in both the young and aging hippocampus, but the neurons that show dad's X have very robust expression of these genes. Very, very clear, very robust. And then she asked, well, does this difference matter? Is it detrimental that mom's X silences genes or imprints genes? She first asked, does it accelerate biologic brain aging? So she took neurons from the same hippocampus that were from mom's X versus dad's X, green versus red. And again, what she found was astounding, that mom's X will accelerate the biologic aging in the hippocampus of female mice, almost double the age here of a neuron that expresses dad's X. Again, this is from the same environment, from the same mice. She then asked, if I could increase those imprinted genes, would it improve cognition in aging females? That was her question. And to do this, she used a CRISPR-A system with dCas9 and took the top 3 genes, TLR7, SASH3, and cis-LTR1, and used dCas9 and small guide RNA to activate them, first in vitro, then in vivo. So now she's boosting up these silenced genes in the aging female. And what she found was, wow. So in this, on the left, you'll see the in vivo injection of lentivirus of the dCas9 and the small guide RNAs. And she showed that the genes were increased. And then what happened in the water maze. This was true for both learning and for memory, that those aged females which had these silenced genes turned on showed better both learning, which is not shown here, but also in memory and probe of the water maze. So the ones that had CRISPR activation were smarter than controls. She showed this in another task, novel place memory. Again, those that had increased expression of the imprinted genes showed improved cognition in the aging female. So in summary, I've shown you that women live longer than men and often show resilience to cognitive aging in many populations, and that the XX sex chromosome complement contributes to better cognitive function in aging males and in female mice. Adding or subtracting the Y didn't alter cognition in aging. And that quite remarkably, that the maternal X chromosome will silence cognitive loci. And if we increase those silenced loci, if we boost up their expression, it actually improves cognition. And why does this matter? Why is this provocative? For two reasons. We're now looking at actual sex differences. So what I showed you is all female data, but males will only have their X chromosome from mom, right? They don't have an X chromosome from dad. From dad, they get the Y. So in males, are these cognitive loci silenced? And could that be a reason for vulnerability in cognitive aging? So number one, importance of sex difference. Number two, heterogeneity among females. So it could be that if I have a ratio of 70% mom's X, remember, blame your mom, because a mom's X silences cognitive loci, which appears to be detrimental. If I'm 80% mom's X, but let's say Rachel is 80% dad's X, who's going to do better maybe in cognitive aging? If we extrapolate our data to the human condition, assuming there's relevance here, then Rachel's going to do a lot better in cognitive aging than I am. So maybe this portends something about cognition and aging, maybe risk for disease, maybe risk for neurodegenerative disease. This has major implications for precision medicine for women's health. And I love that Andrew shared with us his hopes and dreams about technology in the first session. And I'd just like to share with you a hope and dream that, and I think about this every day, that someday, as part of our understanding of biology and of health, that we will understand how an individual woman's hormones, and specifically maybe even how her X chromosome expression will dictate, predict, govern her risk for diseases, her response for treatment, that it may be a very integral part of human health to understand how the X expresses itself, which will be very different in each woman. So at the end of the day, identifying therapeutic targets on the X could lead to novel therapies to prevent cognitive decline in both sexes. So if those genes that are turned off on mom's X chromosome could be turned on through small molecules, through viral-mediated therapies, maybe through lifestyle, biologics, if they could be turned on, maybe these are really valuable therapeutic targets. And I'd like to thank the folks that worked on these discoveries, my collaborators, and the NIA and the Simons Foundation for funding this work. Thanks.
Thanks, Dena. That was terrific. Our next speaker is Dr. Rachel Buckley from Harvard Medical School and Mass General Hospital. And the title of her talk is Sex Differences in Risk and Resilience to Cognitive Decline, Intersections with ApoE4 and Beyond.
Dena, that was an incredible talk, and I'm absolutely going to blame my mother. So as soon as I get home. It is such an honor to be invited to speak here in this session, and I really wanted to thank the organizing committee. Thank you so much, Dr. Wagster, and also to Dr. Kaczorowski, who is online and hopefully can hear me. You are the ultimate consummate professional when it comes to herding cats, aka academics, so thank you so much. So a lot of the focus of my research is on in Alzheimer's pathology and its related cognitive outcomes. But where the intersection of sex differences really lies is in the broader aging milieu, including non-pathological aging, and its impact on cognitive resilience. And we can draw many connections that provide a bridge between age and cognitive decline, with some pathways involving a direct influence of elevated risk factors such as affective disorders and cardiovascular health, as well as immune and cardiovascular pathways. But there's also indirect pathways where the influence of these risk factors either partially or fully are mediated by brain structure and function. And in addition, there are also pathological pathways involving AD proteinopathies and others, And I'll briefly touch upon all of these pathways during this talk, but suffice to say, one can form a definition of cognitive reserve and resilience based on an individual's ability to perform better than expected given the level of risk along these pathways. And just as a quick diversion, I just wanted to give a shout out to my colleagues at the Reserve and Resilience Peer in the Alzheimer's Association, who partnered with us at the Sex and Gender Peer to write this review together that created this infographic. I think it just really provides a nice holistic view of the current literature. And also, for the purposes of this talk, I wanted to define some concepts about sex and gender. While sex, as we've just heard, is sort of regarded as the biological foundation of chromosomal and gonadal development, gender can be defined as the socio-cultural elements that help to shape our gender identity. And in many studies that I talk about today, and in fact, you can think of this across the board in medical research, we don't ask people about their gender identity. And we mostly assume someone's sex assigned at birth. And because of that, these concepts of sex and gender can get incredibly tangled up. And we have a lot of work to do in this space, but I think it behooves us to be transparent about this when contextualizing the findings I'll be talking about today. And something I want to take a moment to highlight in this figure is that along every single one of these pathways, theoretically, lies the intersecting influence of ApoE4. And you can see these highlighted in red. Right alongside that, in the gold squares, there lies the influence of sex, both from a sex steroid hormonal standpoint as well as a sex chromosomal standpoint. But one can't ignore the gendered elements that are layered over the top, such as education level, occupational attainment, gender identity, and other gender-specific determinants of health. But I don't have time to explore these today, unfortunately, but I'm happy to take questions later. I will be focusing on what we already know about the intersectionality between ApoE genotype and sex. I'll posit some theories about why we think this intersection exists. And finally, I'll raise some questions about what we still don't know. So let's pause for a second. Let's think about the theoretical model of how sex differences in cognitive resilience might occur throughout the aging. One potential is that women simply just outperform men across the entire age span. And we now have decades of evidence showing that women do outperform men on many neuropsychological tests of cognition, particularly if they're verbal. And one might imagine a model where women just simply have a slow decline over time, similar to men, but they remain better performing. But there's also another much more compelling scenario where women still outperform men earlier in life, sorry men, but a confluence of events occur, such as the menopause transition or being an ApoE4 carrier, that might knock the resilience off its course, causing women to decline faster past a certain point. And these intersecting influences of sex and ApoE do not just impact cognition. And I'll present evidence to support that supposition, but there have also been very interesting outcomes of sex and E4 on neurofibrillary tangle density, which I think is also worth exploring in this talk. And in both circumstances, women have an outsized disadvantage if they carry the E4 allele. So let's start out with sex differences in incidence rates of dementia. It might be surprising to some people in this audience that it's not actually, there's no entirely convincing evidence to suggest that there's a main effect of sex on incidence rates to dementia. But time and again, many studies have shown that women who carry the E4 allele, whether it's one or two copies, by far and large, show faster rates of progression to dementia relative to male carriers. The figure I'm showing here today is from the seminal paper by Dr. Judy Parr's team that show far higher rates of dementia incidence in women who carry E4 than men who carry E4. But, critically, it's only between the ages of 65 to 75. Now, this is of great importance to our understanding of women's reproductive health, because if we think about the endocrine aging system across the population in the US, the average age at menopause is 51. And at this time of life, you're not just seeing a massive hormonal cessation event, but you're also seeing things like gynecological surgical intervention and hormone therapy use in subsections of the population. And if we assume that the clinico-pathological progression of AD takes approximately 20 to 30 years from the time of a dementia diagnosis, you're working your way back to right around the time of menopause, when the genesis of aging-related processes, including but not limited to AD, might be starting to accelerate. And it's not just the menopause transition that may reduce cognitive resilience beyond a certain age in women. Data seems to suggest that the additional factor of the E4 allele may really exacerbate risk for women post-menopause. So let's just focus on some of the evidence. I've already talked about the findings associated with the sex by E4 risk on clinical progression, but there's also some literature suggesting a sex by E4 effect on rates of cognition. In a large proportion of these studies, women who carry the E4 allele show the greatest detriment to cognitive performance over time, but it is important to note that dissenting studies exist that show either no difference between men and women, or that men might show greater deficits. So we really need to tease this apart further. Jack and colleagues have also showed sex by E4 effects on hippocampal atrophy, but it's the men who carry E4 that show faster rates of atrophy past a certain age, greater than women who carry E4. But most consistently, findings across both CSF and PET imaging seem to suggest that women who carry E4 show much higher levels of tau relative to men, and this is also demonstrated in women who have abnormal levels of amyloid. And here on the left, I'm showing you some of our data using Flotaucipir PET, and on the right is from Dr. Timothy Homan's lab. That's a meta-analysis of CSF total tau. So if we're interested in what might be going on under the hood, so to speak, I think we should really consider the moderating effect of hormones. And here there is a little data, although much more needs to be done in this area. First, there is some much older evidence that current estrogen use via exogenous menopausal hormone therapy might exacerbate E4 effects on cognitive decline, as can be seen on that sort of bottom row to the very right-hand side on the table. While hormone therapy may have a protective effect in E4 non-carriers, as can be seen in the top row, but more recent evidence from Dr. Kantasi's group also suggests that hormone therapy use may result in lower amyloid if you're an E4 carrier. So again, more needs to be done. In our recent unpublished work, we found some interesting signal coming from the sex hormone binding globulin levels in the blood, which may potentially provide a protective effect, but in men only. And that is that higher levels of SHBG are associated with lower levels of tau PET signal. But these findings were only being driven by those men who did not carry the E4 allele. Now, sex hormone binding globulin is a fascinating glycoprotein. It's most widely associated with sort of bioavailability of sex steroid hormones. But there's also an interesting role to play in the literature, suggesting that SHBG might be associated with vascular risk, a protective effect. So more needs to be done here. Yet other evidence suggests an interesting role to play for testosterone, with lower levels of testosterone associated with more detrimental outcomes for hippocampal volume in male E4 carriers. And that testosterone may also moderate relationships between E4 and tau. These are findings from Dr. Sunderman and colleagues that showed lower levels of testosterone were associated with higher levels of CSF p-tau, but only in E4 carriers. And it wasn't across both sexes. This was just apparent in women. In all, these findings paint a really interesting but complex preliminary picture of sex steroid hormones and SHBG playing a potential protective role against the effects of E4 on AD and non-AD related outcomes. But when we're thinking about ApoE, it's not just interest in the effects of E4. This is a really nice paper by Dr. Nichols and colleagues from the Advanced Psychometrics Workshop, looking into the sex moderation of the protective E2 effect on cognitive decline. And the takeaway point from this figure is that E2 shows a robust, direct, and protective effect on cognition across both AD and non-AD pathological contexts. But of greatest relevance to this talk, they found that E2 was protective only in women, which was only partially mediated by AD pathology, suggesting that there might be other pathways that might be sort of inducing this protective effect, potentially immune and inflammatory responses, or childhood cognitive development, where there are known sex differences. Suffice to say, there seems to be a very interesting role for different ApoE variants to play when considering sex differences in resilience with age. Continuing this story, another recent paper examined how sex and race might intersect to influence the protective effect of E2 on cognitive performance. This again is from Dr. Tim Homan and Dr. Loga Dumitrescu's group at Vanderbilt. And the huge strength of this paper was the sheer sample size. This was including more than 32,000 individuals across 4 harmonized cohorts and included more than 4,000 Black individuals. And here I'll just focus on their findings to do with executive function. They found a significant intersectional effect with the cognitively normal sample, whereby the E2 allele showed a female-specific protective effect among non-Hispanic white individuals, but a male-specific protective effect among non-Hispanic Black participants. And these pattern of effects were not found in the E4 allele. So I think this is a really interesting and novel way of looking at the data, and suggests that there might be some interesting and differential ways that these variants play effects on resilience across different subpopulations. So now I'd like to turn to potential theories of why there might be intersections between sex and the E4 allele specifically, as this remains an open question. And we've already heard quite a lot about the metabolism story from Dr. Brinton today, and I wanted to sort of contextualize it within the E4 context. There is evidence of a range of pathways that the E4 allele impacts, from dysregulation of lipids to aberrant accumulation and storage of unsaturated fatty acids, but it also alters cholesterol biosynthesis and trafficking in oligodendrocytes and plays a role in demyelination. Supporting some of this evidence are findings that suggest greater consumption of dietary cholesterol may result in worse outcomes for women who carry E4 cognitively. And of critical importance to our line of inquiry is that there are more widespread metabolic differences between ApoE isoforms, in that E4 carriers, there might be a greater reliance on ketone bodies as opposed to glucose as a bioenergetic fuel in the brain. And this could have major implications for women, considering they undergo a massive bioenergetic shift in the brain during menopause, as Dr. Brinton mentioned. So in summary, there might be an emerging story in destabilized bioenergetics in the brain, which might knock some women into a vulnerable state and reduce resilience to aging processes. But that still leaves us with so many questions unexplored. Firstly, is that the end of the story, or is that the whole story? What are other biological pathways that are yet to be uncovered that might explain the sex by E4 impact? What unacknowledged biases are we ignoring here, like survivor bias, that might impact the biological interpretations that we have? Dr. Dubal has presented some incredible evidence of the power of the X chromosome, and yet we still are trying to tease apart what is the X chromosome doing in combination with E4. And we still don't know the full answer about how cardiovascular risk is involved. And finally, the influence of gender and race and ethnicity are significantly underexplored and need further investigation. And I wanted to end on a positive note here about what we can get excited about in the future. These initiatives have already been mentioned earlier in the hour earlier in the day by Dr. Jacobs, but I wanted to just underscore again what exciting time we're in right now. There's a new White House initiative for women's research led by Dr. Jill Biden, which gives me chills. and the new Ann S. Bowers Women's Brain Health Initiative that's being led by the inspiring Dr. Jacobs. And we had the pleasure of hearing a little bit about this, but one thing that's really exciting about this initiative that we're hoping to do very soon is to kick off the first ever longitudinal menopause project. In this project, we hope to deeply phenotype a large cohort of women across the United States who are smack bang in the critical years of their menopause transition. With these types of groundbreaking approaches, we hope to really drill down on some of the biggest questions out there related to women's resilience during aging. E4 is going to be just one of those questions we can ask. So watch this space. There's lots to be excited about. And I'd like to end there and thank you all for listening to this talk. Special thanks to my colleagues both in my lab and outside of it. Thanks to our funding sources at the NIA and the Alzheimer's Association, and the team of researchers and participants who support this work. It was such an honor to be here, to be given a voice amongst such inspiring scientists today. And finally, a huge thanks to my little girls for reminding me why I'm so passionate about women's health at all ages. Thank you.
All right. Our next speaker is Steve McCarroll from Harvard Medical School and the Broad Institute. His talk is going to be a synaptic neuron-astrocyte program, or SNAP, which varies naturally among humans and declines as we age.
Wow, it's going to be really hard to follow two amazing talks like that. And I'm just grateful that to be invited to come share this work. And I've been learning so much at this meeting. So yeah, I want to talk about a program that we discovered involving multiple cell types. It seems to vary naturally among all of us, but also to show really pronounced changes with age. But just to introduce the idea of it, I just want to go back to, so some years ago, my lab was developing this technology that we called DropSeq, for droplet-based single-cell RNA-seq, that's evolved into like the 10x technology and for single-cell genomics. And at the time we were trying to get this to work, we came up with a metaphor for what DropSeq was, which we said, traditional genomics is like taking a piece of brain tissue and throwing it in a blender and analyzing the smoothie, like the mixture or average of all the cell types together. And we wanted to make it possible to analyze every individual cell. And so, in other words, more like a fruit stand, less like a smoothie. So it was a great metaphor for how DropSeq, but it's a terrible metaphor for how the brain works. And let me explain why. So the brain is like the ultimate non-cell autonomous biology organ. Almost everything important that the brain does involves collaboration among multiple types of neurons and glial cells, and sort of atomizing the brain and thinking of it just in terms of the cellular parts really misses the magic of what the brain does and how it does it. And so we wanted to come up with a way to use these kinds of data, but to ask a really different question, not to like make a parts list or a cell atlas, but to actually identify tissue-level programs that may involve important interactions or collaborations by multiple kinds of cells. So how would you do that if you're going to analyze all the cells individually? So the idea that Emmy Lang, a postdoc in my lab, came up with is to use patterns in quantitative biological variation across many individual humans. So let me explain what I mean by that. So the experiment was, on some level, simple. We did single nucleus RNA-seq of the prefrontal cortex from almost 200 postmortem brain donors, variety of ages, actually half with schizophrenia, half controls. I'll get back to that part in a minute. And in total analyzed something like 500,000 in cells or something like that, profiling gene expression genome-wide in each one. But then Emmy came up with a very different way to analyze the data, which is based on latent factor analysis, a form of machine learning. And the basic idea, by latent factors, we mean inferred underlying states that cause constellations of like hundreds or thousands of gene expression measurements to change together. In the same, they change together, and the way you can tell they change together is that you'll see all the changes in the same people, okay? But here's the secret sauce. Those changes don't all have to be in the same cell type. So they don't, or in the same cell. So they don't have to be like this transcription factor regulates these 20 genes. This is at the level of the whole system. What are the things that always change together and appear consistently in the same people? So, and here's the surprising thing. 10 latent factors explain 30% of inter-individual variation in gene expression levels. And very many of these latent factors, particularly the ones indicated with arrows here, involve coordinated changes in multiple kinds of cells. And in particular, the ones that appear over and over again are neurons and astrocytes. So what this approach led us to, that we actually had no idea when we started this project, is that gene expression in one's neurons is powerfully predictive of gene expression in one's astrocytes, and vice versa. And in particular, one of these latent factors, the one I'm going to focus on today, so actually 3 of these latent factors changed greatly with age. Today I'm just going to have time to tell you about one of them. In this chart, it's latent factor, in this plot, it's latent factor 4, okay? But latent factor 4 involves a constellation of gene expression traits, thousands of them in neurons and astrocytes, that change together and that decline as we age. And you can see from the green line, that's the decline in controls. And you can see from the parallel purple line, that's the decline in persons with schizophrenia. So it declines in both groups, but in schizophrenia, there's a downshifting of the whole distribution, equivalent to about 30 years of aging. Now, that may seem at first, depends on how much you've thought about schizophrenia. That may seem random, but if you've actually thought about schizophrenia a lot, it doesn't seem random at all. So culturally, people talk about schizophrenia in terms of psychosis, the like hallucinations and delusions, because that's like the very striking feature that you recognize when you see it. Okay, but actually that's a relapsing remitting symptom, and it's treated with medication. What people with schizophrenia and their families and physicians will tell you is that the truly debilitating symptom of the illness, what knocks people out of the education and the workforce, is cognitive decline. Loss of cognitive flexibility, loss of processing speed, and when you see someone on the subway, you don't know their baseline, right? So you don't recognize their cognitive decline. You recognize if they're hallucinating, right? But the truly debilitating feature is actually something that's shared in common with aging. Schizophrenia and aging also share reductions in cortical thickness, reductions in the gray matter, reductions in neuropil, and reductions in numbers of dendritic spines. So these are all shared features of schizophrenia and aging. They're just appearing far earlier in schizophrenia. So the question that I think and actually one of the things that's not on this slide actually is, having had schizophrenia and some other psychiatric disorders earlier in one's life is a profound risk factor for dementia later in life. It is as if schizophrenia, and it's not a small risk factor, it's like a several-fold increase in risk. It is as if these illnesses deplete a kind of cognitive reserve. That, of course, we'd love to know what the molecular and cellular substrate for that is. So might these things be arising from shared molecular and cellular changes? And what are the shared? So one of the things that's very clear in the data, and here each point is a different person. The horizontal axis is their age, and then the vertical axis is the aggregated expression level of a specific functionally defined set of genes in one of their kinds of cells, in this case in their inhibitory neurons. And one of the things that I think you just recognize immediately is this expression of different families of genes that encode synaptic components, and you can see this very clear decline in age. Now there's inter-individual variation superimposed on that, but there's this very clear pattern when you look across enough people that as we age, our neurons are reducing their investment in synaptic components. So this is in one kind of neuron, but we actually see this in every single type of cortical neuron. So this isn't just some little special microcircuit. Every type of inhibitory neuron, every type of projection neuron, we see this divestment from synaptic components with age. Again, with inter-individual variation superimposed on top of that, but a clear pattern of decline with age. But here was the surprising thing that the latent factor analysis taught us, is that neuronal and astrocytic gene expression related to synaptic molecules and functions are highly correlated with each other. You can see this correlation across people. Here, the y-axis is neuronal investment in synaptic components. The x-axis is each person's, the investment of their astrocytes in neurotransmitter transporters, you can see strong correlation at a population level. And same is true for other astrocyte functions related to synapses. The synaptic adhesion molecules that astrocytes express, by which their processes adhere to synapses. And also astrocytic commitment to cholesterol synthesis. All of these things strongly correlated with neuronal investment in synaptic components. So why this change in astrocyte cholesterol biosynthesis? Just in case that seems random. So neuron synaptic cholesterol actually comes from astrocytes. So 25% of the body's cholesterol is in the brain. And synaptic membranes are particularly cholesterol rich. There's some of the most cholesterol dense membranes in the human body. This cholesterol is made locally, and the cholesterol at dendrites and synapses is actually made by astrocytes and transferred to neurons at their dendrites and synapses. And it's actually transferred via apolipoprotein particles like ApoE. Okay, so that is certainly one place, among others, that ApoE biology sits, is in this interaction between astrocytes and neurons. So we call this program the Synaptic Neuron-Astrocyte Program, or SNAP. And it involves these coordinated gene expression changes by cortical neurons and astrocytes. Different genes in neurons than in astrocytes, but you can see across people that there's like one set of genes, one set of synaptic genes that's highly co-regulated in astrocytes. We call that program SNAP-A, and there's a different set of genes with synaptic functions that's highly co-regulated in neurons, and we call that program SNAP-N. And when you look across people, SNAP-A and SNAP-A on the left and SNAP-N on the right, they're both declining together with advancing age, and they both have about a 30-year acceleration of that decline in people with schizophrenia. But furthermore, even if you remove the effective age, like if you just adjust the data for age and the effect of schizophrenia, SNAP-A and SNAP-N are still very highly correlated. You can see even in 200 people, that relationship has a p-value of like 10 to the minus 16. So these programs there's coordination above and beyond that chronological age is influencing both of them independently. We're working hard to try to identify the mechanism of that coordination. So just to summarize, cortical neurons and cortical astrocytes coordinate gene expression related to synaptic biology in a program that we call the Synaptic Neuron-Astrocyte Program. And SNAP declines in aging and schizophrenia. So I want to separate what we know from what we think. So what we know is that SNAP is a cortical gene expression program in which neurons and astrocytes are mutually investing in synapse-related components and activities. What we think is that SNAP may be supporting plasticity, maybe providing cognitive reserve, but it's obviously going to take different kinds of experiments in living systems and especially in animals to understand that. And those things we're working on now. SNAP declines in schizophrenia and aging. We're working to understand the extent to which SNAP may also decline in other brain disorders, and we are finding that it does in actually several different psychiatric and neurodegenerative disorders that have in common that they have effects on reductions in gray matter volume and losses of dendritic spines. So we think that this may be a convergent pathway on which many different kinds of brain disorders with different genetic causes actually converge. And finally, this is more of a big thing, but we find that schizophrenia risk is arising not just from the neuronal activities that are part of SNAP, but also from the astrocytic activities that are part of SNAP. So that is to say, the astrocyte role in this is not just reacting to synaptic dysfunction, but that there's an active and important contribution from the astrocytes. So key questions in current work, we're developing living biological models of SNAP in mice and through in vitro interactions of neurons and astrocytes. We're trying to understand how SNAP's affected in other brain disorders. We would really love to find biomarkers for monitoring SNAP in living patients. So imaging biomarkers, CSF biomarkers, but ideally even plasma biomarkers. And so that's an area of interest. And then we're trying to figure out what's the molecular mechanism that's coordinating the neuronal and astrocytic activities that are part of SNAP. So if you'd like to read more about this, there's a preprint on bioRxiv, and then actually the paper just came out a couple weeks ago, and we forked up $10,000 to make it to buy open access. So if you can, hopefully, tell me if it didn't work. But if you go to the Nature website, there shouldn't be a paywall for this paper. So two things. First of all, I want to acknowledge that there's a limitation of this work. So the vast majority of the brains that we looked at are from persons with European ancestry. And in general, persons with non-European ancestry are underrepresented, not just in our study, but in brain banks in general. The Harvard Brain Tissue Resource Center that we worked closely with to do this is making intense efforts to connect with physicians and families in minoritized communities to try to address this. This requires a lot of listening, building of trust. There's obviously an important history to be mindful of that makes people reasonably worried about, privacy and attributing the biology things to groups and things like that. So our colleague, Dr. Sabina Beretta, and her colleagues are working really hard to address this. I do want to mention two other things. So SNAP is by no means the only set of age-related changes that this kind of analysis reveals. So, and in particular, there's two other. My slide about this seems to have dropped, but there's two other things that that are very clear in the data that we're working harder to understand. So one involves age-associated loss of interneurons. So we're working really hard to try to understand the genetic influences on that. And then the other is age-associated clonal expansions of glial cells with somatic mutations. So something that's analogous to something we discovered some years ago that we called clonal hematopoiesis, where as we age, a substantial fraction of our blood is actually clonal. It's derived from a single hematopoietic stem cell that acquired a growth-promoting mutation decades earlier. We're starting to see this in the glial populations in many people, and that's something else that we would like to work really hard to understand. So, as you guys know, one of the best things about this business is being able to work on interdisciplinary teams, and this was just a lovely, cohesive team of people to work with. In particular, Emmy Lang, a postdoc who really led the whole project, Jem Nimesh, a data scientist and software engineer who also contributed to a lot of the innovation in terms of the machine learning methods and things like that. And my close partner and co-author, Dr. Sabina Beretta, and Kylie French from her lab, but also a much larger team of people who contributed in many different ways. So thank you very much for listening, and I'll look forward to your questions and feedback and suggestions.
All right. The last talk for this session is from Catherine Kaczorowski from the University of Michigan. We will be doing, okay. The title of the talk is Systems Genetics Identifies Novel Resilience Factor in Normal Non-Pathological Cognitive Aging. And I think we're bringing it up now.
Hi, I'm Catherine Kaczorowski, the Eleanor Levine Endowed Professor of Neurology at the University of Michigan. I'm so sorry that I can't be there with you today in person, but I'm delighted to share work from my laboratory that was led by Dr. Sarah Nooner, who's now a scientist at Regeneron. And we were really interested in using a systems genetics approach to identify novel resilience factors in normal, non-pathological cognitive aging, particularly in the context of resilience. As we all know, aging is the leading risk factor for many diseases, particularly decline and dementia. And we also know that the process of cognitive aging is complex and different for everyone. Some individuals follow this trajectory with little to no cognitive decline. Others, a more normal aging trajectory with some amount of cognitive decline. Or this, a more severe descent into dementia, which is when cognitive impairment becomes so severe it interferes with daily living abilities. identifying the mechanisms involved in regulating these individual differences in the trajectory, particularly those that are acting at this midlife critical time point, will not only enable a better understanding of the causes of age-related cognitive decline and dementia, but also highlight potential therapeutic targets. We know from twin and family studies that genetic makeup plays an important role in determining the trajectory of cognitive decline. So we've been really focused on identifying the genes and pathways involved in regulating individual differences in cognitive outcomes using animal models in order to apply forward genetics to discover genes that are causing age-related cognitive decline and hopefully highlighting potential therapeutic targets. In order to identify genetic factors that regulate variation in cognitive aging, which is quite difficult in humans, we performed cognitive tests in a very large cohort of genetically diverse mice. These are termed the BXDs, and they're ideally suited for using forward genetics in order to identify novel genes that influence differences in cognitive outcomes. Specifically, we trained middle-aged mice from 21 different BXD mouse strains on a standard contextual fear conditioning memory test, which measures spatial and episodic memory. And as you can see, there's wide variation in the memory abilities of these aged mouse strains, ranging from almost 0%, which would be equivalent to severe cognitive decline, all the way up to almost 80%, which would be our cognitively resilient or unimpaired mice. Now, our calculation of heritability suggested to us that 70% of the individual differences in the variation in memory across these BXD strains is actually a result of genetic factors. Therefore, we use genetic mapping to identify specific regions of the genome that were harboring differences in DNA sequence that were involved in cognitive aging. And what we found was a significant peak on chromosome 4 that had DNA sequencing differences significantly associated with how well a given BXD strain performed on contextual fear conditioning at midlife. Given that there were many genes within this peak on chromosome 4, we needed to narrow in on those that had sequence variance between the parental strains, the B6 and the D2. The next thing we did was use whole genome transcriptional data from the hippocampus of aged BXD strains to identify those genes that had cis variance. That means there was DNA sequence that was responsible for controlling the expression of that gene. Lastly, we tested gene expression for the degree of correlation with the contextual fear memory trait. From that integrated behavioral and gene expression data, we were then able to nominate a single gene, HP1BP3, as the putative causal gene in this. HP1BP3 is a heterochromatin binding protein that's thought to regulate gene expression through its actions in the nucleus. We next wanted to confirm that this effect was specific to cognitive aging as opposed to general memory function. So we used data from a study that had previously trained adult BXDs on contextual fear conditioning, and we broke them down depending on whether they had inherited the B or the D version of HP1BP3 in their genome. And what you can see is that HP1BP3 genotype had no effect of cognition in the adult BXDs, but strains inheriting the D allele exhibit robust memory deficits when they're middle-aged. And that demonstrates a genotype by age interaction where reductions in the expression of HP1BP3 in the hippocampus correspond to specifically to age-related cognitive impairment. The next question we had was, how might HB1BP3 be acting to regulate individual differences in memory at midlife? So there's evidence that groups of highly correlated genes are likely to play a similar biological function or act as a part of the same biological pathway. So we identified all of the genes whose expression was significantly correlated with that of HB1BP3 in the hippocampus of middle-aged BXD mice, and we used this for gene set enrichment analysis. This plot is showing you the calculation of the enrichment score. And the farther away each line gets from 0, the stronger the enrichment. Each black line under the graph represents an individual gene belonging to that set. And here I'm just showing you that positive correlates of HB1BP3 were significantly enriched for localization to the nucleus, with functions including RNA splicing, nuclear transport, and transcription activation. Now, we are excited to see this because it confirms a few known roles for HB1BP3, including in regulating cell cycle, chromosome organization, and transcription. Contrast these known findings with the negative correlates of HB1BP3 on the right, which were actually enriched for localization to the plasma membrane. These negative gene correlates of HB1BP3 were ascribed to functions including ion channel activity, transporter activity, and G protein coupled receptor activity. So when we combine these results with what's previously known about HB1BP3, we hypothesize that HB1BP3 might be acting to negatively regulate the expression of some critical ion channels or transporters in the plasma membrane, many of which have been previously associated with memory. So to directly test whether HB1BP3 was really critical for successful memory formation and involved in regulating neuronal excitability as well as synaptic plasticity, we trained mice that had viral-mediated knockdown of HB1BP3 in their hippocampus, which is a key memory region, on contextual fear memory, followed by performing electrophysiological characterizations of neuronal functions. After confirming that the knockdown was sufficient to disrupt contextual fear memory, we sacrificed these mice. This allowed us to measure neuronal excitability and synaptic plasticity in neurons from brain slices using whole cell electrophysiology. And what we found was that knockdown of HB1BP3 in hippocampal neurons resulted in a larger and more negative post-burst slow after hyperpolarization, which is indicative of a decrease in neuronal excitability. And this parallels aging and AD-related changes in neuronal excitability that had already been published in a variety of models. So what I've shown you so far is that HB1BP3 regulates cognitive aging in the BXD panel, and that loss of HB1BP3 is sufficient to recapitulate deficits observed in the global knockout and in aging animal models. I also showed you that decrease in HB1BP3 results in a larger post-burst after hyperpolarization and reduction in neuronal excitability. And although I don't have time to show you today, we also found that this reduction in HB1BP3 impairs synaptic plasticity. To assess the translational relevance of the loss of HB1BP3 on cognitive aging, we next wanted to measure HB1BP3 in human postmortem brain tissue from the hippocampus. And we found humans exhibiting age-related in the absence of overt AD pathology in their brain actually had less HB1BP3 protein from postmortem hippocampus tissue compared to their age-matched unimpaired controls. And these findings were actually published in 2016. Fast forward to 2023, this same gene, HB1BP3, has come up in a new publication from Li Wei Tai's lab. What they did was they performed the broadest analysis yet of the genomic, epigenomic, and transcriptomic changes that occur across every cell type in the brains of Alzheimer's disease compared to controls and cognitively resilient individuals. And what I hope you can appreciate is that HB1BP3 is upregulated in almost every cell type that they measured to be associated with Alzheimer's disease diagnosis. So we decided to directly test whether or not there's a change in HB1BP3 protein in the AD brains as well. And we looked in the hippocampus of AD mouse models as well as human AD patients, and we actually saw that there was an upregulation of HB1BP3 in both conditions, which at first was puzzling to us because it's opposite of what we see under normative aging conditions. But what I know some of you can appreciate, especially after the last few talks, is that mislocalization, especially of nuclear proteins, is a very common feature of neurodegenerative diseases. And so we did immunohistochemistry to see if that was the case here. And I hope what you can see by these images is that under normal conditions in the mouse hippocampus, HB1BP3 is entirely nuclear. However, when you look at the AD brain, you can see that we're now observing pretty robust expression of HB1BP3 in the cytosol in both neurons and astrocytes. So what I think could be going on here is that although the overall levels of HB1BP3 in both transcript and protein are higher, it's not actually functional HB1BP3 because it's not in the nucleus where it needs to be doing its job. This consequence is actually a loss of function of HB1BP3 that we believe is contributing to the memory deficits observed in both our normal aging BXD mice as well as in the context of Alzheimer's disease dementia. So just to give an overall summary of what I've shared with you today, I think the discovery of HB1BP3 adds to a number of examples of mouse studies that have really demonstrated that some forward genetics in mice are leading discovery. And really, this has gone unrecognized. Certainly, it also contributed to the discovery of genetic mechanisms underlying normal non-pathological aging and showing that some of the things that we've discovered in the context of normal aging are super relevant to Alzheimer's disease. And I think the Alzheimer's disease field and researchers should really be paying more attention to some of the discoveries that we're doing in the context of normal cognitive aging. I think there's a huge gap to fill in the context of mechanisms of normal cognitive aging. And again, I think the outcomes are going to be very translationally relevant. We know that 35 to 40% of the variance in memory outcomes in Alzheimer's disease dementia are ascribed to, all brain pathology. But there's a lot of the annual rate of cognitive decline that is not explained by brain pathology. And that unexplained variance or that residual, I think has a lot to do with just normal non-pathological cognitive aging. And our ability to identify those genes is going to be paramount. The last thing I want to do is just say that although I've demonstrated that increasing genetic diversity in our mouse models is a huge boon for modeling human relevant disease, we are absolutely still missing real world generational trauma, social and environmental exposures that are differentially impacting individuals based on their gender and their orientation and their race and disability status. And one of the gaps and opportunities I'm really interested in working towards is finding ways to integrate some of this rich environment and socioeconomic stress and other things into our models so that we can really start identifying treatments that are going to be advantageous for everyone and not just some of us. So with that, I really want to thank you all for your attention. And I also need to thank a number of individuals from my lab at the Jackson Laboratory, especially who is an outstanding graduate student in the lab and has gone on to do really fundamentally amazing work with Allison Goat during her postdoc, and now leading really exciting programs at Regeneron. Benjamin Garfinkel was a really outstanding collaborator that helped us get the HB1/BP3 knockout mice. I learned systems genetics and forward genetics from Rob Williams and Megan Mulligan when I was faculty in Tennessee, and I owe them a lot. We had a number of collaborators at the Hebrew University of Jerusalem. And of course, we would be nowhere without all of the funding support that we've received to do this work. And especially Dr. Wagster for creating opportunities for so many of us to be able to share our work, not only with you, but also with the leadership at NIA so that we can really advance some of these models and approaches to improve cognitive function in our elderly population as long as we possibly can. So with that, again, I'm so sorry I can't be there in person, but I'm glad that I have this opportunity to share our work, and I am really looking forward to the rest of the sessions today. Thank you.
Is Dr. Kaczorowski with us for the discussion session?
I'm here, Molly. Thank you.
Okay, wonderful. Thanks, Catherine. I don't know if you heard everyone clapping enthusiastically, but they were, if you missed it. So thanks so much. Really, that was a wonderful session, and we're now going to open it up for discussion and questions. Do we have the microphones? No. Okay. We'll get them now in just a moment. We're bringing microphones to the aisles for any questions. Is it on? Okay, go ahead. Yes, yeah. Thank you.
I have a question to Dr. McCarroll. I was wondering, I'm not an expert on studying the psychiatric disorders, but it sounds familiar from family reasons. Is this cognitive decline related to the psychotic drugs, or it's strictly related to schizophrenia? Because I believe it's very difficult to separate those in the schizophrenia patients.
Yeah, so there's actually been some really careful studies of what's sometimes called the prodrome or at-risk population of young people who don't yet have a schizophrenia diagnosis, are not yet on antipsychotic medications, but have symptoms that about a third of the time will progress to psychosis. And it's very clear in that population, A, that there already is some cognitive decline, but B, that cognitive decline is the best predictor of subsequent progression to schizophrenia. So to say, the cognitive decline actually starts years before the psychosis does, and years before antipsychotic treatment does. So it's again, culturally, we focus on psychosis because it's kind of like the more unique feature, but cognitive decline is the early and continuous and disabling feature of the illness.
Okay, wonderful. Thank you.
You're welcome.
Thank you. Yeah, and congratulations, wonderful presentations, all of them. That was amazing. I have a question for Steve. I have a lot of questions, but I think I'll just focus on one question. There are at least 5 or 6 transcriptionally different astroglial types, and I think over a dozen for glutaminergic and non-glutaminergic cells. So I was wondering if you have seen any specificity in the transcriptional glial versus neuronal types. And also, I mean, we know that during aging, there is simplification and selective vulnerability of these glial cells and neuronal types. So does it happen during aging that maybe that specificity gets swapped or inverted, or there is mislocalization of glia to neuron snap?
Yeah.
You know what I'm talking about.
Yeah, it's a good question. There's definitely astrocytic diversity, and that has a transcriptional correlate. So from the transcriptional profiles, you can readily distinguish, for example, the protoplasmic astrocytes from the fibrous astrocytes and the interlaminar astrocytes. The changes in SNAP are most apparent in the protoplasmic astrocytes, so the ones that are living in the gray matter, close to neuropil, and then among neuronal types, like as you said, there's many kinds of GABAergic neurons in the cortex, many kinds of glutamatergic neurons in the cortex. We actually see effects of SNAP in all of them. There are differences in the details. So I wouldn't say that it's like exactly the same in all of them. Certainly the presynaptic changes are very, very similar across them. And there's maybe a little more variety in the postsynaptic changes, which may also be a bit more pronounced in the projection neurons than in the inhibitory neurons. So it's definitely, they're all responding to SNAP. It's affecting synapses in all of them, but not in exactly the same ways. There's differences. It's just there are differences in the details.
Okay. And during aging, is there any swapping or
No. It's still same. They're transcriptional. The neuronal and the astrocyte transcriptional programs are still a mile apart from each other, and it's a layup to tell them apart.
Thank you.
So I'm just fascinated by the blame your mother situation. And what I was wondering, because some of the epigenetics is actually heritable, right? So we are passing it through generation. So if a mother passes the bad one to the son, and then the son passes it to the daughter, that should be the better one now because it comes from the father. So is there like an improvement when it passes through, when that X chromosome goes through the paternal lineage, goes through a male? Is there improvement in their epigenetics, or do we just kind of degrade our epigenetics?
Yeah, it's a great question. Whether this is heritable in a transgenerational way from mom or dad. And it hasn't been tested. That's the bottom line of my answer. But having said that, there is an epigenetic wipe of the chromosomes in the gametes themselves. So again, it hasn't been tested, and so one would predict it wouldn't matter, but we don't know. But there is an epigenetic wipe before the sperm and the egg come together.
So it's kind of like, like Huntington's disease, whether it comes through the paternal, maternal lineage, it's a very different outcome. So that's why I was thinking that maybe there's something here that actually passing it through the male, maybe because they don't go through the hormonal changes and they don't get that impact on their genome. Something like that. Just something to think about.
There is also slightly unrelated, but still related to paternal and maternal family history. There seems to be interesting evidence, and we have some data coming out, hopefully it's under review right now, supporting the idea that the maternal family history increases risk for higher levels of amyloid, and that's been published for a while as well in smaller studies. So there's an interesting component to that heritability via the paternal and maternal line.
Thank you.
All right.
Great. So really great, wonderful talks. And hi, Catherine, virtually. So my question is around, sort of the genetics and the interactions with lifespan, genetics, and cognition. So there's kind of a couple examples. One of them being obviously, sort of the X, right? The maternal X versus the paternal X. You have this beautiful data that we see. It doesn't matter what happens to the Y, 2Xs is great, they live longer, that beautiful study. And here you're showing us, if you get the X in the neurons, your epigenetic clock actually looks like you're accelerated, you're older. So that is really interesting. Somehow the 2Xs is really good for lifespan, but then if it's, if you do have that maternal X, it seems to be accelerated aging. And likewise, Catherine, in your studies, it's great because you have all this genetic diversity. In that context, do you see an interaction between lifespan, the genetics, and also the improved cognition? So I'll start with Dena, and then Catherine, maybe you can chime in a little bit.
So can you remind me what your question is?
Yeah, so you're basically the lifespan, right? 2X is great for lifespan, but the maternal X seems to promote aging in the neurons when you did the sequencing by the epigenetic clock. So does that tease apart, they're dissociated between lifespan and cognitive improvement or not? And I'm hoping also Catherine can chime in to give us a little feedback from her as well.
Yes. So we have not brought those two stories together. 2X extends lifespan, and then the maternal X is detrimental. But the idea is that because females have neurons that express mom's X and dad's X, that there's a heterogeneity there that would buffer age-induced decline compared to males who only have mom's X. So the idea is that the two X's, yes, there's mom's X and there's dad's X, and that mosaicism provides a buffer against potentially mothers-induced decline, which only have mothers-induced decline. So the sex difference is what makes the difference here, is mom's X in males, and then mom's X and dad's X in females provides a buffer.
Yeah, and I guess I can pick up from there. So in our recent studies, we did caloric restriction interventions and intermittent fasting in a very, very large population of outbred mice. And what we found was that the most successful intervention for lifespan, which was caloric restriction, extreme caloric restriction, extended lifespan by 40%, but the vast majority of those mice showed cognitive deficits at midlife time points. If you then go into those cohorts of mice where we dialed down the intervention, so intermittent fasting and 20% caloric restriction, these interventions, on average, in a genetically diverse population, extended lifespan. And then what mattered was whether or not you inherited a certain version of a glucose transporter, right? So I think that there are going to be environmental and, pharmacological interventions that will be, on either side of the, continuum for a population, not good enough, you know what I mean? And potentially damaging. And then that sweet spot is going to be something where we're taking into account some pretty strong genetic modifiers coupled to those interventions. This study that I'm talking about, the first author is Andy Willett, and this was only in female mice. So I just want to make sure I say that caveat. But and what's interesting too is it's like whether or not you're looking at it from an epidemiological standpoint, right? A population average versus taking into account individual genetics. So I think the verdict is out, and it's a really important gap and opportunity that we should pursue, I think. Thanks.
Thank you. Jen?
So thank you for excellent talks. I learned so much each time I hear you. I want to push the group a little bit further now that we're in the discussion to talk about some of the limitations that many of you pointed out in your talks about the cohorts of the brain bank and also of sort of the conceptualization of sex and gender. And what I want to ask you is, one reason for us to make that note and to talk about that is that inclusion is incredibly important, especially for understanding generalization. But that's not the only reason why we want to, raise these issues about diversity in our cohorts. The other reason is scientific. And I was hoping each of you could maybe take some time to talk about the scientific expectations for once those issues are addressed within the cohorts that you look at with genetic research, with neuroimaging research, with brain bank research. Once those changes are addressed, what are your expectations scientifically about the diversity that will then be presented? And once with Rachel specifically, I want to ask you about gender a little bit, that it's more than just a difference from sex assigned at birth, that there are forces, structural and social forces, that I'm hoping maybe you can talk about in terms of their relative impact to some of the other factors that you're studying.
Thank you so much. That is such a really powerful question, and it's one that actually is addressed in almost every time I speak. This question comes up, and it's a very difficult problem that I encounter because, as you would know, a lot of the work that I do involves secondary data analysis, and due to that, I end up very limited in the types of things that I can address and the answers that I can try to draw from a lot of the research questions that I have. So for me going forward, the biggest thing that I want to try to do is to change that paradigm, is to really think about collecting data and promoting and advocating for more transparent ways that data is collected. It's specifically when I think about sex and gender, for instance, because that's where a lot of my work is focused, is really being transparent about the questions that we ask and the inclusivity that we have in representation for recruitment. This is not exactly the way you said. This is not because of generalization, but because of the exact question that when I started doing sex and research, a lot of people pushed back on me and said, who cares? I put sex as a covariate in my analysis. But actually, it's not about, so let's say incidence rates are similar between men and women. It's not actually about the outcome. It's actually the journey. The way that the pathways that are involved to get to that endpoint can be very, very different. And without that representation and inclusivity, we don't have a precision medicine approach, full stop. It doesn't exist. On top of that, so what I was just referring to is mainly sex assigned at birth, but if we think about structural determinants of health, both from a race ethnicity standpoint, but also a gender standpoint, then, a lot of that gets ignored, particularly when you're thinking about either current data sets or just thinking about the sort of the medical conclusion of brain neuroimaging or something like that. There needs to be a lot more focus on the other colliders that can be involved, that can have a major impact to help to explain the outcome of interest. But I'll stop speaking too much and let other people have a go. But thank you for that question, Jen.
Yes, thank you for bringing this this very important and, as Rachel said, powerful question to bear. Well, I think we all are very ambitious, and we hope and dream, and then those dreams come true. But we start somewhere, and where we're starting with is sex as a binary variable in our mouse studies. But that is not representative of human populations and the human experience of gender, which is much more of a spectrum. And has multiple layers that you have mentioned and Rachel just mentioned. And I too have started from just the standpoint of wanting to study sex because women hadn't been included in clinical trials, and many of the drugs that we take today we know are effective in men, and we don't know what happens in women. And it turns out even something basic like aspirin was tested in men, but isn't actually effective for primary cardiac prevention in women. I mean, I think we should have pink box warnings on medicines if they haven't been tested in women. But we start from there, but that doesn't mean that we don't dream big and that we want to be as inclusive as possible and including. I think that's one objective, to be very inclusive going forward in thinking about gender as a spectrum, with different hormonal profiles, with typical and atypical sex chromosomal complements. There's many ways to do it. I think that it's still to be imagined, and there's still so much to do. And I thank you for raising the question. I think that's only one tiny part of it, was my answer.
Yeah, and just to add to that, I think that's such a really nice response. It's also inclusivity of ownership of the research. So being explicit about who exactly you feel that you're able to be asking questions of. Is it cisgender women? Is it, women with a uterus? When we think about the ways that we want to use the language to include people in this work and for everyone to feel ownership, but also we need to have more representation in the research side as well. That's the other huge thing.
Catherine, did you want to share something for this question?
I mean, I think from my perspective, I think a lot about the limitations of the animal models, right? The design of the transgene, the places that it's inserted, the Thy1 promoter. And then in a lot of the human cohorts, limitations on representation, different environmental contexts, socioeconomic status, like a lot of these things. And so I think the way that my group has been approaching it is really to focus on integrating the mouse and human data and drill down on the mechanisms that seem to be conserved across this species with the, you know what I mean? So that it's not addressing the issues that you really want to address, but it's getting at sort of like the biology that is conserved between those to start with. So I don't find, mouse studies or human studies alone nearly as useful as the power of combining the two.
Steve?
Thank you for that insightful question. So I mentioned the work that our colleagues are doing to try to increase diversity in research participation in the sense of like brain donation by families and things like that. But I think, as your question points out, that's diversifying like participation in cohorts is only part of a larger struggle, and in particular, and in some ways, it's the easier part. So I think a lot about this too. Like brain donation that's made at the end of a person's life, how do you know when what you measure, by however sophisticated a molecular approach, is due to genetics or is due to a life experience of structural racism and all kinds of things. And I think there's in general a risk in science, which is that scientists just tend to focus on things we measure well. Right? And we can measure, today, genetics is very sophisticated. We can measure to, multiple decimal points of precision what fraction of a person's ancestry is from each continent. We have a much harder time measuring, life experiences. And that's in particularly true with brain donation, where often the question even only comes up in the hours after someone's passed away. Right? So I think there is like a real kind of gift to science that an institution could make, which is to kind of get ahead of this and talk with people, maybe years before the end of life, collect other kinds of information, narrative information, clinical information, biomarkers, measurements of things, so that then, maybe several years later, if that person's brain is part of brain banks and being studied and things, things in addition to just like, their age, their chromosomes, their genetic ancestry. And you actually you might know how the things that you measure at a molecular level correlate with things that could be measured at a clinical level, because obviously the goal ultimately is going to be, we want to be able to make clinical measurements that tell us about what's going on in there, right? Well, and so if you could make clinical measurements in the years, toward the end of a person's life, and then, collect CSF, collect plasma, and all kinds of things, and then there's a brain donation that you can apply all these other kinds of analysis, you really start to be able to connect things that you can measure in the clinic to things that you can only measure in like a laboratory context. So I think that's obviously like, but I would love it if someone was, excited to like think about something like that, because I think it would be a real gift to science.
Thanks. Thank you. Randy, you still have a question? We have time. We have time. And we have time for you too.
So this is, I guess, to the panel, but the talk's inspired, and you alluded to it, Steve, so I'm going to push you on it. To take these discoveries, which are now potentially molecular pathways in the brain, and broadly explore them in cognitive aging, we need accessible biomarkers that are proxies for them and selective. And so you alluded to this as plasma biomarkers, and even things like to what degree there's paternal versus maternal expression. I have no idea of how to do it, but I've been shocked at how people have figured out to get things from plasma. So how far away and how do we accelerate that? And how do we generalize this so that people can be measuring all these different pathway-specific cascades from blood?
Well, I'll let you guys speak about the exon activation one, but specifically the plasma and biomarkers one, I think if we just, a program that was collecting those things in older people, and then also, who had, already consented to donate brains when they do reach the end of life, you really then could you could relate these things also to like a cognitive history and also like a history with discrimination and racism and all kinds of things that you just can't ask of samples in a brain bank. So I think that would be a powerful thing. I should say, you guys are the exon activation ones, or especially Dena. I should say, in postmortem tissue now, through an approach we've developed and that we're writing a manuscript, we can actually tell for every cell which X chromosome it's inactivated, because humans have so much genetic diversity. There's so many heterozygous sites on every chromosome. So it makes it possible to actually measure that in like every cell type. But of course, that's only at the end of life. That's not during life.
Thank you. Stan.
Yeah, questions for Stephen as well. So we go back to the SNAP data on schizophrenia. You're showing this kind of offset between the typically developing, I guess, and schizophrenia. The parallel lines are really kind of interesting. So can you help me kind of think through it? It doesn't seem like, or I should say, I would expect that if there was a difference in the rate of aging between the schizophrenia and the control group, that there would be a difference in slope, not a mean offset, right? So can you kind of help think through like what the implications of that are?
Yeah, I mean, I think it's that there was a precipitous decline earlier in life, but that precipitous decline does not protect one from also having further decline with age. As far as we can tell, are indistinguishable slopes, but you ideally do this in a lot more people to measure the slopes more precisely. But yeah.
So would the model be that there was some early event caused an immediate drop, and then they just continue on the same trajectory that everybody else is on?
Yeah. So there's really the cognitive decline. Exactly. So and this is work by other people, but the cognitive decline in schizophrenia seems to start a few years before the onset of psychosis, and kind of continue for a decade or so after that, and then to sort of stabilize. But I don't think it exempts one from the kind of further age-associated decline. Yeah.
Thank you, Sarah.
To just follow up on what you were talking about with respect to cognitive decline, how is that being measured with respect to memory, speed of processing, function? What's the cognitive decline you're talking about?
All of the above. I mean, again, this isn't my work. This is work of like teams of psychologists and things like that, but all of the above and more. I mean, these all kinds of, batteries of cognitive tests of processing speed and word memory and all kinds of things. Many of which overlap with tests that are given to older people to monitor cognitive changes.
And then I'm fascinated by this conversation about the relationships between schizophrenia and these associations that you're finding. Do you think the division of neurology and psychology is fundamentally to blame, that we're really only talking about this now?
Well, yeah, I mean, I think it's a porous boundary in some ways between neurology and psychology. I mean, things tend to, wasn't Dr. Alzheimer actually was a psychiatrist, I think, if I recall? And it's just like, things tend to be classified as neurology when there's like a really like very specific pathological finding, so like if it, and but it's like the distinction is kind of comes down to stuff like that and to history. So it's not that what we call like psychiatric illnesses or mental health illnesses are less biological. It's just that they may not have had their big pathological discovery yet. Like someone may not have, like there's not like aggregates or death of striatal projection neurons or something like that, right? And so, but it's not that it's less biological.
Well, yeah. And so the conversation that we're having about women and race and culture just seems to be so important. I know it's a comment, but I'll sit down now.
Thank you. So, and Robbie will let you have the last question.
Thank you. And first of all, fascinating. Thank you. Dena, I now know why my identical twin sister and I are aging differently. I am aging like my mother, and my identical twin sister is aging like my dad. So, yeah. So I would encourage you and Rachel to actually dig in and maybe do a twin study.
Thought you were going to say to use my twins, but that's good.
No. They're not old enough. But this would just be fascinating because I have been I have been so surprised. I'm going, why is this? Why is this? Why is this? And because it's so different. We've been identical until now. So there you go. Thank you so much.
Yeah, thanks for that.
Illuminating me.
Awesome.
All right. Thank you. Thanks, everyone. So we're running about 10 minutes behind at this point. The break was to be from 3:35 to 3:50, so a 15-minute break, and I'm going to shorten it to 10 minutes. So please be back at, well, I don't know, guys. What do you think? Should I? Well, I now see I'm losing more time. Yes? Okay, all right. Please return at 4 o'clock.
It's tempting to want to talk and walk around and so forth in between these. I know this is the very last session of the day. You've been very resilient and awake all the time. We intend to keep you awake through this one as well. But listening to all the presentations that have gone on this morning, I have to say that my introduction to this session is rather changed in that we can't just talk about cells anymore. We can't just talk about molecular pathways. We've got to talk about collaborations between cells and collaborations between cells with different X chromosome contributions and the networks between these cells. And so this session is going to give us a good overview of molecular pathways that are important for cognition and aging, as well as single and population responses in aging, and whole brain imaging and interactions across the brain in the circuits that are important for the cognitive processes that we all care about. Okay, come on in. I don't want the speakers to start without you guys sitting down. So thanks for coming back for the afternoon session. It's going to be great. The first speaker today is Catherine. Is Janine Kwapis, and the title of her talk is Local Dysregulation of the Clock Gene Pair 1 Contributes to Age-Related Memory Impairments. Janine.
All right, thank you, Carol. And thanks for making it back after that short break, everyone. I'm just delighted to be here, so I'm really grateful to the organizing committee for giving me this chance to share my work with all of you. So thanks for coming, and thank you for inviting me. So today I'm going to be talking about my lab's work, trying to understand the connection between the circadian rhythm and memory formation, and how these are impaired in tandem in the old brain. So we've known for a long time that there's a very fundamental connection between the circadian clock and memory formation. If you've ever experienced jet lag, for some of you this is hitting home right now, you've probably noticed that when there's a fundamental mismatch between your internal state and the actual time of day, you not only feel quite off until your body adjusts, but your memories are impaired. So sorry to say you're not going to remember anything about this conference. We also know that memory is not static across the diurnal cycle. It oscillates based on the time of day. So memories are really good at some times of day and are relatively poor at other times of day. And you can see in this chart a really interesting second observation, which is in normal aging and in basically every major neuropsychiatric condition, there's some component of memory dysfunction and circadian clock dysfunction. So my lab wonders if there's a molecular link that connects these processes. You can imagine if there's some mechanism or some set of mechanisms that are involved in both the clock and memory, impairment of this mechanism across the brain of an aging individual could contribute to symptoms of both memory impairment and circadian dysfunction. So when I started my lab, one of the first things we wanted to do is figure out when memory is best and worst across the 24-hour day in our model organism, C57 mice. So in the literature, it's really mixed when you see better memory and worse memory. And we're memory experts. We said we're going to control everything we can and really figure out when memory is best and worst across the 24-hour day. This work was all led by my very first graduate student, Lauren Belfi, who's been a real force in the lab in performing some of these systematic studies. So what Lauren did is she took young C57 mice and trained them in a very simple spatial learning task called object location memory, which you've heard about a little bit already. But the way we do it is we take a mouse, we get him very familiar with the context, and then on the training day, we expose him or her to two identical objects in very specific locations in that environment. So the animal gets 10 minutes to explore those objects. And then the following day, we test long-term memory for that task by simply moving one object to a new location. So mice are driven to explore anything that seems novel. If the animal remembers where these objects were the day before, they should preferentially explore that moved object that seems new and interesting. And if the animal doesn't remember that information, those two objects at test should seem equally novel. So it's a very simple hippocampus-dependent task, and mice are really good at this. It kind of capitalizes on something they're already doing. So what Lauren did, you can see in that schematic on the right there, she trained animals every 4 hours across the day-night cycle. So here, ZT0 is when the lights turn on in our vivarium, 7 a.m. ZT12 is when those lights turn off at 7 p.m. So she has 3 time points during the day, 3 time points at night, and she tests those animals exactly 24 hours later to really ask when memory is best and worst for this simple spatial task. And so what she finds is that we reliably see that memory performance is much better during the daytime or during the light portion of the cycle compared to at night. So on the y-axis here, you can see it's a discrimination index. That's simply a measure of preference. How much does the animal prefer that moved object, where a higher value reflects better memory? So you can see the yellow bar is the daytime points. The animals are showing a robust memory performance, whereas at night, those animals are showing really poor memory performance. And that's very clear when you collapse the day and the nighttime points. And I want to mention that mice are nocturnal, so perhaps unsurprisingly, we do see slightly more exploration at night compared to during the day, although it's not significant. But I really want to emphasize that the animals during the day that are showing less exploration are the ones showing better memory. So it's not the case that these animals are simply not exploring enough to learn. The ones with lower exploration actually have better memory performance. So Lauren actually went through and identified what phase of memory is oscillating, pulled out acquisition and retrieval, and kind of pinpointed memory consolidation as the phase of memory that's oscillating across the diurnal cycle. And so we said one critical component of the consolidation process is transcription. We know you have to have expression of a subset of genes for memory to be properly stored into long-term memory. So maybe what's happening is some genes that are critical for memory formation are being expressed properly during the day and are simply failing to express at night, leading to this consolidation failure. So what Lauren did next is a massive RNA-seq experiment where we tried to figure out in an unbiased manner what are all of the genes that could potentially support this oscillation and memory consolidation. So she trained animals at the same 6 time points, but now she sacrifices the animals an hour after training and simply looks at what those genes are doing. So every single one of these time points has its own time-locked home page control, so we can look at both baseline gene expression and how that oscillates, and then critically, how those genes change in response to that learning event. So we did a bunch of analyses on this data set, but I'm going to focus just on kind of one take-home analysis here, which is looking at all of the genes that are upregulated or induced by that learning event. So you can see in the yellow circle, what we see is a lot of genes. This is just focusing on the peak and trough of memory performance, so ZT5 or 12 p.m. and ZT17 or 12 a.m. So when the animals have the best memory at ZT5, we get 119 genes that are only upregulated by that learning event during the daytime. Those genes are not expressed at night. We have a really small subset of just 5 genes that are only upregulated when learning happens at night. And then we found 11 genes that are agnostic to the time of day, and they're upregulated no matter when learning happens. So we ran pathways analyses on the 119 genes to figure out the identity of those genes that are kind of our putative candidate mechanisms that could support that good memory we see during the daytime. And so we see some key players that have been implicated in memory, like the FOXO signaling pathway, the MAPK or ERK signaling pathway as well. But we were super excited to see that a clock gene came out of this unbiased analysis. So we were really excited to see the clock gene PER1, or period 1, came out of this unbiased analysis as potentially being able to contribute to this time of day effect on memory. So period 1, or PER1, is a circadian clock gene. It's one of a handful of genes that operates in this molecular negative feedback loop that takes about 24 hours to complete. So it's how your cells know approximately what time of day it is. And usually PER1 is studied in the suprachiasmatic nucleus of the brain's central clock. But we're seeing here that PER1 is not only present in the dorsal hippocampus, it's actually upregulated in response to a learning event, and the size or magnitude of that induction changes across the day-night cycle in tandem with memory. So we think, and this is just showing you qPCR for PER1, this is showing you the magnitude of PER1 induction across the day-night cycle. So you can see that during the day, again, those yellow bars, we get a big induction of PER1, and PER1 is repressed at night when memory is bad. So we think maybe PER1 is a mechanism that's connecting the circadian clock to memory formation. Perhaps it's operating locally in the dorsal hippocampus to exert some time of day control over memory formation and telling the hippocampus when memory should be robust and when they should be relatively weak. So in full disclosure, part of the reason we received PER1 is work I had done as a postdoc in a completely separate series of experiments had already identified PER1 as important for memory and contributing to age-related memory impairments. So I was with Marcella Wood at UC Irvine for my postdoc, and we spent a lot of time looking at the epigenetic negative regulator HDAC3, a histone deacetylase. And to make a long story very short, we basically found if we take old mice and we delete HDAC3 just in the dorsal hippocampus, we can improve memory or ameliorate age-related memory impairments. So in this graph, we're looking at behavioral performance in OLM. You can see the wild-type animal in gray is showing really poor memory for this task, so we see this age-related impairment. But if we remove HDAC3 from the hippocampus, these animals suddenly show beautiful long-term memory, and they look very similar to young mice. And we had run a completely separate RNA-seq experiment here to identify what genes were being regulated by this mechanism. And lo and behold, one of the same genes came out, PER1. So here we're just looking at PER1 expression in response to a learning event. So in the gray and black bar, we're looking at our old wild-type animals. Those are the animals that don't show good long-term memory. And in that case, in these old wild-type mice, we don't see any induction of PER1 in response to learning. But when we remove HDAC3 and show good memory in these animals, those animals now show a significant increase in PER1 in response to learning. So we think PER1 may be a mechanism contributing to age-related memory impairments. So putting this together with what I just showed you, we see that PER1 is operating locally in the dorsal hippocampus, possibly to regulate what time of day memories can be formed and stored. And in the old hippocampus, PER1 is repressed, possibly contributing to this sort of persistent nighttime state. These old mice don't show a PER1 induction even during the daytime when memory should be good, suggesting that maybe PER1 is limiting memory during the day, kind of imposing this persistent nighttime state and limiting memory formation. So our next question was, well, what happens if we manipulate PER1 locally in the hippocampus? We're going to leave the circadian clock itself intact and just focus on the hippocampus here. So we have these really fun CRISPR-A and CRISPR-I systems. So the first thing we decided to do is use our CRISPR interference system to knock PER1 down. This simply targets a couple of transcriptional repressors to the endogenous gene. So you're just reducing PER1 transcription from the endogenous locus. And when we do that, we're injecting directly in the dorsal hippocampus. We get a significant reduction of PER1 mRNA directly in the dorsal hippocampus. And you can see on the right there that impairs memory for OLM in a young mouse. So PER1 is necessary for successful long-term memory. And then what if we do the opposite and we overexpress PER1 in a case where PER1 is not properly expressed? What happens if we upregulate PER1 just in the hippocampus of these old mice that don't show an induction of PER1? Again, we're expressing this in the hippocampus. we get a significant upregulation of PER1 mRNA like you would expect. And what's really nice is we see these old mice that are showing memory impairments suddenly show robust long-term memory if we upregulate just this one clock gene in the dorsal hippocampus. So it seems like PER1 is possibly exerting diurnal control over memory locally in the dorsal hippocampus, but this is a circuit symposium, so I wanted to make sure I kind of emphasize for all of you this is not something PER1 is just doing in the hippocampus. It seems to be performing this role kind of all across the circuits important for memory, at least as far as we've tested so far. So I'm not going to show you the data today, but we've actually looked at fear conditioning memory as well, and we see very similar effects where PER1 seems to be exerting diurnal control over memory. I will show you some data from the retrosplenial cortex, however. So this work was all done by Chad Brunswick, another really creative and talented grad student in my lab. And he basically tackled the question of whether the retrosplenial cortex is also showing this pattern of PER1 expression. So for our purposes today, the retrosplenial cortex is really important in spatial and navigational memory. I've been interested in the RSC for a long time, so if you're also interested, catch me later. But basically in this series of experiments, Chad was just using it to test if PER1 is playing the same role in other memory-relevant brain regions. And so here, Chad just processed tissue from Lauren's RNA-seq and just looks at what PER1 is doing in the RSC. And basically what he finds is in the RSC, we get a very similar pattern of expression. Notice that you get a big induction of PER1 in response to that memory event during the day, but at night when memory is poor, that PER1 induction is repressed. It's still intact somewhat in the RSC, but it is dampened compared to during the day. Chad also directly compared young and old mice to see what PER1 is doing during the daytime. Because we know that PER1 is repressed in the aging hippocampus, is it also repressed in this cortical region? And so here what we see is that, I've lost my colors somehow, but in the young animals, notice there's a big induction of PER1 during the day, and in the old animals at the same time point, that induction is again massively dampened. And that's not true for every gene we looked at. CFOS expression is pretty well intact, so a measure of neuronal activity seems to be fine. And then finally, we asked, well, what happens if we manipulate PER1 in the retrosplenial cortex? Do we see the same thing as we see in hippocampus? And so Chad did the same series, knocked PER1 down in the RSC of a young animal. And what he finds again is that knocking PER1 down, even in the retrosplenial cortex, is impairing memory for this memory task. And then he did the opposite as well. He asked, what happens if we upregulate PER1 in the old retrosplenial cortex? And again, he sees that upregulating PER1 locally just in this one brain region can improve memory in this task and ameliorate these age-related memory impairments. So our story so far is pretty consistent with this idea that PER1 is sort of locally operating as an almost autonomous regulator of memory across the diurnal cycle, locally within these brain regions. But everything I've shown you so far has been in males. And I know what you're thinking, why didn't you do females? We did. The problem was that when we did our first kind of initial series of experiments, it became really apparent really quickly that we were going to see big sex differences. And we simply couldn't handle one more variable in those massive circadian experiments that already had 12 groups. So when Gretchen joined my lab about 1.5 years ago, she really tackled, well, what's happening with the females and how is it different from the males in a fully powered cohort? And so the first thing she did is she teamed up with Lauren and just looked at young and old mice and when their memory is best and worst across the diurnal cycle. And what she saw was really surprising. So for our young female mice, we saw no diurnal oscillation in memory. The young females show beautiful memory really no matter when we train and test them. But interestingly, if we look at our old females, suddenly we get this reemergence of that diurnal oscillation, and they're showing better memory during the day and worse memory at night. So of course, we next asked, well, what is PER1 doing? So this is our little cheat sheet just to remind you what we saw with memory. So again, the young females show good memory during both the day and the night. The old females show this reemergence of a diurnal oscillation. When we look at PER1, in the young females, we need to add more N here, but you can see a trend toward an increased expression of PER1 during the day that's repressed at night. So in our young females, PER1 expression is divorced from behavioral performance. Even though these animals have low PER1 at night, they're somehow capable of showing beautiful long-term memory at this time of day. But in the old females, now we again see this reemergence of PER1 possibly being able to exert diurnal control over memory. So here, PER1 is induced during the day and repressed at night in tandem with memory. So we're not sure of the mechanism behind this yet, but it's really exciting to us, and we think one possibility is that in the young mice, it's possible that estradiol is supporting that good memory at night. So as you've heard about before from Emily and others, estradiol does show a diurnal oscillation, but its major oscillation is on the order of 3 to 4 days. So we think perhaps the higher levels of estradiol at night in these young females, maybe that's capable of supporting good long-term memory, even though PER1 levels are low. And we know estradiol promotes memory formation, so that is a potential player. And then in the old animals, when estradiol levels precipitously drop, we think PER1 might reemerge as this mechanism capable of exerting diurnal control over memory. So we're actively starting to test this now, but I'd love to hear your thoughts on this as well. Okay, so with that, what I've shown you is evidence that in addition to its kind of canonical role in regulating the circadian rhythm, PER1 is moonlighting in these memory-relevant brain regions to exert diurnal control over memory consolidation. And repression of PER1 and the retrosplenial cortex in an old male mouse might contribute to age-related memory impairments. And then in the young females, we're not seeing this diurnal oscillation in memory, and we think this might be, this improved nighttime memory might be supported by some mechanism beyond PER1. And I didn't have a chance to share this with you today, but we're looking at the mechanism through which PER1 is promoting memory, and we think it might be doing this by regulating the process of memory allocation. So PER1 is enabling a lot of cells to be activated and incorporated into the memory during the day, and repressed PER1 at night might be limiting that process. So with that, I'd like to thank my wonderful lab. I talked about a lot of them as we went through it. Of course, I have a really great team. Of course, my funding sources, and I guess I don't take questions now. So thank you so much for your attention, and I look forward to your questions soon.
Thanks, that's great, Janine. Okay, our next speaker is Lisa Giacomo, Understanding How Aging Impacts Entorhinal Spatial Coding.
And thank you so much for the invitation to share our work today. It's been a real pleasure to be here. We just started working on aging a few years ago, so this has been a really wonderful event to be a part of. I've learned a lot, and I'm really excited for the rest of the talk. Okay, so I like to run, and this is a map of one of my favorite open spaces to run in the Palo Alto area, Windy Hill Open Space Preserve. And this is a map of the preserve. You can see all of the trails denoted and various landmarks denoted. But this is my cognitive map of the preserve. This is the map that my brain has created through interaction with this space and movement through this space. And the last several decades of work have really revealed that our brains contain all of the components that are needed to build this type of cognitive map of space. There are neurons that act as a type of longitude and latitude coordinate system and tell me the distance that I've traveled through this space. There are neurons that, some of them aren't showing up. There are neurons that tell me where landmarks are in this space. So for example, where I parked my car, as well as boundaries in this space so that I can stay on the trail. There are neurons that tell me what direction I'm moving at as I move through this space, as well as how fast or how slow I'm moving. And these together allow me to explore this space with this knowledge. Now, there are a few key components about this cognitive mapping system I want to highlight. The first is that once you have a cognitive map of a space, it's very flexible. You can take a novel shortcut through a space and have a general sense of where you're going to end up. The second is that it's very adaptive, so you can learn about a new feature in this space, like the location of a water source, and you can incorporate that into your spatial map. And the third is that it's highly evolutionarily conserved. So the same underlying neural substrates that generate this cognitive mapping system have been observed in species as varied as fish, flies, bats, rodents, non-human and human primates. So in my lab, by understanding this system in rodents, we hope that we can say something about how it operates in humans. Okay, so what does this system look like in rodents? Well, many of the components that build this cognitive map of space reside in the medial entorhinal cortex, which is shown here in blue. The first of these cells are called grid cells, and I'm showing you an example of a grid cell here, where what you're looking at is the bird's eye view of the activity pattern of the grid cell after a mouse has explored an open arena for about 20 minutes, where blue colors indicate spatial locations the cell was not active, and red and yellow colors indicate spatial locations where the cell was active. Now, you can see just by eye that in many ways this pattern is somewhat reminiscent of a longitude and latitude coordinate system. And grid cells have two key features that make them really ideal in terms of knowing where you are in space. The first is that different grid cells have what we call different spatial phases. So they are active in different spatial locations across the environment, even though they have the same underlying geometric structure. And what that means is that no matter where you are in space, at least a subset of your grid cells are active. The second is that they come in different spatial resolutions. So this is a sagittal slice through a mouse brain, and if you look at the dorsal tip of entorhinal cortex, there's a very small physical distance between the grid firing nodes, about 30 centimeters, and as you move to more ventral portions, this increases to several meters. And what that means is that you can actually combine the activity pattern of a very small number of grid cells, and as long as they have these different spatial resolutions, you can really efficiently decode your position in space or know where you are in space. So these grid cells exist alongside several other cell types that are defined based on their firing patterns observed as animals move through space. There are border and object vector cells that tell you where boundaries and landmarks are in the world. There are head direction cells that tell you the direction that you're facing in the world and act like a neural compass. And there are speed cells that change their firing rate on the y-axis with the running speed of the animal shown on the x-axis and act like a type of neural speedometer. Now, these neurons in the entorhinal cortex all interface quite closely with place cells in the hippocampus. These are neurons that are active in just one place in the environment and are thought to underlie episodic or autobiographic. Now, despite the really fascinating physiology of this system, there's very, very little known about how this system changes over healthy aging. And so that's what we really set out to look at. So why study the spatial navigation system over aging? I'll just give you a little bit of motivation. I think many of you probably know this literature better than I do, but there's a loss of orientation that often occurs in early, healthy, age-driven cognitive impairment. And determining and maintaining routes really allows people to retain mobility and quality of life and independence as we age. And the key thing for us, though, is that the neural system supporting navigation, the system that I just described to you, is extremely well characterized at youthful baselines. All of this data exists in young mice, so we have a really good idea of what these coding properties should look like in young, healthy animals. So the data set that I'll show you today is work all led by Charlotte Herber, who's a really fantastically talented MD PhD student in my lab. And I'm going to show you data that comes from mice that are navigating through virtual reality environments, where we've done electrophysiological recordings using neuropixel probes. These are probes that allow us to record from almost the entire entorhinal cortex simultaneously. And I'll show you data that we took from two different types of tasks, which I'll describe to you in a couple of slides, a random foraging task and a split maze task. And this represents mice that are young, aged, as well as a middle-aged cohort. They are split across male and female sexes. And I will just tell you now that the data that I'm going to show you today, we see a lot of individual mouse variability, but we haven't seen any systematic differences in terms of sex. So we started with this random foraging task to just ask the question of what's the baseline stability of spatial maps as animals age. And so we used this virtual reality task that we call the random foraging task. In this task, animals walk down a linear corridor, and when they see this sort of diamond-shaped pattern on the floor, they can lick in order to receive a water reward. And on each trial, the location of that reward changes. So in essence, they're kind of just randomly foraging through this environment. So old and young mice can perform this task quite well. So over the course of 6 sessions, both young and aged animals will accurately request reward when they get to that reward zone. But what about their neural activity? So this is an example of 2 neurons that we recorded, one in a young animal and one in an aged animal. And just to orient you, because I'm going to show you a number of plots that look like this, those top plots are raster plots. So the position of the animal is plotted on the x-axis, and different trials are plotted on the y-axis, where individual spikes from this neuron are represented as a black dot. The colored plot below, the blue or orange kind of oscillating looking thing, that is just the average firing rate of the neuron over those VR positions. And what I hope you can take from this just by eye is that in the young animal, this spatial cell is what we would call stable. So you can tell sort of by the fact that there are these long columns, this neuron is active in the same spatial position over hundreds of trials. Whereas if you look at the aged spatial cell, this neuron is not active in the same spatial location over trials. It's kind of drifting around. We can just quantify this by calculating a stability metric, and indeed we see that there is lower spatial stability in these maps of aged animals versus young animals. And this results in us being losing some ability to decode where the animal is. We can use a decoder and show that in the aged animals we can't decode their spatial location as well as in the young animals. Okay, so given that there's sort of this preserved task performance in the young, in this random foraging task, but there's this degraded neural activity, we wanted to try a behavioral task that was a little bit more challenging. And so we had animals run on this split maze task. So in this task, animals walk down a corridor that is the same for both context A and context B trials. But when they get to the back half of the corridor, they're presented with one of these two environments, context A or context B, which differ in terms of the visual landmarks in the environment. In addition, they also have to lick in a different location between context A and context B in order to receive their water reward. And this is unmarked in the task, but I've highlighted it with these pink and blue squares here, just for illustrative purposes. And this task has two phases. It has a block phase where animals either lick at A or where they do a bunch of trials in context A and then a bunch of trials in context B, and then an alternation phase where we go back and forth between the two contexts. And this is a lot harder for even young mice to do. I'll just tell you that in this cohort of mice, there are no visual impairments. So we were a little worried initially that maybe the aged animals just wouldn't be able to see the landmarks or something like that. But all of the mice that we used for this data set can see the landmarks. We know they can see the visual features of the task. Okay, so when we look at the block condition, this is where animals are running a bunch of trials in A or a bunch of trials in B. Young, middle-aged, and aged animals all improve their performance over the course of 6 sessions. But the aged animals are performing much worse. This is the center plot here. And this becomes really pronounced when we look at the alternation trials. So this is where the animals have to, on each trial, could encounter context A or context B. And here we see that aged animals really show significant impairment on this task. So what's going on then neurally? So here what I'm showing you again are these raster plots. And I'm showing you two neurons, two individual neurons from young and then middle-aged and aged animals. And I'm showing you an example from day 1 and then an example from day 6. In addition to plotting the trial numbers along the y-axis, if you look to the right side of the y-axis, you'll see these blue and pink bars, and those indicate the context that the animal's in. And what I hope you can appreciate, but I'll quantify this in the next image, but what I hope you can appreciate, at least just by looking at this data, is that in the young and the middle-aged animals, by day 6, you're starting to see these columns of activity. So there are stable spatial maps, and these columns of activity are also context-specific. So they're different for the blue trials versus the pink trials. Whereas in the aged animal, you see less stability, and you also don't see as much switching. There's not unique representations for each context. So this is quantified here. So first, I'm just showing you the stability of the map across different sessions, and aged animals here are in orange. And you can see that aged animals tend to not stable, they don't form these stable spatial firing patterns across days, whereas the young and middle-aged animals do. In addition, this stability metric really correlates with the animal's behavioral performance. So if we look at the performance of the animal, where here a higher number is better performance on the y-axis, you can see that the aged animals, one, don't perform as well on average, but in addition, when we take into account sort of the individual variability that we see in aged animals, it's highly correlated with how stable their maps are. Okay, so what happens now if we look at the whole population? I just showed you kind of individual cells, but we have recorded from tens of hundreds of thousands, way too many cells to even think about individual cells at this point. So what we can do is these population-level analyses, and that's what I'm showing you here. So these are what we call population-level similarity matrices. But all we're doing is we're taking a set of grid cells that we've recorded from, and we're looking at their activity on a given trial, say trial 1. And then we're just correlating that activity with every other trial, trial 2, trial 3. And if maps are very stable, if you have a really stable spatial map, what you'll get is a black square. And if you have a different map for each context, then what you'll see is this checkerboard-like pattern that you see in the young animal on day 6. So what that means here in that image is that the mouse has a very stable spatial map for each context, and it has a different spatial map for each context. But if you look at the aged animal, for example, on day 6, you still don't really see this very clear checkerboard pattern. They actually have 2 maps, but they switch between them kind of randomly. They don't pull up the map for context A when they're in context A. They don't pull up the map for context B when they're in B. So by day 6, young and middle-aged but not aged animals have this map alignment where they have a map for context A and a map for context B, and they're pulling them up at the right point in time. And what we see in addition is that the aged animals, they do still show map switches. We call this remapping. And this actually happens more frequently in the aged animals, and you can kind of see that by the sort of little tiny blocky patterns in that example of the day 6 aged animal. But they're not aligning with the context. And this, again, correlates with behavior. So when the map alignment is correct, when animals are pulling up the right map for the right context, then we see that the performance of the animal is improved for young and middle-aged animals. Okay, so in the last few minutes, I want to talk just a little bit more about grid cells specifically. So grid cells, as I said, aren't the only cell type in the entorhinal cortex. There are also these speed cells, and these are important for knowing how fast you're moving through the world. And so we also looked at speed cells, and what we found was that the gain of the speed signal was much higher in aged compared to young and middle-aged animals. What that means is that as an animal starts to run faster, these speed cells are increasing their firing rate much faster than they are in young animals. Well, why might that matter? I'm not going to go into all of the details, but there's been about a decade of really beautiful computational work that has illustrated how speed signals directly relate to properties of grid cells. And in these models, if you have an animal moving around in space and you record from a neuron in the control how large or small your grid scale is, how large or small that distance is between the firing nodes, by increasing or decreasing the speed signal. So the fact that we see this increased speed signal in this computational framework actually makes the direct prediction that we should see smaller grid cells. We should see smaller spacing between the firing nodes of the grid cells in our aged animals. And so that's exactly what we see. So what I'm showing you here on the x-axis is a metric of the spatial scale of these grid cells. And on the y-axis is the grid cell location along the dorsal to ventral axis. And in young animals, we would expect to see an increase in that scale from dorsal to ventral, which is what we see in the far left plot. But in our aged animals, we actually see much smaller grid scale across the entire axis. And this is a small but very significant shift across all of our animals. And this is very consistent with these computational predictions. So why would that matter? You know, why does it matter if you have a smaller grid scale? And I won't go into it today, but I'm happy to talk about it after my talk if people are interested. But we've done a lot of work showing that grid scale is actually very related to how stable your place cells are and your memory performance. If you start messing with this dorsal ventral gradient of grid scale, you tend to get less stable place cells, you tend to get degradations in behavioral performance in memory tasks. Okay, so just to summarize and also just kind of hint at some of the ongoing and future work that we're interested in. I showed you that aged mice exhibit this degraded, unstable, and drifting medial entorhinal spatial map, and that they exhibit this impaired flexible spatial memory. In addition, I showed you that aging impairs the ability of grid maps to accurately differentiate between these different environmental contexts. So aged mice are pulling up their map for A or B context kind of almost randomly. And I just want to highlight some future directions, and these are actually ongoing directions. Excuse me. First, we want to identify some of the transcriptomic changes that are occurring in these animals so that we can target them and then look at subsequent neural activity. And in a longer-term perspective, we're also interested in trying to intervene to enhance spatial cognition and coding. But on this first front, I just want to highlight that we have a really wonderful collaboration with Solvaleda, And we've actually taken the brains of the animals that I just showed you electrophysiological data from. We've given them to Saul's lab, and we're now looking at what differentially expressed genes are in those brains. So in this set of mice, we'll actually have electrophysiological data, behavioral data, and also gene expression data. And I think that'll be a really exciting set of data to look at in terms of relating this neural coding to other types of changes that occur with aging. Finally, I just want to acknowledge all of the folks in my lab, especially Charlotte Herber, who collected all the data that I showed you today. We're also a really collaborative lab, and so everyone in the lab has provided various components of infrastructure that contribute to all of the projects. And in addition, we post all of our data and code from all of our projects on our lab website. If you're interested in looking at tens of thousands of NeuroPixels recordings, that's the place to go. And after this paper is published, we will publish all of this data on our website as well. So thank you so much for your attention.
That was great. Lisa, thanks. Okay, our next speaker is Beth Buffalo, and the title of her talk is Age-Related Changes in Cognition in Non-Human Primates.
So thank you very much for the invitation to come today. Just like Lisa, I'm sort of new to the aging field, so I've only been doing this for a short while, and I've thoroughly enjoyed the meeting so far today. I've learned so much, so thank you for allowing me to be a part of this. So I'm going to tell you about some work that we've been doing looking at age-related changes in cognition, working with non-human primates. And just to point out, the students who conducted most of this work are shown on your left. So Ellie Bakatish, Sophie Landy, and also Casey Vanderlip. And then one of my collaborators is shown here, Courtney Glavis-Bloom. So just to give you a little bit of background, so as we've been hearing today, all mammals, I think, studied so far show age-related changes in certain brain and cognitive functions near the end of their maximum lifespan. And I'm going to channel Carol Barnes by saying that animal models do not develop Alzheimer's disease. So I think this is a sort of point that Carol has made often in meetings, and the data so far support that. Interestingly, for primates, similar behavioral approaches can be tested across primate species, and also sometimes the identical task can be used in humans and non-human primates. And so I think they provide an important animal model of normative human aging. And a lot of our work has been motivated by really some early studies from the late 1960s from Alfred Yarbus, who taught us that eye movements can really provide an important window into cognition. And so what I'm showing you here on the left in white is the scan path of subjects when they were asked to examine the painting freely. And then on the right, you see the dramatic change in the scan path when they were just given a different instruction, and now it's estimate the material circumstances of the family. And so with this appreciation, many studies now have taken advantage of changes in eye movements to tap into different forms of cognition. In particular, in my lab, we've been using these eye movements as an index for recognition memory. And what you'll see here on the left is a monkey scan path on two different viewings of this complex image. So in yellow, you can see the monkey scan path the first time he looked at this image. So you can see he made lots of saccades and fixations. And then in blue, you see his scan path the second time, where he just made a few saccades and then looked away. And as Janine was saying, just like mice have an innate preference for novelty, non-human primates and humans have an innate preference for novelty. And that's what's being shown here. They show this increased looking time for pictures when they're novel compared to when they're repeated. And through many studies in my lab and others, in monkeys and humans, this ability has been shown to be hippocampal dependent. So apart from just looking at these kind of novelty or familiarity responses, we also can now look at more details of the saccade statistics. And so that's what I'm showing you here. So I think these are not my new slides, so I'll have to just tell you a little bit. I'll have to have you trust me with the data that I'm going to tell you in a minute. But what these data are showing is the fixation duration across different fixations, so from 1 to 15, as the monkeys are looking at different images. And on the bottom line, you see their fixation durations for novel images, and then you see that they're enhanced or they're lengthened when monkeys are looking at images when they're repeated. And this is the data for young monkeys. And this is what we find for our aged monkeys. So they tend to show an offset in their fixation duration, so they're spending more time with each fixation as they're exploring the scene. And there's also less of a difference between the novel and repeated viewings. And the data that are not on here are the data from hippocampal lesions. And so what I'll tell you is that we've also performed this task with a group of monkeys who have selective lesions of the hippocampus, and they look very different from the aged and the young monkeys. So surprisingly, what they show is reduced fixation durations for both novel and repeat, indicating that aging is really not just recapitulating a full hippocampal lesion, but it's going to be more complicated than that. So I want to move on to another task that we've been using, trying to look at more sort of nuanced deficits that we might see in these aged monkeys. And this was work that was done in collaboration with Mike Arce, who I believe is here in the audience, really motivated by his task called the mnemonic discrimination task, which is a task of pattern separation and is shown to be impaired in aged humans and also appears to be dependent on activity in CA3 region of the hippocampus as well as the dentate gyrus. And the way this task works in monkeys is they're seated in a chair and they're holding onto a response bar while these images are presented on the screen. And the monkey is rewarded for paying attention to the first image that's shown at the top there and sort of ignoring all of the other stimuli that come on that don't match, including the one at the bottom that looks very close to the first one. And then they're rewarded for releasing the bar only when they see the exact match of that first stimulus. And so on the left row of trials, you can see what are called the lure trials, where you have this one stimulus that's very similar. And on the right are the foil trials, where there are no stimuli that are particularly similar to the choice. And we can look at their performance across two different ways of kind of looking at pattern similarity. So in the top, you can see similarity that's sort of distinguished into these three categories, high, middle, and low, based on features that were determined from human viewing. So these are the kinds of groupings that Mike Arce and others got when they performed this test with humans. And what we can see in red is that the aged monkeys show significant impairment, particularly for the middle and high similarity stimuli. And at the bottom, this was a different way of discriminating because we don't know if monkeys showed the same kind of discrimination as humans. So now this was using computational modeling to look at the perceptual similarity of the stimuli, irrespective of any semantic similarity. And this also showed impairment in the aged monkeys for these perceptually similar stimuli. Importantly, you probably noticed for the foil trials, the aged monkeys perform just as well as the young. So they can be trained to perform this task and show a normal memory performance, but it's when you tax it by changing the perceptual similarity that we see these deficits. I'm going to skip this one and go on to this. So another task that we've been using looking at working memory is called the delayed recognition span task. And this is work that we've done in collaboration with John Reynolds at the Salk Institute and Courtney Glavis-Bloom, who I mentioned at the beginning. And what they've done is trained marmosets to perform this task using a touch screen that's positioned inside or just on the edge of the marmoset's cage. And we do the same thing with our rhesus macaques, so performing exactly the same task. And this is kind of like a game you may have played when you were young. I think it was called Merlin or something, where the monkeys touch the square to start, and then one stimulus comes on, and they have to touch that stimulus to get a reward. And then there's a variable delay, and then two stimuli come on, and the monkey gets rewarded for choosing the new one. And this continues until the monkey makes a mistake and chooses a stimulus that was not the newest one. And then we count how many stimuli had been presented before the mistake, and that indicates the monkey's span for that trial. And so at the bottom, you can see the performance of both groups of marmosets on the left and rhesus monkeys on the right across different ages. And what we find is that the aged animals tend to show a more difficult time learning to perform this task and have trouble reaching the same span length as the younger monkeys. So particularly for the rhesus monkeys that were tested in my lab, you can see we had 3 aged monkeys, all over the age of 20. And there was quite a bit of variability, which I think we've heard a lot about, increased variability with aging, where one of these monkeys actually, one of the aged monkeys actually performed just as well as the young monkeys after additional time of training, whereas 2 of the aged monkeys could never reach that higher level of performance. And then this is just looking at different measures of learning, so both in terms of trials to reach above chance performance, the max learning rate, and their final span length. You can see that there are correlations decreasing with age in terms of their performance. And this is true for both marmosets and rhesus monkeys performing exactly the same task. And so what Courtney and John did after they trained the monkeys and looked at their performance on this task, they also wanted to see if there were changes in the microstructure of the prefrontal cortex that could maybe be underlying some of these changes in performance. And so what they found in the dorsolateral prefrontal cortex was that there was an age-related loss of synapse density or reduction in synapse density, also a reduction in Bouton density, but no change in the number of synapses per Bouton. And also, for those, in terms of the synapse density and the Bouton density, there was no difference between marmosets that were unimpaired versus aged marmosets that were unimpaired versus aged marmosets that were impaired. But then they started looking at other measures of the microstructure around the synapse. And what they found was that for the young marmosets and the age unimpaired, there was this correlation in scaling between the size of the Bouton and the size of the mitochondria. And remarkably, this was not true for the age-impaired marmoset. And so that's what you can see at the bottom, where you see this large Bouton, and in purple, a really tiny mitochondria. And so their idea around this is that perhaps this sort of mismatch between energy balance is what may be underlying some of the synaptic dysfunction that they're seeing in the aged impaired marmoset. Okay, so the last experiments that I want to tell you about are some new ongoing studies that we're doing in the lab, looking at memory-based foraging. And it really, I think, follows nicely from the work that Lisa was telling us about, her beautiful work showing these spatial representations in the entorhinal cortex and hippocampus, and how they're elicited through foraging, and then also how they change in aging. And so we can actually train monkeys to perform similar kinds of foraging tasks. What we do is we have them trained to use a joystick to navigate a virtual environment. And in these new experiments, we're training them to do 2 different kinds of foraging. So one that's based on the color of the stimuli. So in the top view here, the monkeys get rewarded at different values, so either a high, middle, or low value for foraging the spheres that are of different colors. And then in a second version of this task, we have a spatial version where the spheres are all the same color, but the monkey has to learn where there are zones unmarked within the environment. So there are zones in which they'll receive high reward, medium reward, or low reward. And so probably the best way to explain this is to show you a video. And I'm not sure if I can click through this. I may have to see if this works. Perfect. All right, so this is the first trial of the monkey performing this color task. And you can hear the little beeps are the sound of the monkey getting reward. So that was 1 beep for red. You got 2 beeps for green. And I said he, this is actually Manchita, so the female monkey. And then she gets 4 beeps for the blue. And the key is that the trials are only 30 seconds long, so she can't forage all the stimuli in one trial. So she has to learn to be efficient. And you can see just 14 trials later, she already readily knows which ones are the high reward objects, and sort of makes a beeline to those. The little white and blue dots that you see are her eye movements, and so we can learn a lot about her foraging strategies and planning by looking at both her joystick movements as well as her eye movements. So you see she got all the high reward ones and then went to one of the medium rewards. And then at the same time, in the same session, we can alternate trials and have the monkey perform the spatial task. So I just want to show you some learning on that. So this is the first trial of the monkey in the spatial zone task. So all the stimuli are the same color, and the monkey's just foraging, trying to identify or getting higher reward for stimuli in a certain zone, which she hasn't gotten to yet. So she starts each trial from a different location. So she, in order to do well, she really has to build up an allocentric map of this environment. And so she found the zone with the high reward there. And now this is just 15 trials later, again within the same session, just about 10 minutes later or so. You can see she immediately orients to the high zone. She knows where she's going and has learned this task. So this is a young monkey. We've tested several young monkeys on this now who learn this task very quickly, and they can learn to do multiple maps within a session, either like I'm showing you here, a spatial and a color map, or 2 color maps and a spatial map. They can learn all of these within a session. And what this allows us to do is now probe neural activity that accompanies that learning. And so in some of our experiments, we've been recording from multiple probes positioned in the hippocampus with multiple channels, so we can record from an ensemble of hippocampal neurons all at the same time, so up to about 100 neurons simultaneously with these probes. And we can start to look at the network activity that might be supporting performance on this task. And so just to give you a sense of one way that we're analyzing the data, if we think about if we were recording from just 3 neurons, and we plotted the activity of each of those 3 neurons in this space, this 3-dimensional space, and then we sort of moved it through time, we could get a sort of projection, a low-dimensional projection of that ensemble activity as the monkey moved through the trial. And so this is what we're doing. This is showing the sort of projection of that low-dimensional activity of the ensemble now of 100 neurons projected into PCA space, and or using PCA projected into PC space here. And what you can see is that for the color task, now we see that the ensemble activity kind of groups according to the value of the stimulus. If we look at the same neurons now recorded while the monkey is performing the spatial task in alternating trials, we also get kind of a grouping of the ensemble activity that reflects the value schema now for the spatial zone. Now, an important control or an important question is, do we get the same kind of representation that across the 2 tasks? And so what I'm showing you here is data from the color task, but grouped by the spatial zone in which the monkey is positioned. And there's really no separation there. So what this is telling us is that the neural ensemble is really segregating across the 2 different schemas. And I think this could be really similar to kind of showing this change across the different maps, and something that we'd really like to now test in aged monkeys, also with recordings of these ensembles. So I'll just end with some challenges and opportunities, and really a sense of where we might be going next. So I think with these kinds of techniques, we can identify changes in circuits and network function associated with age-related behavioral changes. We can use behavioral tasks that are similar, in some cases identical, across non-human primates and humans. And I think these sort of naturalistic type tasks, like this foraging task, really might help serve as a bridge between humans. So I'll end there. And I just want to thank the members of my team who contributed to this. I mentioned Ellie Bakatish, who did all of the aging work with the viewing, and then Sophie Landy and Solana Fernandez, who did the work with foraging. Thank you very much.
Another great talk, thanks. And our last speaker is Liz Krastel, and she'll speak to us about human navigation during midlife, sex differences in behavior and circuitry.
Thanks so much for having me here today. Thanks for the organizers and Carol for inviting me. It's been a real privilege to be here to hear so much fantastic work happening and to just see all the exciting work that's happening. And thank you so much for sticking through for one more talk. I know it's been a long day. So I'm also relatively new to aging research because my primary work is on spatial navigation. This is something we do every day, all the time. And, you know, it's a very everyday behavior. And so sometimes we don't think about it, but in fact, it's sort of critical for everything about getting around from any place, finding your way here, finding your way to your room, and exploring a new city, anything like that. So we do this sort of thing a lot. But what's also really important with navigation is that a lot of the circuits overlap with those of episodic memory. So primary areas include the hippocampus, entorhinal cortex, retrosplenial cortex, as well as basal ganglia areas in terms of gray matter. And then we also think a lot about the white matter, and I'll be showing you some white matter tracks today, particularly looking at fornix and cingulum bundle, which are outputs of the hippocampus, and a lot that connect a lot to prefrontal areas, which are also critical, as well as others. So we're trying to think, again, about a lot of the brain, whole brain stuff, as well as gray and white matter, and thinking about how they interact. What's also really exciting, and one of the things that I love, but it's also a big challenge for navigation, is that it involves so many different cognitive processes. So it involves perception of your self-motion, of things in the environment, attention to certain properties of the environment, or other factors, memory of various kinds, both working memory and episodic memory, decision-making about thinking about where you're going to go, how you're going to explore, and so forth. So a real fun challenge for us to think about how it works all together. And perhaps most importantly for today, there's large individual variability. I'm sure we all know people who cannot find their way out of a paper bag, and there's others of us who could be somewhere 20 years ago for 10 minutes and would still remember the layout of that building. So there's this huge variability when we're thinking about how people do this. That's going to be really important, and we're kind of leaning into that variability to help understand these circuits, and in particular, what happens when we age. So now connecting this a little bit to some of the aging work, navigation has also emerged as an early marker for detecting dementia risk in humans, and especially thinking even potentially decades before any symptomatic onset. There's been some evidence suggesting that the change in our grid cell-like representations in our entorhinal cortex, which we've just been frequently, is related to ApoE status, and then it's eventually related to some changes in behavioral performance, even again in young adults. Women have twice the risk of dementia. We've talked about this, whether or not that's, to what extent that is manifested in different ways, and the sources of that is something that's important, but there certainly are these large sex differences when we think about dementia risk. And again, particularly when we think about navigation, sex differences have been frequently observed. And in this case, we're actually looking at males tend to have the advantage in navigation. So that's maybe bucking the trend that was talked about earlier. And of course, the nature of these sex differences is highly uncertain. There's a lot of social factors and experiential factors, as well as sex biological factors that might be part of it, but that's something we're interested in discovering more about. And then sex hormones have this huge influence on the organization of the hippocampus as well as prefrontal cortex and a lot of the areas that are involved in navigation. So kind of putting this all together, we want to find out a lot of information. So thinking about how, in particular, women might be affected by changes in sex hormones during menopause, we kind of want to think about this all together. So first of all, can we use navigation as a tool to determine risk for dementia, especially for women? How do navigation abilities change during early aging, especially, because that's what we're going to focus on in this talk? Do those circuits are associated with navigation change as we age? And then do those sex differences in navigation change during the course of aging between young and midlife? And then finally, do those changes relate to menopause? And so we're particularly interested in this menopausal transition, again, as a real inflection point, thinking about how it might be important for understanding and sussing out these individual differences combined with sex differences, combined with this complicated behavior. So there's a lot to parse out, but we'll see. So first of all, this work has been done in conjunction with Emily Jacobs and Mary Haggerty at UCSB. So we've been really combining forces for a lot of this work I'm going to talk about today. So what we have here is early aging, so midlife, so 45 to 55, so not that old, but looking again across this menopausal transition. So we have some younger adults that we're going to compare to some of our findings to as well, and we're looking at pre-peri and postmenopausal women for much of this. So we're looking both at chronological and endocrine aging, and hopefully we can piece some of that apart. So we have three main tasks. I'm going to talk to you about them in turn. And they each talk about different aspects of navigation, because navigation, as I sort of alluded to, is not a singular entity. And so when we think about individual variability, some people might be good at certain aspects and not at others, and vice versa. So there's a lot to think about as we sort of put it together. So our first task is a wayfinding task, and I'll actually just start showing the video because that's the best way to see it. So we have over here an overhead view of the maze, which just has 9 objects in it. It's a virtual maze. As you can see what's happening, this is a person navigating in it. They just press a button at each intersection to say whether they want to go left, right, or straight. So it's a really simple interface, and they just get around with this. And first we give them explorations. They're freely allowed to explore wherever they want for about 16 minutes during this maze. And we just tell them to find all the objects and learn their some paintings on the walls of the objects, as you can see here, which provide some landmarks. And then during the test phase, we ask them just to go from one object to another object. So example, in this case, they started at the spaceship and would go to the umbrella. That is, in fact, the umbrella. They got it. But they don't know that they got it right because we actually replace all the objects with red spheres, so it limits the feedback. So people can and do get nothing right. But there's also people who get 100% right, and so we see a huge variability here. So we had cohorts of both young adults, young males and females, and midlife males and females. And here, just first looking at the behavior, and this work was done by Xu-Ying Yu and Santa Barbara, and we found that our young males are doing quite well. Our young females are doing not as great, but our midlife males and females are both doing pretty not so good here overall. So it's a pretty substantial decline here in performance in this task with midlife, and again, 45 to 55. We also looked at some of the exploration behavior, which actually some of our folks alluded to earlier, looking at how people explored this environment. So just seeing whether or not their performance differed, but how do they approach this task? Are there strategy differences? Are they thinking about it differently? Can it be reflected in terms of how they're doing this? So we found that the midlife adults actually visited the objects more during the exploration times than the young adults. But they spent less time in the hallways. So those object visits were increased, but their hallway visits times were decreased. And there was a sort of a qualitatively different pattern in that respect. And we then did a principal components analysis on several of these variables of exploration patterns. And we found there were sort of 2 main variables. One that was related to quantity, essentially, like number of button presses, how many times you visited objects, and things like that. It was related to performance in midlife. It was not related to performance in our young adults. But we also have an exploration quality variable, which is really thinking about how evenly you explore the environment, how often you how long your hallway visits are without visiting an object, some other variables that are sort of related to how you might approach this task as a sort of more of a qualitative measure. And so we found this was also related to performance in midlife, and only that one in your young adults was related to performance. So I want to show you a little bit about, it's a separate cohort, so just as a little caveat, we can't directly compare this to our older adults, but just to give you a flavor for it. Our thing's not working. We did some relationships with performance in our younger adults. And in this case, we found that navigation performance in our younger adults was correlated with white matter integrity in the cingulum bundle and fornix, which are those hippocampal outputs in our younger adults. And so we found, again, that really strong relationship to those limbic structures going on. It was also correlated with total hippocampal volume. There's been sort of a controversy in the human neuroscience literature about whether spatial tasks are related to hippocampal volume. We did see it here. It's not a super strong correlation, but it is related. We also have some relationships with some hippocampal subfields. Now, in contrast, when we look at it in our midlife adults, we found no relationship with total hippocampal volume and performance in this task. We also just to clean things up, but we also, the results are pretty similar with that. We also did an controlled for an associative memory task as a covariate to control for general memory, so to make sure that these were specific to the spatial part and not sort of memory decline. And we found here that performance, when we did this, performance is correlated with white matter integrity and cingulum bundle, but not fornix, but corticospinal and corticostriatal tract. So a little bit of a different pattern. But perhaps more interestingly is the sex difference in our midlife adults. We also saw some sex differences in our young adults, but the midlife one is even more striking. So in this case, we saw that females overall showed a sort of a positive relationship with performance in the fornix and white matter integrity and performance in this area, which is what we saw in young adults. But our males actually have negative correlation, so a significantly opposite direction effect. And so we definitely see some interesting things happening here. And we also saw some differences across menopausal staging, such that our premenopausal women looked more like the overall women, but our postmenopause looked more like the men. And so we see, again, differences in the directionality of this relationship between performance and white matter integrity. Our second task is a path integration task, which is a continuous updating of position and orientation during movement. And we often think about this in a desert environment or a place without landmarks. And so it might be something like tracking a location where you're going on a circuitous outbound path and then have to walk straight home. We often do this in sort of virtual hallways and virtual deserts. We actually take away all the environment for our participants here. It relies on entorhinal cortex, hippocampus, and other medial temporal lobe regions. But previous studies have found sort of mixed results in integration deficits and aging, perhaps because of what type of information you provide, whether you provide just visual information, just proprioceptive information, or both. Here we're going to provide both, and so I'll show you what that is. So this is what people are seeing. So there's two eyes, which is why I saw two things. And so that's their start location, but then we remove it, and the people are walking in a virtual desert. And what's happening is we're taking people on a circular path, and we're just walking them in a circle. And all they have to do is tell us when they think they return back to the place they start. So it's really simple to understand, difficult to do. And you can see Xiying practicing there. And so all they do, we have several different circle sizes, and we just return back to the start in this virtual desert. So what did we find here? In contrast to sort of these stark differences, both across sex and age, in our maze task, in our path integration task, we found that males tended to overshoot and females tended to undershoot. So that 360-degree mark here is the dotted line. Women tend to be overshooting and men tend to be undershooting, which we sort of suspect is a strategy. But in terms of overall like error, they're actually very similar. There's a little bit in the young men, but overall there's no main effects here of age or sex. And so in terms of getting to the target, they can do it very well, even at midlife, no matter male or female, but there just tends to be, we're thinking a strategy difference in terms of how they're presenting their performance. So given that there's no strong behavioral differences, what's perhaps most striking is that we are going to see some differences in the brain. And so we found that total hippocampal volume in midlife was associated with that degrees traveled, but not errors. And so there was no relationship with position error, how far off you were, but it tended to be more related to whether you overshoot or undershoot. In the young adults, we do find a relationship with total hippocampal volume and smaller errors, but not with degrees traveled. So there's definitely something happening a little bit different in terms of the circuitry. We also found an interesting relationship between testosterone in females, only, not males, with degrees traveled and with undershooting. So we're kind of unpacking that. Then when we look at the white matter differences, we find that our young adults display this positive association between white matter integrity and path integration ability in terms of errors. And so it's lower errors, but it's so that better fornix in white matter integrity is related to better performance and lower errors. It's one of the cleanest, prettiest fornix results I've seen. In contrast, our midlife adults show a negative association with the fornix. So it was a very different pattern in terms of what they're doing. And we also see some differences in the midlife between males and females, differences in terms of white matter regions associated with those path integration abilities, in terms of the circuitry that they're sort of related to across the board in addition to fornix. Our young adults, we don't have enough sample to do sex differences, but we will in the future. So our final task is a strategy task. And in this task, it's a dual solution paradigm where they follow a route 5 times in a virtual maze, and then we ask them to go from one object to another. And we don't tell them anything about how to get there, we just say go from one object to another. And they can either take the route that they know, or they can take a new path, and they can just choose that. And so we ask whether people are going to take the route they know or the learned or new. We find, first of all, that people tend to get to the goal pretty well. There's a little bit of a decrease in our midlife adults, but overall people can successfully do this. They see the objects are still there, so they can usually get there eventually. But in terms of this shortcutting, we find that really only the young men, the young males are taking the shortcuts, that both our young females and our midlife males and females are taking the learned route much more than the shortcuts, and our young men are only doing that. So then let's relate that to the brain. We found relationships with actually some of the sex hormone levels that we were taking, so increasing FSH. So FSH increases during menopause, estradiol decreases. They kind of go in opposite of each other. So decreasing estradiol and rising FSH across this menopausal transition are associated with increased reliance on habitual routes in midlife women. So as you make that menopausal transition, you're more likely to rely on the routes that you know. When you look at the white matter overall in our midlife adults, white matter integrity in that fornix, again, is associated with taking more shortcuts in our midlife adults. So our next steps to think about this, we really want to expand this now to a more complete spatial battery with more immersive VR tasks. So including a complete lifespan, so not just young and midlife, but to fill in some of those gaps, look at older adults, so really spanning the whole aging spectrum, including some more tasks of general cognitive function, as well as some other navigation tasks that are happening. And just as a plug for immersive VR, that's my lab there at Irvine. It really helps with motion sickness and with providing proprioception and vestibular cues. So thinking about doing some of this work on a desktop versus an immersive system is really important, especially when you think about aging and sort of the changes that you might occur with ability to integrate body-based information. Of course, hormone assay and AD risk factors will be included with a nice imaging structure and function. And we actually are including this grid cell task, so a measure of grid cell-like function in the brain, so we can help monitor whether people's grid cell stability is going to be related to this performance in this task. And of course, because we're interested in looking at this across the menopausal transition, we hope that this will be the first cohort in a longitudinal menopausal study, combining with some of the other work that's been doing, that people are doing across other sites, but to be involved in thinking about how this is going to happen long term. So just to summarize, we wanted to ask whether navigational abilities change during aging. We do find that some are impacted, but not others. And the circuits that are associated with navigation seem to change when you do cross-sectionally across our young and midlives, that some, even though we have some similar behaviors, there are some differences in terms of the systems and the circuits that are being relied on in, rather than hippocampal systems. Sex differences do sometimes occur for some tasks, but not others, and the circuits seem to differ for men and women, particularly when looking at the maze task and loop task. And then do changes in navigation ability relate to the menopausal transition? We do see this in a couple places, relationship, especially with strategies and with some of the menopausal staging in terms of the brain, relationships between brain and behavior. And then the big question that's still the long-term outstanding question is whether we can use this as a tool to determine dementia risk. That's where we're going long-term, and hopefully we'll get there soon. So thanks so much for your attention. Thanks to my collaborators. All the people highlighted in bold were people who worked on this stuff, and particularly, again, Emily Jacobs and Mary Haggerty at UC Santa Barbara, so we can really, again, do a lot of work across sites to think about where we're headed. Thank you.
Thanks so much, panel. While I wait for people to come up and ask questions, I wondered if what popped to my mind is what you think is different about the circuits that you're looking at, Lisa, in your mice that show grid cell instability and your solid path integration findings, Liz.
Yeah, I was interested to see that. So I wrote that down. So I'm not sure. So, I mean, we'll have to see also because we don't yet have the grid cell markers for the relationship with our midlife adults and our younger adults. So in terms of the brain circuits that we're getting that, we're collecting that now. So we'll see if that is affected. Maybe, I think one thing that's really possible is that people are changing their strategies and sort of how they're approaching this task. And so that it may be that, some are relying on more of the sort of grid cell path integration. Others are trying to do something different. We are seeing some differences in a big individual differences just in the young adult study, looking at strategies and how people are when we ask them their strategies, how they're relating to that. And then we do see some differences both in sort of in the behavior right now. So.
Yeah, and I'd also say, I mean, when we think about grid cells, we don't think just about path integration. So, I mean, we think about grid cells as also providing information about position in space. And I think for path integration itself, there is probably sufficient coding to do a decent job of path integration. The real detriment we see is that the grid cells seem to be assigning the wrong map to the wrong or not to the wrong context. They just don't seem to be able to pull up the right map for the appropriate context. But they do show a degree of stability that, I mean, you can still decode the position of the animal. You just can't do it as accurately.
It's just harder to ask your mice what strategies they're using as well.
That's right. Yeah, and we haven't done a path integration task in our aging animals. It's something we could do, but yeah.
So important to be able to identify that if you're trying to compare circuits and say that the behavior is changing, but what if just the strategy is changing? Yeah. Beth, do you have any insights from your monkeys?
Not yet, but hopefully we'll get there. I mean, I think with the foraging task allowing us to look at learning across, lots of different schemas as well as measuring eye movements, I think we'll be able to get a sense of monkey strategy as they're learning the task, and then, who knows if we'll see changes with aging, but it'd be fun to look.
Yeah, this is absolutely great symposium, partially because you also start to imagine how reading out representations in brains relevant to memory and navigation can be studied for the first time. Lisa, I had no idea how far that work had gone, and seeing the structure and how it changes with aging. And I guess it relates to your question. It didn't, and you kind of answered it, it didn't look random. There were periods of brilliance in those plots where there looked like structure. And I guess one question I have, are you able to quantify that to ask the question at a more micro level? Are there actually epochs where the representational state is more like a young animal? And the reason is because it could speak to the mechanisms of breakdown, because if it's just random all the time, that's one class of explanation, whereas if there are moments where the representational state is stable, but it doesn't get there, I kind of, in my own mind, kind of looked up other buckets. I'm just curious, can you read that out and predict like periods of accuracy from the data?
Yeah, that's a great question. So sometimes the representations are accurate, but just on average, they tend to be more randomly pulled up than the young and the middle-aged animals. But there's high individual animal variability. So we have one super ager mouse that has like beautiful map, like it pulls up the right map. It's an old mouse, but it pulls up the right map when it's supposed to, and it behaves brilliantly. It's like, does the right behavior. So there's also a lot of individual mouse variability. I just showed you one example of one mouse on one session. The thing we haven't done is like when there's map coherency, do we see kind of like trial by trial performance or degradation? That's something we could look at. I think there's a little bit of complexity too, where the aged mice tend to start to kind of give up a little bit at some point. And so there's a set of things that are happening, I think, behaviorally. But we haven't yet looked on like kind of a trial by trial basis.
So have you given Saul your super aging rat? Mouse?
Yeah, so we have that brain banked, but we have not we haven't run the genetic analysis on that one yet. I'd like to know what that mouse has in its brain.
In the middle. You were first. I can't see anything. We're blinded by this light shining.
That's okay. I'm not the one shining anything, but so thank you for those interesting talks. I had a question, particularly in spatial navigation, about have you considered I kept thinking of video games. And I kept thinking of it because there's an issue of training, that some people might actually be trained, particularly when we talk about younger, so age, and also gender, male versus females. And I wonder whether you've looked at, depending on the background of whether people, whether in humans played, because I was looking at the maze, and I said, well, you know, the studies that show that actually provides training, and how much of it is influencing, I don't want to say confounding necessarily, but or stratified by that in terms of.
Sure, yeah. So video games are a super important question, both in terms of thinking about how people might be learning different things, or it's just like an enrichment activity, but also thinking about for our test specifically. So we typically get information on video game play. We didn't get that in our midlife cohort, but we do have that on younger adults. And we do see some relationships for some stuff with video game play, and there certainly are some sex differences in terms of men and women playing video games. What's interesting is that we were just looking at this for the young adults in that maze task and looking at mediation analysis and path analysis of that. And so we found sort of two important things. One was video game use related to proportion correct, and then another one was this evenness of exploration, how evenly you explored in the environment. And they're all related to each other. But when we put them together in the model, basically video games dropped out. So it was really about the evenness of exploration that was more informative than the video game use. So one question, of course, is, you know, the causality of all these things. So that's something we want to look at more in detail in the future. And that's another reason to get away from desktop stuff, because we want to be able to have our more naturalistic walking environments. There's still going to be these differences, though. So even in things that they're very just like walked from one place to another, we see these big sex differences. And so understanding that relationship is going to be critical.
Thanks. Go ahead. My name is Kritika Venkataraman, and I'm the scientific lead for the Simon's Collaboration on Plasticity in the Aging Brain. So, hi, Lisa and Beth. My question is for the both of you, and I wanted to know if there are any sex differences in what you see, or if you've had a chance to look at sex differences. I think you mentioned, Lisa, that you had.
Yeah.
And then the second part of that is, if you are looking at females at all, are you seeing any estrous cycle or menstrual cycle dependent changes or social environment dependent changes? Like how are your animals housed, and do you think that makes a difference with age?
Yeah, I'll go first. We haven't seen any sex differences in terms of neural coding or behavior. There's one small difference. I can't remember what it is now, but it's like on one particular version of the task, the males tend to perform a little bit better with age, but generally speaking, we haven't seen any sex differences. For housing, those are really interesting questions, and those are the kinds of things we'd like to think about in the future. We don't have the estrus cycle data for the data I showed. And then we do run the mice at the same time of the day, though, so that's good. But in terms of housing, our animals are all housed alone once they're assigned to like be on an, because they have the head bar and things like that. But I do think that there could be some really interesting interactions with other types of components that we could explore in the future. They also are exercised mice, they're already at like an exercise baseline, because we have to give them running wheels, or else that's just like they won't run. So they're already fit.
And I could say, especially after all the talk we heard today, I think the sex differences question is such an important one, and we don't have a good answer yet. We have such small N sizes for our cohorts, and it turns out that most of the only age monkeys that we've gotten so far are all female. They're typically retired breeders, and so we've only worked with aged female monkeys. In terms of the housing, we work really hard to try to pair house them, at least, to have them with a social partner. Not all aged monkeys are amenable to that. Sometimes they like to be by themselves. So there's a mix depending on the preference of the monkey.
That leads into the question I was going to ask specifically about primates because of the social environment they are raised in. It's similar to us. So that kind of hierarchy among gendered groups matters. maybe for the others that didn't get to talk, can you imagine or talk about how some of your results might not be clear if they're really sex differences, or there might be really gender differences? Because I'm thinking about the test you shared about women being more conservative in their approach. I'm thinking about safety and all of the experiences we have for sexism, what that might mean for residential segregation and women who are curtailing the way that they move based on fear of police. Or contact with males. So maybe talk about some of the results you think might differ by structural stuff.
So that's certainly something we think is really important. So these social determinants of some sex differences is one thing that I didn't mention that we are collecting in a lot of stuff is some of the anxiety measures, but not just general anxiety, but spatial anxiety specifically. So thinking about safety, especially for women, thinking about the fear of getting lost and why that might be different, or just in general, people might have different tolerances for getting lost across individuals, but it also might very much be different by sex. And then thinking also about lifetime experiences. So we've actually developed a questionnaire about spatial activities. So thinking about things that you might have done. So boys tend to be allowed to maybe run around more than girls do. And then, did you go hiking a lot? Did you go running? Could you do those things, right? Or things like, small scale things like drawing, computer programming, all of the kinds of things that might involve different levels of spatial activity from sort of different types of spatial activities to kind of get a sense for your experiences. And so we've developed that and have validated it. So we're starting to use that in our things as well.
I want to just comment on some of the non-human primates that we use at the primate centers come from these large pens. And I know that some of the monkeys that I've been involved with testing, the female is definitely the dominant, the matriarch of this whole, 200 monkey pen. And her behavior was really quite striking, but still showed memory changes with age, but her behavior just cooperating or not wanting to cooperate, or it was quite interesting. And also, we try to opposite sex pair house our males have to have vasectomies, but that tends to really work well, even when they're implanted. So we do make.
Yeah, I'll just add one other anecdote about the non-human primates. One thing we discovered too, a lot of this work really requires a collaboration, I think, with the monkey, because these are complicated things that they're doing, and we really need them to feel comfortable and secure with the environment. And we've noticed that the monkeys that were housed if there were two sort of heights, and they were housed in the lower cages, they were not as excited about doing the tasks. And so we've just gone to having no monkeys on the bottom shelves. That just, so now they're sort of multi-layer things, and the monkeys can move around. And it just felt like they were, feeling, I'm anthropomorphizing, but there was something about that setup in the room being lower that was not working for them.
We have a question. Oh, Molly first? Okay. I'm not sure who was first, sorry.
You were first. Go ahead.
Thank you. I'm Alan Gow from Hertford University in Edinburgh. I wonder if any of the panel want to comment on the kinds of things we'll often see in the press when we talk about navigation, and that's the fear that younger generations have their map in their pocket, so they're useless at basically any task that involves. Now, we see that for everything, don't we? Whether it's spatial navigation or information looking up or whatever. But I wonder if you want to comment on what should we be thinking about with children and young people in terms of training spatial navigation, and is it a fear that we should have in the sense of what that might look like in generations to come if they don't have the same kinds of spatial skills, spatial training that maybe their grandparents' generation might have had? So a bit of an open question, but hopefully someone will have something interesting to myth bust for us.
I guess I'll just start by saying I do think that most of the systems that I described are have been studied under local navigation conditions. So usually mice are in a 1 by 1 meter box, which to us, would be like this room. And I at least think we haven't lost the ability to navigate local spaces. I hope no one's using like Google Maps to get through this room. So I do think that at least some of the spatial mapping system can be very, very useful and well trained in local environments. I do agree for more global navigation, moving from here to a restaurant or something, I might rely more on my phone, and then I'm not really building a spatial map of that environment. In humans as well, I think the systems are also used. There's been really beautiful, this isn't our work, but really beautiful human work showing that the grid cell system can also be leveraged for things like semantic tasks or knowing where you are in task space. So I do still think that those are systems being used when we do other kinds of things. I don't, the degree to which rodents do that is a little bit less certain, but yeah.
Okay, Molly, I think you're next. Sure.
Well, this is actually a question I asked of Janine when we were having some pre-meetings, and you presented this data, but I was just going to ask you to expand on it or at least comment on this to the audience, because I found it very interesting. It seemed like an almost paradoxical finding. So in your animals, you saw that during the light cycle for a rodent, which happens to be, the daytime, let's put it like that for the rodent, that happens to be their sleep time, okay? But you saw improved cognition during that time. And so I just wondered if you would expand on it a little bit for the audience, because I found your explanation to me when we were talking earlier quite interesting.
Oh, good. Yeah, we were surprised to see that, honestly. So we see better memory during the daytime when these mice are normally asleep. I will say it's not just mice that do this. Drosophila also show the best memory during their inactive phase too. So it really seems to depend on the model organism. And what it suggests to us is that the activity pattern of the animal is simply not informative about when they're worse. It's probably something happening at a molecular level like PER1 or something at a cellular level like SCN activity. The central clock itself is actually more active during the day in both nocturnal and diurnal animals. So it could be something happening at a cellular level. At like an evolutionary perspective, it's a little bit hand-wavy, but we're wondering if perhaps mice show better memory during the day when they're, most vulnerable because their predators are out, their prey, they need to remember things happening when they're really vulnerable, whereas, humans, we are, less vulnerable, I think, while we're asleep, perhaps, so we have better memory during the day. We're primed for that. We're not sure exactly why, but we reliably see this every time. I only showed like one example here, but we've replicated this and we see it for multiple tasks. It's not just object location memory. When we do fear conditioning, we see better memory during the day than during the night. So it's definitely reliable, and yeah, hopefully we'll have a better answer for exactly why it's happening soon. Thanks, Molly.
Caleb Finch, USC. Comment about the socioeconomic gradient, which was implied with the many discussions this morning and this afternoon. I would like to point out that the full range of this is 15 plus years in lifespan between the highest, most educated and the most impoverished. And this is observed, the scale of this 12 to 16 years is observed across industrial countries. For example, Finland, which has much more homogeneous population, the UK a little less, ours which is quite heterogeneous. Not only is the lifespan 15 plus years different, the entire survival curve is shifted left or right. And this is on a gradient that is seen linear, whether it's measured as quartiles or deciles. And there's some biology there that we simply don't know about that may start prenatally with the exposure from a mother who is working 1 or 2 jobs until just before labor and can't prepare fresh food, and there's higher exposure to pollution and secondhand smoke. So these effects are larger than the ApoE alleles. And on that, I would like to just recall that ApoE is part of a multi-gene complex that includes TOM40, which was the second Alzheimer's gene, which is a neighboring gene, and 8 or 10 other genes which do many of the same things that ApoE and TOM40 do in regulating blood lipids, metabolism, reproduction, and resistance to viruses. So I think the ApoE discussion in due course will expand to consider ApoE haplotypes across the entire ApoE complex region. That's for the next 5-10 years.
Thanks for those comments. Anybody want to respond?
I think that's just a really important point to think about how the diversity of our sample. So we see, most of our work is in, healthy, young, sort of college-age cohorts, and our midlife adults also tend to be pretty well educated and somewhat higher SES. So I think getting a broader spectrum, first of all, across the lifespan and not just chunking by group is going to be important. And then thinking about it, then, intersectionality with all these other factors is going to be super important, and expanding, again, with some differences in terms of, but also TOM40 and others are expressed differently across different groups. I think it's going to be critical to sort of understanding these really broad characterizations of deep phenotyping of people, to really think about all the possible contributions. And we have to sort of make some choices in there. But that's really, I think, the take-home message from a lot of what we've seen today is thinking about how all these things are impacting us, and how we can sort of put it together in the best way.
John?
Hi, and just briefly, for Dr. Krastel, you used the FNAME task, also known as the face name memory task, which is available in the NIH Toolbox version 3. And as you may have noticed from the TV out there, there's the NIH Toolbox conference next month that you can all attend on April 8th and 9th. Not to mention its inclusion in the mobile toolbox effort, which will be available widely this summer, which will allow you to administer all these tests on mobile devices, iOS and Android, which I think is really important. But past the commercial part, you use that as it went by fast, as a covariate in the maze task?
Yeah, we did use it as a covariate. We also have done the analysis without it as a covariate, and it's pretty similar. It kind of cleans it up a little bit, partially because we did want to look particularly with the maze task, because there's other, you can do this task in a lot of different ways. Yeah. And that's part of the strategy question. And so we really wanted to see what was more spatially specific rather than sort of an episodic memory general thing. So sort of at least tease it apart a little bit. Even though, of course, they're strongly related. And so that's also this big factor. And so when we look at this, we have this like an even larger young adult thing where they do like 12 hours of testing and fMRI stuff, all this stuff. So to try to really piece apart the various non-spatial cognitive factors with the other ones to kind of think about how this all fits together.
Well, okay, so I guess the question I had is whether or not you had men's performance on NAME being actually worse than women's, which is what I think you look at. Yeah,
I don't know. Actually, I have to look at the data. So we have it, but I just don't know.
Oh, okay, I'd be curious to know, because that would be interesting, because to the extent that men are less good at that, and they seem to be, then by adjusting for that, you're going to heighten sex.
Yeah, I think that's a really important point. Thank you.
Which is really cool.
So, John, if you mean partially just associative memory, Matt Huntelman has, like 100,000 people from 18 to over 90, and at every age, women are better at associative memory than are men, but at every age from 18 to 90 as well, men are faster. They show faster speeds of responding than women. So there are going to be real differences that we don't really understand, but some of the, profound differences are not going to arise unless you have really large cohorts. And you do need the whole lifespan to get the perspective on what's really going on.
Oh, no, I agree. And that was, I guess, sort of what I was wondering about, is because that task was designed to be essentially like super sensitive to associative memory in the hippocampus. It was I was just wondering if it was, this is the best one to use. But you still need big sample sizes, and what Matt's project scientist for that project too. So
yeah. You know about this.
Yeah, but I think it really is quite interesting, because to the extent that we can get some of these tasks either in real life or down to something that you can administer in very large cohorts.
On the web, fast, etc.
Very compelling, yeah.
Huge. Yeah. I agree. Any other comments? I was going to ask if, one of our goals in this particular group, or one of our objectives, was to be able to cross levels of species so that we could get deeper and deeper understanding at a reductionist level, but we know that we have to go back to the whole network. Do you have any comments about how we can get there? I mean, the critical factor, like Janine, you brought up the circadian cycle, and when you test, there's so many people who do not test their animals, say, at the same time of day, and maybe even humans. So it's just so critical to be able to go back and forth and to use tasks that aren't verbal so that we can go across species as well. So any comments there?
I was just going to say that in addition to that, also like having a report of what time of day you're running your animals or your subjects is really important, because a lot of times it'll just say like the light cycle or not even that at all. So I think those things are really important to disclose when you're writing your paper.
And what about analytic difficulties in these huge populations of cells and across the brain analysis and so on? Anybody want to comment on how they're solving this problem? Because we're all facing it.
Yeah, I mean, I think that's just a general problem. We generate now, terabytes of data every day. And so figuring out what metadata needs to be saved so that the data sets that we do share are useful. The infrastructure for doing that and ways to organize it in a way that is useful in the longer term, I think is just a general challenge of science that's going to have to be addressed.
And I'll just add, I think specific to non-human primates, again, our cohorts are so small that really being able to pool data across studies and across labs is going to be really critical. And so having some standardized way of collecting the metadata, sharing it, storing it for reuse is going to be really important.
One of the results of the last Cognitive Aging Summit was this Resilience and Reserve Collaboratory that Jakob Stern organized. And one of the real successes of that is getting people who use animals to actually talk to each other about how are we going to get all these data that we collect that we don't share normally into some sort of database that we can share. I think we've been somewhat successful in rodents, but I think the non-human primate would be a challenge that we should directly address if we can. Okay, any questions to wind up here? We have 26 seconds. John?
Actually, it's 1:12. But no, Molly just that the reception, which was scheduled for 6, will actually happen at 6, which is to say in 12-ish minutes, or maybe right now if you get out fast enough. But after this, and again, it's from 6 to 7:30. It's a great opportunity to ask the questions you didn't ask and meet the people you didn't meet. But also, if it would be possible for those of us who are session chairs to meet with Team NIA very briefly to figure out how we're going to assemble the gaps and opportunities, that would be great. But it's been a wonderful day one. Thank you very much, and see you outside.